Hi, I'm Amy Proal, and I'm very excited to welcome you to the PolyBio Spring 2026 Symposium. And just a quick note on the format of the symposium, there's a question and answer session at the end of every hour. And to maximize how that works, if you're in the audience, post your questions in the chat throughout the symposium, and then in those 10-minute Q&A periods, I'll direct your questions to the relevant speakers. Okay. Moving forward, there's a lot of momentum in the long COVID and related infection-associated chronic illness space on identifying different drivers of disease, and you'll hear many interesting talks on that today. So, for example, you'll hear people talk about SARS-CoV-2 persistence in tissue, for example, tissue of the gut. Teams like Tim Hendricks at UCSF or Marcus Buggert at Karolinska Institute. You'll also hear groups talk about the reactivation of previously latent pathogens. These include the herpes viruses, but we're also looking closely at the chronic tick-borne vector-borne pathogens in that capacity as well. You'll also hear groups like Mike VanElzakker's lab and his postdoc, Mario Murakami, talk about imaging studies that are finding neuroinflammation, some signs of vagus nerve dysfunction or cerebral spinal fluid flow issues in patients with long COVID. Also, teams talking about immune dysregulation, like Nadia Roan. And, for example, Henry Van Brocklin at UCSF will talk about imaging that can identify T-cell activation throughout the body and brain of long COVID patients. There's also the endothelial dysfunction, the coagulation issues, the gut barrier permeability issues, metabolic dysfunction, and a lot of different things that will come up today. But I want you to keep one central idea in mind while you're watching the talks, which is that a lot of the teams that are finding the strongest signal of disease or dysfunction in long COVID or ME/CFS or related, even in chronic Lyme samples, are doing that because they're the teams collecting tissue samples from patients with the conditions via biopsy procedures or surgical procedures. Or some of the teams using those high-resolution imaging methods that I mentioned before that can identify immune activation or other parameters, again, throughout a patient's entire body and brain, which is the tissues of their body. And understanding that that's where a lot of the signal is being found, many of us in the long COVID space are starting to look at long COVID as a largely tissue-based disease. And what I mean by that is not everything in long COVID is only happening in tissue, but there's strong signal in tissue with sometimes variable detection of those factors that could be dysregulated in accessible fluids like blood. So it's important to keep that in mind. And the truth is that as we study patients with chronic Lyme disease and ME/CFS, there's also a similar trend emerging there, which is that throughout the tissue of the body and brain, we can find a lot of the major dysfunction. Honestly, in my opinion, this trend is one of the reasons why patients are most misunderstood when they go to a doctor is because, and we've known this for so long, standard blood tests not only sometimes are not innovative in the methods they're using to find dysfunction in people, but they're even looking at a body fluid that might not contain important parts of the disease process that are in the tissue of the patient that the doctor just doesn't know about. That can lead to dismissal or gaslighting or things that are very problematic. So to address this, we're continuing the tissue and other work, but we also, and everyone knows this, we need diagnostic tests right now in long COVID and related infection-associated chronic conditions. And these tests have to be able to be run to a large extent on the accessible fluids collected from a person at a clinic, like blood, saliva, or urine. And so a big thing we've been doing, and you'll hear speakers talk about this today, is working with teams to maximize the development, iteration, and validation of technology platforms that can capture tissue-based drivers of disease in accessible fluids as long as you have the right innovative methods. So, for example, you'll hear Alessandro Lucchini from George Mason and Eduardo Retigui from Ohio State talk today about platforms that can capture the Borrelia pathogen, the Lyme pathogen, or SARS-CoV-2 as a virus in extracellular vesicles derived from tissue and fluids. This is a big trend, honestly pulled sometimes from the cancer space with innovation, and it's important. Okay, so then to maximize the ability to both understand, iterate, and validate those diagnostics and other diagnostic tests that measure other drivers of long COVID disease, including metabolic markers or even eventually autoimmune diagnostics, we created a program called VIPER. It's based out of UCSF, but samples are sent to dozens of other teams who have promising diagnostic platforms. And it's a program to identify and also validate the top long COVID diagnostic test. We're very excited that the program is now fully underway out of UCSF. Thanks to generous donors, we were able to get $8 million to begin it in full force. And key is that both tissue samples and accessible fluid samples are collected on participants in VIPER, so we can compare diagnostic tests in those fluids to tissue-based drivers of disease, which is really huge. So you'll hear Michael Peluso talk more about the program later today. We have a scientific advisory board now for VIPER. It's a stellar group of 5 people, another thing that's moving forward in a really great way. So this is the thing, though, anticipating already the results of VIPER, anticipating some of the results from other PolyBio programs that have great promise. We definitely want good academic papers. So we're hoping that we have great papers in Nature or other journals that have, amazing data presented on what works in programs like VIPER. But we don't just want the academic papers. We want real-world uptake of the diagnostic test that VIPER helps to validate and define as meaningful in the space. And so anticipating that very early in the game, our PolyBio team built a program called the Long COVID Cure Initiative. And it has 4 steps. The first step is to validate the top diagnostic test in long COVID via the VIPER program. Then, however, we've created a commercialization advisory network to help make sure that those diagnostic tests can actually start to be maximized in the real world. Then we're gradually building a clinical trials network where we can strategically position in the tests that work along with the drugs that most make sense to target the problem that the diagnostic measures in patients with long COVID. And then finally, we're building what works into a CME accredited medical education program that can educate thousands of physicians on what works from the rest of the steps. Okay, so the commercialization advisory network, we're really excited about this. We spent the last year forming this group. We've networked with people, first of all, from different foundations, from the American Cancer Society, from the Michael J. Fox Foundation, from the Multiple Myeloma Foundation, all foundations that help support not just academic research, but also have incubator programs that help make sure that findings that they support in academic labs that are promising can get, and the teams there can get guidance from people in the real world on how to commercialize the projects and make them exist. So our group, for example, now has people with years of experience on diagnostic manufacturing and scaling in the real world. These are even contacts at labs already, like LabCorp or Quest, that really understand what's needed to make these tests fully happen. We have people in the network with substantial regulatory experience. Whenever you're moving a diagnostic test forward, you have to choose an FDA approval path, and that's a big decision. You need to know what you're doing, so we have people who can guide teams on that front. We also have groups who can work with people who have promising tests very early on to maximize the strategy for insurance coverage of the diagnostic test, which is key to making it actually succeed in the real world. We also have people from the pharmaceutical industry in the group. They're helping to strategize, again, how these tests can be best positioned as companion diagnostics with treatments that make a lot of sense. This also maximizes their uptake because an MD in a clinic, when they order a diagnostic test, they want to know what to do if the test is, positive or negative, shows problems. They want to know what treatment to order. So that companion diagnostic dynamic is very good. And we held our first meeting on May 15th of this group. This was about a week ago at Harvard Innovation Labs in Boston, and it's very good. These are members of the group, so you can see this is now on our website. And like I said before, we're also building this trials network gradually. Key to the network is we're going to have a very good registry of long COVID patients who can sign up to show they're interested in trials in different geographic locations across the country. This will incentivize the pharmaceutical industry to run trials because they'll easily be able to know that they can recruit a lot of patients at a lot of sites if they want to move their drug into the space. Like I said, we're building out the medical education that can help make that work. And then finally, it's important to emphasize that the Long COVID Cure Initiative is not a one-off program that ends with Long COVID or the SARS-CoV-2 virus. We're already beginning the process of taking this model, validate diagnostics, help incubate them to get commercialized, use the diagnostics to guide trials, and maximize the medical education in the space into chronic Lyme disease, ME/CFS, even conditions tied to viruses like Epstein-Barr virus, like MS, and even into the health span extension community, where people are increasingly realizing that having pathogens or problems in their bodies over time is not a great situation. Okay, so those are our plans. With that in mind, I want to introduce the first speaker, who is Resia Pretorius. She's a distinguished professor at Stellenbosch University. Thanks, Resia.
Hello, everyone. Again, it is a huge pleasure to share our latest research and developments with you. I wish to focus on 6 of our recent published and in-print manuscripts. We have 2 framework review papers and 4 laboratory papers involving method development. Lastly, I will say something very quick about our collaboration with Michael VanElzakker and the LINK cohort. As always, we've been looking at various components of pathology, including endothelial dysfunction, platelet hyperactivation, net release, and a hypercoagulable amyloid microclot state. In all the data discussed here today, you will see glimpses of research focusing on these specific pathologies. I start with our recent paper, the Fahy et al. Review in Communications Medicine, a 20-author interdisciplinary review. In this paper, we frame long COVID as the intersection of 8 mechanistic domains: immune dysregulation, viral persistence, autonomic dysfunction, microvascular pathology, mitochondrial dysfunction, neuroinflammation, gut vascular dysbiosis, and the glaring diagnostic gap. The cost in numbers: 400 million affected, over $1 trillion per year. And this is why this matters as a policy at a policy level too. This paper positions microclots as a microvascular endpoint of multiple upstream insults, directly motivating the work that we've done on proteomics that follows. Paper 2 is our cell death and disease paper currently in press. This paper provides the mechanistic framework we have needed. It proposes that we use the term fibrinolytic microclot complexes, or FMCs, and that there are different FMC phenotypes. These are complexes resulting from cell debris that form prothrombotic seeding areas. Another type of complex is fibrinolytic microclot immune complexes that contain neutrophil extracellular traps, or nets, seen in the micrograph labeled E. Our third type of misfolded fibrinogen is where spike or other amyloidogenic inflammatory molecules have had protein-protein interactions, resulting in fibrinogen to become misfolded. And lastly, another type of complex is amyloidogenic protein aggregates that seed onto intact cells, seen here in micrograph labeled F. This is a high magnification of a red blood cell with microclot deposits on its membrane. In this paper, we specifically focus on the mechanism or formation of these 4 microclot complexes because every step is a potential drug target. We suggest that the first events are upstream insults like spike, ROS, hyperglycemia, or viral persistence, producing circulating inflammatory molecules that stresses the cell membrane. And then these membranes are directly binding to these different inflammatory molecules, exposing a catalytic and prothrombotic surface. This is called the PS, or phosphatidylserine flip, on the membrane, so that the intact membranes become pathological and flip outwards and inwards. This allows for coagulation cascade assembly, including fibrinogen, that can bind on these prothrombotic surfaces. The cell membrane disintegrates and is then in circulation. This may include membranes from platelets, endothelial cells, red blood cells, and neutrophils. Typically, the immune cells will get rid of these damaged debris, but we now know that in post-viral diseases, the immune system is sometimes muted. So these prothrombotic and damaged microclot seeding areas are not adequately removed. In parallel, amyloidogenic inflammatory molecules may co-aggregate into insoluble complexes. that also, as I mentioned, entrap nets. Each may result in an actionable research line for targeting therapeutics in future. The next manuscript I wish to highlight is with first author, my postdoc Claudia Christovitz. It is currently under review. This was an experiment that we wanted to produce. Claudia exposed human umbilical vein endothelial cells, or UVECs, to long COVID platelet-poor plasma. Plasma we know that contain microclots, and also to control plasma with much less microclots. We used healthy UVECs and also UVECs that is in rest or in senescence. Previously, we have discussed the physiological processes of endothelial senescence in post-viral diseases. Long COVID plasma significantly reduced intracellular von Willebrand factor, indicating endothelial activation, also reduced syndican signal, indicating glycocalyx degradation. We also saw significant morphological changes. Then we also looped in DMSO that kills cells. Plasma from long COVID patients showed very similar morphologies than cells exposed to DMSO, cell death after exposure to microclots. Paper 4, the platelet monocyte aggregate paper by my student Anel Thompson, is currently under review. This is a methods development paper that has given us a quantitative accessible biomarker using two-color imaging flow cytometry of CD14 on monocytes and CD62P on platelets. We show that long COVID patients carry platelet monocyte aggregates on 29% of their monocytes versus only on 5% in controls. Long COVID was also found to have more multiple platelet binding events on single monocytes. This is important as these aggregates are also age independent in long COVID, meaning we have an objective reproducible measure of thromboinflammatory burden that can serve as a trial endpoint. The next paper I wish to highlight is from my PhD student Maxine Waters and is our post-vaccine, post-infection syndrome proteomics paper. We compared microclot proteomes with patients and with controls, and we also directly compared it with the older data set from 2022. These findings stand quite out. First, attractin, a previous unreported molecule inside digested microclots, was significantly upregulated. Second, the intrinsic pathway is prominently involved where factor 10 and 11 are up. This is distinct from long COVID's von Willebrand factor net signature. Third, platelet factor 4 and S100A9 go in opposite directions to long COVID. That last one is important to us. It means that proteomics can discriminate vaccine-related injury from infection-related illness. Exactly something that we need to look at in future and that we have struggled with. The last manuscript I wish to discuss is from my PhD student, Renata Boyens. This is the first map of post-translational modifications on protein sequence inside FMCs. Renata compared pre-COVID pots, long COVID pots and controls. The exciting finding is that it is quite counterintuitive for us that 1 in 3 proteins differ by abundance, but the cargo is essentially the same in all 3 diseases. The chemistry on these proteins is profoundly different. Long COVID FMCs show age glycation-related changes on fibrinogen, which is a diabetic pattern. Pre-COVID pots show oxidation and deamination of apolipoproteins, while long COVID pots is a hybrid. This is quite interesting, and we believe that these diseases lives in its chemistry, not the composition or regular lab tests, as Amy already said. Putting the 6 manuscripts together, 3 intervention axes, each now backed by a published biomarker and quantitative endpoint. Axis 1 is to restore fibrinolysis with microclots clearance as a measurable readout. Axis 2 is to calm platelets, monocytes, and platelet neutrophil aggregates. This is in our Thomson 2026 preprint with the platelet monocyte aggregates, which might be a trial-ready biomarker, complementing combined antiplatelet and anti-net strategies as treatment regimes. And our last axis is to address upstream chemistry. Renata Boysen's papers show glycation and oxidation as disease-defining, so glycation inhibitors and antioxidants might become stratifiable and not generic. Our current priorities is finalizing our proteomics data analysis from the LINK study. Also looking at POTS and control samples from Artur Fedorovsky at Karolinska Institute and optimizing our immune complexes detection methods. And we hope to have some good news soon regarding a clinical trial in South Africa. This intervention axis, our priorities for the next few months, is really with a deep gratitude for PolyBio Research Foundation and all the funders that support us. I'm happy to take questions afterwards. Thank you.
Thanks, Resia. That was a great overview. And I'm also so glad you're working on patients with post-vaccination syndrome in addition to long COVID. Our next speaker is Marcus Buggert. He's an associate professor and group leader in the Department of Medicine at the Karolinska Institute. He'll be talking about SARS-CoV-2 gut and lymphoid pathology plus a longitudinal long COVID study.
Perfect. All right. Thank you very much, Amy, and thank you for the invitation. As Amy just said, I'm going to talk about two studies. For today, I'm going to first actually talk about this longitudinal study, just very much in brief, that we're working on right now. And then I'm going to briefly go through some new data that we're working on when it comes to our SARS-CoV-2 gut and lymphoid pathology work. So at the Karolinska and at COVM cohorts, these are two separate cohorts that we're working on here in Sweden, which I'm going to show some data from today. We have almost 800 patients. We have longitudinal disease data from these donors, disease data which are in different scale scores, some of them highlighted down here, where we also collect longitudinal serum plasma sample from most participants and also PBNC in selected subgroups. So at this point, what we have been able to do, and this was particularly in collaboration with David Putrino from the UK as well, is to identify inflammatory signatures in long COVID. And we're particularly able in this paper to identify them to individuals with different type of respiratory problems, particularly individuals with breathlessness, severe issues. And this is just showing some different proteins which are upregulated using Olink biomarker panels. And what we essentially were able to do in this paper were to identify specific networks which were elevated, and they were centered around pro-inflammatory signatures and also caspase activating factors. But one of the questions that we have had is that this was all cross-sectional 2 years after the infection, but when do these inflammatory signatures then sort of emerge? And in order to do that, what we've been doing is to work together with other researchers in Sweden from Umeå and Örebro, where we have been following 171 donors. We have taken samples from them, or they have taken samples from them at 4 different time points during acute infection, 6 months after, 1 year after, and 2 years afterwards. So in total, we have 669 different samples where we've been running different type of assays on it. And I'm going to show only Olink data here today. And what we have been doing is to sort of identify different clusters of patients. Essentially, individuals in cluster 1 with very few symptoms afterwards, so they have no long COVID symptoms at visit 1, 6 months afterwards. And then we have these two different set of clusters, where particularly this fourth one is interesting, with more multi-system symptoms and more severe long COVID. So if we look into pro-inflammatory signatures and the top 30 proteins by looking into thousands of different proteins, it's not very surprising that if we look at here the red, every point here is a patient. The first visit here is now in red. We can see clearly that visit 0, that's acute infection. You can see that clearly there is differences when it comes to pro-inflammatory molecules. But the question we were asking is, how are these now persisting afterwards? So if we just take these 30 proteins and we look at module scores for them, and this module now contains IL-6, TNF, 6L8, IL-32, classical inflammatory molecules. What we can see essentially is that they certainly go down in most individuals, but they sort of stay up and they almost plateau already after 6 months, and they stay sort of elevated, and particularly in these individuals which have multiple symptoms after clearance of, or after potential clearance at least of the virus. So essentially what this shows here is that this onset of it happens quite early. You can see that there's elevated early on, it goes down, but it doesn't really go down to levels where individuals having fewer type of symptoms afterwards. So we can essentially identify that now, that it happens quite early. And what we're trying to do now in this course is to understand what happens when people become better and when they don't become better, to fully understand if we can identify some key biomarkers in the plasma. But one important question, and this is a center phenomena of the work that we're doing in my lab, is to not only look into blood, because not all individuals show increased inflammatory signatures. So could it be so that some of these signatures, they still exist, but they are compartmentalized? They're present still in tissues. And that's the reason why we have these sort of three subgroups of individuals that we're looking into. And what I'm going to focus on today is work that we're doing on colonoscopies, where we basically are taking samples from different sites of the gut. I don't have to go into great detail on why this is important. I think everyone is well aware of this study and other studies in the field that SARS-CoV-2 can be present in the gut for a long time after acute viral infection. So what we did in the study, this is something I demonstrated last time I talked about it, but I will quickly just go through it again, is that we have severe long COVID patients with GI symptoms. They've had this now for more than 4 years. They have IBS-like symptoms, abdominal pain, bloating, cramping, diarrhea, but we can't find any macroscopical findings that is linked to bowel disease. When we take biopsies from cecum, ileum, sigmoid, as well as blood in 10 long COVID patients and 7 healthy controls. And essentially, the work I'm going to focus on today is the single cell work, although we do a lot of other type of work as well to look into other aspects of the pathogenesis in these individuals. So this is data, again, some data that is quite similar to what I've been showing to some of you before. And one of these distinctions that we do find, if we just now focused in on different clusters of immune cells by the single cell data, this is very deep sequencing. I will say as well, over 50,000 reads basically per cell that we're doing. But essentially what we can see is that the interferon stimulated genes, ISGs, are broadly upregulated in long COVID. We can see it's primarily elevated in adaptive immune cells, which sort of suggests that maybe there's an upstream or extrinsic signatures that could drive these features, but it could also very well be some extrinsic signatures as well, which I will touch upon soon. And essentially what we can do now is that we can basically score them based on an ISG score, and we can then do that across cecum, ileum, and sigmoid, and we can more or less almost, like here in this case of sigmoid, almost identify that all individuals have a higher elevated ISG signature in comparison to what the median values are usually for normal healthy controls. But in addition to that, we also see strong pro-inflammatory signatures for other type of cytokines. We see increased TNF signaling, and that means that elevated baseline expression of TNF is across multiple different T cell subsets, but also germinal center B cells are elevated. But we also see that some features are downregulated. So it's not only one sided story, things are also downregulated. So we see that there's reduced baseline TGF-beta expression across multiple T and B cell subsets, and there's other molecules which are linked to regulatory features such as furin, and as well as other factors which are also downregulated in long COVID patients. So finally, what may cause then these changes? And one of the ideas that we had now, given the fact that we do have so much single cell data, were to use metagenomic analysis with a tool called Kraken-2. So based on our single cell RNA sequencing data, we can then use Kraken-2 to taxonomically sort of identify and classify non-human sequencing reads and identify microbial signatures that includes viruses, bacteria, fungi, and parasites within single cells. And as I mentioned to you before, I mean, it would not be very surprising if we would potentially find SARS-CoV-2 in the gut, given the fact that people have identified that in the past. And indeed, when we look into, in an unbiased way, with Kraken-2 counts per cell for SARS-CoV-2 reads, we can't determine yet if that is SARS-CoV-2 1 or SARS-CoV-2 or 1, but most likely it was a SARS-CoV-2. But what we can see essentially that the vast majority of reads that we can sort of align to any virus is coming from SARS-CoV-2. And even if this is very preliminary data, I felt anyway that I wanted to sort of show at least that what we can see then is that these reads are particularly elevated in single cells in long COVID patients. We have some donors with very high recounts, while the healthy donors in general have lower levels. It's not so that all individuals have higher levels, some have lower, but in general, tendencies are that is increased levels in these donors as well. So that's very preliminary data, but so far what the data shows in summary is that longitudinal plasma inflammatory signatures, it changed over time, as I showed in the beginning, but it remain persistent in individuals with multiple long COVID symptoms. That seems to be fairly clear in now a couple of different cohorts that we've been studying, both in Sweden and the UK. We see an absence of strong systemic inflammatory signals does not necessarily reflect resolution within tissues. And that's something that we know from other diseases, particularly autoimmune conditions, but I think is an important context to put into this disease as well. Because in deep tissue profiling of the intestinal compartment, it really reveals persistent immune dysregulation, which is characterized by interferon, TNF-associated inflammatory programs, and we see the reduced regulatory signals, which includes TGF-beta. We do see increased SARS-CoV-2 associated reads that remains detectable, and particularly within long COVID gut samples. And together, these findings are supported model where compartmentalized tissue pathology could persist despite limited signals in peripheral blood. So with that, I'm done. Particularly thank you to people in my group and of course PolyBio as well as some of our funders. And with that, I'm happy to end here. Thank you.
Amazing, Marcus. That was a great overview. And it does verify my slogan that the issue is in tissue. You're finding things along those lines for sure. Okay.
Yes.
All right, I'll introduce our next presenter, who's Peter Brodin. He's a professor in the Department of Women and Children's Health at the Karolinska Institute, also the Garfield Weston Chair of Neonatal and a professor of pediatric immunology at Imperial College London. He'll be talking about whole genome sequencing and immune profiling in long COVID.
Thanks so much, Amy. Thanks for the amazing work that you guys are all doing at PolyBio. It's really incredible. And I love the new initiatives you were talking about, pushing towards diagnostics and treatment. That's really important. So yes, I am going to give you a very brief update on this because this is very much work in progress and I don't want to overpromise. So here we are. So this is a continuation of a collaboration that's been running since 2020, basically, where these clinical colleagues of mine established a phenomenal cohort and did really deep functional and physiological assessment of patients presenting with severe long COVID. And this is the basis for the types of analysis we're able to do, because if we don't have very careful assessment of the patients and their symptoms, everything else becomes impossible to interpret. So I want to acknowledge that again, and those of you who've seen me present before know that this is a long-standing collaboration. So this cohort, which includes now about 200 or so severe long COVID patients, the first thing that this clinical team did was to assess how the different phenotypes, POTS, autonomic dysregulation, various neurological symptoms, abnormal breathing patterns, and things like this, which are all objective for some symptoms that we can objectively verify, which is another important aspect of this. How they vary between patients. And as you can see through this, there's no obvious clustering of patients. I think other cohorts have seen similar things, that there are no obvious groupings that patients with POTS always have something else or vice versa. So it's really a heterogeneous population. And I think that's really important to keep in mind when thinking about things like genetics as an underpinning for this disease. And so we're doing all of this work within the framework of this very large international consortium called the COVID Human Genetic Effort, which is basically a group of individual researchers who study patients with rare presentations prior to COVID, which includes unexpectedly severe infections due to immunodeficiencies, or, as in my case, immune dysregulation in patients with rare, let's say, monogenic diseases of their immune system. And so when we then started to hear about patients with long COVID or post-infectious disease, the presentation, we thought we can use the same logic to try to understand these conditions and treat long COVID as another unusual, even though I know many people on this call now don't consider it unusual, and neither do I. But still, the logic is to study patients with an unexpectedly severe form of infectious disease response to infection. And so we do this by sequencing the genome of these patients, which is the start, to try to understand if there are any obvious variants that we either know or that we can try to understand because we don't know them, that are enriched in the patients as compared to the healthy population or people without long COVID. And then obviously, like everyone else in this call who study different patient cohorts, we look for things like T-cell responses, cytokine levels, and circulating viral RNA or protein. And so this is what I've already said, that whenever a new infection hits, there will be patients that have an unusually or unexpectedly severe form of disease. There might be some patients that are resistant. I'm not going to talk about that today, but there are people trying to understand whether there are genetic variants that can also pose resistance to infection. And as many of you know, during the pandemic, one of the first things that came out of this large consortium network was an understanding that the really important factor in limiting the severity of SARS-CoV-2 infections already in the naive population without vaccines is the type 1 interferon pathway. And so sensing the viral RNA, responding to it, producing type 1 interferons is the most important factor. And the reason that was found was in two ways. One, by finding genetic variants in either of these sensors or the signaling pathways downstream, which then led to really severe life-threatening SARS-CoV-2 acute infections. Or in the other fact, or the other sort of branch, was an unexpected finding that patients with severe SARS-CoV-2 infection had autoantibodies that neutralize their own cytokines, type 1 interferons, also leading to severe disease. So it was tempting to think when we started looking into patients with severe long COVID, is there any parallel here? Is there any reason to believe that these same pathways are dysregulated or dysfunctional in patients with long COVID, even though they did not present with severe acute infection? And so we started out looking at autoantibodies to type 1 interferons, as they have been so broadly reported by many cohorts in acute severe SARS-CoV-2 infections. And the notion was that if you would have your autoantibodies to type 1 interferons present prior to SARS-CoV-2 infection, then perhaps you would end up with severe acute infection. However, if you develop your autoantibodies as a part of your immune response to that first infection, then maybe you're more likely to not have severe acute infection, but rather viral persistence as a consequence of failing to keep that type 1 interferon response going over time. Well, it turned out when we started looking at these patients that that was not the case. And these are 4 different kinds of type 1 interferons. And we're looking at autoantibodies binding to these. And what we could find is that these are healthy blood donors on the left and 2 groups of long COVID patients on the right. And there's absolutely no increase in the autoantibodies to type 1 interferons in any of the long COVID patients we have looked at. And this is collaborative efforts together with Nils Landegren's lab in Uppsala. So that didn't pan out. And then what about the genetics? And so we are in the process of sequencing whole genomes of all of the patients we can. We've so far sequenced about 260 really severe long COVID patients from these cohort, both the Swedish cohort I mentioned, but also an Italian cohort. And what you're looking at here are the long COVID patients in gray and orange. These are 2 different cohorts, one Italian in orange and a Swedish one in gray. And then in blue in the background are patients with SARS-CoV-2 infection, but no, and they can be variable severity, but they have no reports of persistent symptoms following their acute infection. And as you can see, their genomes, when looking at 88,000 variants, overlap. But more interestingly is looking at genome-wide enrichment among the long COVID patients. And I am not in a position to disclose which genes we are currently focusing on, because this is work that's very much in progress, and we want to be careful that we know that we're looking at actual important variants that actually can cause disease or increase the risk of disease. But what we're finding across the genome are specific variants that are very much enriched, and they have highly likely loss of function variants in these patients with long COVID. And here are some additional examples. Many of these are highly significant. What is very interesting about these gene variants is that very few of them point towards known function in white blood cells. And so I really I want to resonate what Amy initially said, which is that looking at the pathology in long COVID, we are also turning more and more into tissues. And so the challenge that we're currently facing is to take these variants into the relevant cell type where we think they actually may exert their functions mostly. And those are things like epithelial cells of the intestine, which we are currently trying to work up with collaborators in using intestinal organoids and things like this to look at function of the barrier tissue, signaling in response to viral stimulus or TLR ligands, but also interactions with immune cells. And so this is about everything I have to share at the moment, but this is we find this to be quite promising. So thanks very much.
Great, Peter. Really interesting. Very cool that you're moving into the intestinal organoids and those other models to help kind of follow up on the initial findings. Awesome. Our next speaker then is Dominique Salmon. She's a professor of infectious diseases at the University of Paris-Cité. She'll be talking about biological analysis of platelets and plasma in long COVID patients treated with maraviroc and pravastatin.
Hello, everybody. Thank you very much for giving us the privilege to present this small pilot study. Sorry. As you may know, at least 25% of long COVID patients develop gastrointestinal symptoms with several potential causes: inflammation, histamine intolerance, autonomic dysfunction, viral persistence in some reservoirs. And our recent results with Morgan Bonsell shows that persistent virus detection in platelet is one of the hallmarks of long COVID. Moreover, the combination of maraviroc, a CCR5 antagonist, and pravastatin, a CX3CL1 pathway modulator, is known to exert anti-inflammatory effects. It was shown by Patterson, and it may target mechanism involved in long COVID. After having observed a striking improvement in one young patient who had severe gastrointestinal symptoms with this combination, our study objective was to assess whether this treatment may improve generally severe GI symptoms and also has an impact on viral persistence in platelets and the associated biological abnormalities shown by Morgan. So we set up an observational pilot study in which we included 19 long COVID patients who were very severe with multiple symptoms, among which they had severe GI symptoms that were assessed by a Likert score. They had to have at least 4 severe GI symptoms. They were treated with maraviroc with the usual dose and pravastatin once a day in real-life setting. The median follow-up was 16 months, but not less than 12 weeks. We defined the clinical success by a 30% reduction in the number of severe and very severe GI symptoms between D0 and month 3. So let's see first the clinical improvement. We observed in the majority of the patient a rapid and sustained improvement of GI symptoms. You can see, for example, it was striking for stomach cramps, diarrhea, bloating, less for some symptoms such as food intolerance that may be due to another mechanism. A total of 13 patients out of 19, that is 68%, achieves the clinical success as defined previously. We had 5 failures that were due to adverse events in 2, insufficient efficacy and stop of treatment in 2, and loss to follow-up in 1. In those who continued the treatment, the improvement continued to increase at 6 months. And interestingly, we observed 2 treatment interruptions that were planned by the patient because they had to pay the treatment. It was not easy. Those 2 interruptions led to relapse, but were reversible upon reintroduction. The patient also declares that there was an improvement for some on fatigue, on neurocognitive symptoms, and on cardiothoracic symptoms, but there was no improvement on the ENT disorder and musculoskeletal pain in this very short study. In a subset of patients who participated previously to the PolyBio platelet study, we did a sample before, we had a sample before treatment and decided to do another sample during treatment after a mean duration of 4.3 months. We assess platelet microclots by imaging cytometry. Spike protein and viral RNA in platelets also, they were quantified by imaging cytometry. Spike in plasma was quantified by very sensitive ELISA, and serotonin level in whole blood by HPLC. Let's see first the platelet macroclot results. Before treatment, we found macroclot in all 12 patients, and under treatment, there was a reduction in the size of macroclot in 7 out of 12 patients. You can see on the left 4 examples of patients that were taken by cytometry pre and under treatment. In purple, it is the labeling of the platelet by the antibody CD41, and you can see that in those in 3 out of 4 patients, there was a decrease of the size of microclot before and after treatment. And down on the bottom, it's 1 patient who didn't respond and 1 control. Then we looked for viral RNA and spike in platelets. We found both in 8 patients out of the 12 tested, and under treatment, we find a decrease in 7 out of 8 for SARS RNA in platelet, and also for spike, mainly in patients who had large platelet aggregates. And again, you can see on the right, the spike in platelets, the labeling of spike in green that decrease or disappear, and the labeling of SARS-CoV-2 RNA in those 3 patients that demonstrated a success. And finally, we also quantified spike in plasma and found that in 7 out of 12 patients, there was low level of spike. You know, it's a question of only a few picograms per ml. However, there was a tendency for a reduction in spike in plasma in 5 out of 7 patients tested. And concerning whole blood serotonin, 90% of the serotonin is in fact in platelet. Before treatment, the level was low in 6 patients and increase during the treatment in 6 patients. So that's all for this study, short study, but it's encouraging. Maraviroc and pravastatin combination seems to really improve severe GI symptoms in long COVID patients. These beneficial effects occur rapidly and seem to persist over time. Now I have patients that are treated for more than 2 years, but they cannot stop unhappily, if it works. It may have a biological impact affecting mainly the size of microclot and viral RNA in platelets. That was surprising for us, and this suggests an impact on viral persistence. And also for some patients, it increased whole blood serotonin and decreased plasma spike protein. So this approach is, of course, non-curative. Patients have many other treatments to support them, but it's clinically promising and warrants evaluation in randomized controlled trials. So our next step is to plan a multi-center randomized trial that will compare combination therapy versus maraviroc alone versus placebo or no treatment. And we have submitted this trial for support for European funding, but it's not yet required. And I would like mainly also to thank all the team of Association Espoir who did the clinical work, the team of BICET for the epidemiology and public health work, and of course the team of Morgan, who is working at Institut Cochin and CNRS, and Jacques Calabert, who did the dosage of serotonin, and of course Amy and PolyBio to allow these assessments that would not have been possible without your help. Thank you very much.
Thanks, Dominique. It's really interesting to have the data on the responders in this trial. And what's cool is we're actually running a pilot study, a trial of maraviroc at Mount Sinai as well. And we're measuring some other viral persistence markers and immune markers. And it will be really interesting to see how those might change and just compare data across the two groups. So really interesting. Thanks. Okay, let's move into the Q&A then. I have some questions in the chat. Let's see. There's one for Dr. Salmon. Does finding SARS-CoV-2 RNA in platelets prove there is live replicating virus as opposed to viral fragments? And in your study, if patients are positive for spike protein in plasma, does that correlate with having SARS-CoV-2 RNA in their platelets?
We don't know yet for those patients if there is a replicative virus, but for some patients of the Megalon study, Morgan showed that this virus, when you put this virus on viral cell, it replicates. So it seems that at least for some patients, it's possible to make it replicates, but we don't know for those specific patients in this study. And I think the correlation, it's interesting, of course, but it's really a short study. Only 19 patients. We cannot do correlation at this stage. But we will do in the Megalon study.
Makes sense. Also for you, is the reduction in microclots suspected to be primarily due to the pravastatin or a combination of both drugs? Or do you know? Or maybe you don't know.
Yeah. I think we don't know, but what I notice is that we use often pravastatin because we know that it exerts an anti-inflammatory effect on endothelial cells, but we don't observe really striking effects. So I think that alone pravastatin maybe has a small anti-inflammatory effect, but not high. And it's really the maraviroc addition that is potent. So what we don't know, is it useful to put pravastatin? So that's why in the future trials that we plan, we will have one arm with both drugs and one arm with maraviroc alone. It would be also interesting to have one arm with pravastatin alone, but it would be too many subjects.
Okay. Yeah. Got it. Okay. Resia Pretorius, a question for you. Do you think that transporting samples internationally will affect the results of microclot flow cytometry testing because some people are interested in tests that with your partner lab in Switzerland?
As long as the samples are kept cold, so at -80 storage, so blood should be taken in at your hospital or your pathology lab, then spun down, not longer than an hour or two after collection, and platelet poor plasma removed and stored immediately in storage tubes at -80. And as long as it's cold couriered to any lab without interruption, so it should not be frozen and defrosted.
Yes.
So if one can do that, then it will be perfect. That is the regular way that research samples are also shipped between labs. So it will be fine, but you cannot ship whole blood.
Yes, I agree with you. And usually for this little trial, we call the postdoc student. He comes to the clinic where I work. We sample the patient and he goes directly to the lab to observe the microclot. It was made on fresh blood. So maybe we can find technique, but it will be difficult, huh?
Yeah, I mean, that's a challenge with a lot of tests is that they need to be done on fresh samples or they need, a certain kind of preservation method. But I think, there are some strategies. That's part of why we're building our network so that there's a lot of people, and especially even in Boston where I'm based, that just that's their whole background is how to sort of, work with those considerations and try to get around them and to help people sort of scale or even automate their testing, which I know you guys have good leads on, but that's a consideration for a lot of the tests, I think, in long COVID, which makes sense. Okay, Resia, for you also, someone said a recent paper, Sanjuezca et al. Suggested COVID can trigger a new onset, hard to detect type of insulin resistance, which leads to NETosis. Could this be an important driver of the type of microclots and microvascular dysfunction that you see?
That is very interesting. And yes, I think it definitely would be the result of one of the effects of SARS-CoV. What is very interesting in our latest preprint currently under review, where we look at post-translational modifications, we find age glycation products, so end products, glycation end products, on our fibrinogen, where the fibrinogen itself is changed, and it leads to a pre-diabetic type modification that you also see pre-diabetes and diabetes. So this is quite interesting for us, and I think there's still lots of work to be done to understand exactly what type of modifications there is, but this age-like modifications that's related to insulin and pre-diabetic predispositions is very interesting for us. And I know that some clinicians have been treating long COVID, patients with just a clinician-initiated treatment regime where they give them metformin, and some individuals have had good response to metformin. And we think it might be in that subpopulation phenotype of patients that might have some sort of insulin pre-diabetic predisposition, and it might be targeting the ages in our microclots and the post-translational modifications. But this is, something that we're still working on, and we also found it in the link samples. So that is some nice information that coming through, and we shared it today with you.
Yeah, interesting. Because I know, we knew each other before COVID, even when you were actually looking already at some types of microclot formation in samples from patients with, diabetes and who can have related issues. So there is like some, connections between these different patient populations, which would make sense. Okay. I think, one person asked, I can just answer it. If people can participate in VIPER, if there would be an extended VIPER site outside the US. Right now, no. We just are, it's actually a lot to get it started just out of UCSF. The big thing is that it requires a massive infrastructure, and Michael Peluso will talk more about that later, because you're bringing in 150 patients with long COVID and then 150 control participants. And all those participants undergo gut biopsy to get tissue from the gut. And then you have those samples, the blood, the saliva, the stool, the urine collected on those participants. Obviously, we've just been discussing even the challenges of the storage protocols and all of the different ways that those samples have to be, stored and banked and everything like that. So it just takes a lot of infrastructure. That doesn't mean, though, over time, though, that we wouldn't consider something like that. We really do want patients outside the US eventually to participate in as many things that we do as possible, including trials. It's a thought that we have often. So we'll see how that goes. Okay. All right. Well, now we've reached the noon mark, so I can introduce our next speaker, who is Nicolas Hutt. He is a researcher in the lab of Michaela Trotwin-Muller at Institut Pasteur in France, and he'll be talking about SARS-CoV-2 persistence in the intestine, innate immune control of gut viral reservoirs.
Okay, so it's a real pleasure for me to have the opportunity to share our preliminary results regarding the SARS-CoV-2 persistence in the intestine and the capability of the innate immune system to control gut viral reservoir. So COVID-19 is not only a respiratory disease, but also affects other organs. There is a lot of clinical observation which shows that, in fact, there is symptom which could be linked to the gastrointestinal tract, such as diarrhea or abdominal pain. And there is a lot of study nowadays which shows that, in fact, the virus could persist in different, especially in the ileum, as shown previously by Marcus, but also by other team. And here I put a picture of the paper from Augustine, which was published this year, showing the nucleocapsid in the ileum of patients living with long COVID. So in this context, we decided to investigate the possibility that the gastrointestinal tract could be a source of SARS-CoV-2 reservoirs. So to perform this study, we use a non-human primate model, which allow a deep characterization of deep tissue as compared to human. So in our unit, we have 4 groups of monkey. All the monkey were sampled longitudinally, and we euthanasia the monkey, and at euthanasia we collect different organs such as the gastrointestinal tract, duodenum, jejunum, ileum, and colon. So for the purpose of the study, I will focus only on the group 2 monkey, which have been previously infected with the BQ1 strain, then vaccinated, and then reinfected with the GL1 strain. And then we wait for 21 weeks, and we euthanasia the monkey, and we perform some staining. But just before to go into the details, the monkey were classified into 2 groups, according to the level of viral RNA that we could detect in the bronchial lavage during the time course of infection. And as you can see on the graph, after 10 weeks after the last infection, we can start to see a difference between the group of monkey. Some monkeys start to control the virus, while others do not control the virus. The monkey which do not control the virus was classified as persistent, and monkey which resolved the infection were classified as resolved monkeys. To perform the characterization of the gastrointestinal tract, we at the euthanasia, we use a 1 centimeter gut segment, which was cut in section of 8 micrometers. So we generate per section approximately 1,000 sections, and for to have a huge, I would say that, to characterize the response of the gut in the convalescent monkey, we analyze a section every 40 micrometers, which lead to at the end, a total of 250 sections, which were analyzed per tissue and per monkey. So the first thing that we have done is the characterization of the lymphoid structure, especially in the ileum, in the healthy monkeys. So as you can see, in the ileum, there is, this is a site where we can find the most of pair patches. So in control monkey, we were able to detect some pair patches, but there is a long region without any pair patches. When we characterize to confirm the presence of pair patches, we perform a staining of CD4 and CD20 in purple and CD4 and CD3 in green, and you can see that in fact the aggregate of cells correspond to pair patches due to the distribution of both B cells and CD40 cells, which defines the different region of the pair patches. So next, we investigate, in fact, we do the same things in the convalescent monkey, and we were surprised to see that there is a huge amount of pair patches in the ileum of convalescent monkey, especially in the monkey with classified as persistent monkeys. As you can see, we found there is a lot of pair patches as shown in the picture. And when we try to quantify, in fact, the number of pair patches per millimeter square of tissue section, we notice a significant upregulation of pair patches in the monkey with persistent infection. When we compare with healthy control or with monkey with resolved infection. It's also, we also found a significant upregulation of pair patches in monkey which resolves the infection, suggesting that, in fact, even if the virus is not detected, there is still inflammation in those monkeys. So we next decided to look in other part of the intestine, of the gastrointestinal tract. This is something ongoing, but we also found some second lymphoid structure in the genome of the persistent monkeys, as shown on the picture, and also in the colon. Whereas the structure was not expected in this part of the gastrointestinal tract, and we do not detect this structure in the control monkeys, suggesting, in fact, that as for the ileum, there is an ongoing inflammation and an endotopic organization of the immune response in those tissues. So we next try to see if there is a link between the persistence of the virus and the induction of this second lymphoid structure. And to perform that, we perform a staining of the NSP3 protein, which is marked in red on the different picture. And as you can see, we are able to detect sometimes a lot of positive cells for the NSP3 protein. And most of the cells are very close from the second lymphoid structure. And sometimes we are able to find some cells inside the lymphoid structure, not directly in the germinal center, but mainly in the mantle of the lymphoid structure. Those cells do not, there is no co-staining between the B cells and the NSP3 protein, suggesting that, in fact, B cells are not infected, but another kind of cell could be infected. Currently, we are trying to investigate which kind of cell could be infected in those tissues. So next, we decided to quantify the viral RNA in lymph node, in the gastrointestinal tract. So to perform that, we extract total RNA of 3 million of cells of each different part of the tissue. As you can see on the graph, we were able to detect the viral RNA only in the ileum, whereas we could not detect any signal in both the duodenum, jejunum, and the colon. Okay, so taken together, this result suggests, in fact, that the ileum could be a seat of persistent for SARS-CoV-2, but could be also a reservoir for this tissue. To investigate this more deeply, we decided to perform an outgrowth assay. So in this assay, we co-culture 1 million of intestinal cells with Vero cells, which are really permissive for SARS-CoV-2. And as you can see, when we put cells from ileum from the persistent monkey, we are able to amplify the viruses, whereas when we take cells from different parts of the gastrointestinal tract, we are not able, suggesting, in fact, that the ileum is the only reservoir for the SARS-CoV-2 in those monkeys. So just to conclude, we found, in fact, that SARS-CoV-2 RNA could persist in the ileum of a subset of animals. We also show that the persistence of animals show altered ileal immune structure with an increased number of pair patches and lymphoid structures. We were able to show, in fact, that the virus isolated from the ileum is competent for replication. And so, based on this, we suspect that, in fact, the ileum represent a potential intestinal viral reservoirs. So where we are now, we have started to, in fact, characterize the immune responses in those monkeys. So to perform that, we sort CD45 positive cells, which correspond to the total immune cells. And we use two different techniques to characterize the cells. We use a spectral cytometry, which allow to define 43 markers. And the first results are shown on this slide. We are able to find, in fact, on these immune cells, 31 clusters, which have been divided in different parts. We found some 4 clusters of CD8 T cells, which seems to be already activated. We also found some cluster of monocytes, cluster of NK cells. We were able also to define a cluster of CD4 T cells, which represent the main proportion of cells observed in those monkey animals. And we also found some cells which express a very low level of CD3 and which do not express neither CD4 and neither CD8, which might correspond to double negative T cell or other kind of cells, which we are still investigating currently. In the same time, we have also performed a single RNA sequencing, which have been conducted on the CD45 positive cells. We have generated the library 2 weeks ago, and we hope to have the first result during the next summer. The idea is to define the cellular heterogeneity and the transcriptional signature associated with the viral persistence. And based on this, we also decided to perform a spatial proteomics using the Maxima technology, which allow, in fact, to stain on the same section more than 100 protein. So because we perform sequential analysis of the different sections of the intestine, we will focus on section in which there is a lot of pair patches. The idea is to map the immune and viral interaction in situ and to link the tissue architecture with the viral reservoirs. So to finish, I would like to thank, of course, all the members of the labs which perform a very outstanding work every day, especially Louis and Hugo, which conduce most of the experiments. I would like to thank also the INMIT Center, which take care of the animals. And of course, our founder, especially PolyBio, which support mainly this project.
Great. Thanks so much, Nicolas. Really interesting work overall and really cool new updates. Our next speaker is Leo Yonker. She's the Chief of Division of Pediatric Pulmonology and Sleep Medicine at University of Texas Southwestern, and she'll be talking about neutrophil-driven endothelial injury and microclot formation in long COVID.
Thanks for the invitation again. Good to see you. And I'm a pediatric pulmonologist. I've been studying post-COVID syndromes in children, multisystem inflammatory syndrome in children, which we don't see thankfully as much anymore, and then also long COVID. And I'm going to share some of the pathology that we've been learning from these kids that I think not only informs what's, and validates what's happening in kids, but also helps inform what's happening in adults. And so, just a few disclosures, some funding, thankful for the funding from PolyBio, also some NIH funding, and I am a steering committee for unrelated purposes. And so, as I mentioned, long COVID does exist in children. So I just want to take a moment because many of these talks are not focused on children. I just want to highlight the symptoms that have been established by very large studies. And this is a summary in JAMA Pediatrics that children can have symptoms persisting that are impactful after having been infected with SARS-CoV-2. And in toddlers that can present with sleeping, stuffy nose, chronic cough, poor appetite. And then it can sort of in slightly older preschool kids look like fatigue, low energy, cough. And then in children 6 to 12, more symptoms that look like long COVID in adults, but also some pretty significant GI symptoms, brain fog, fatigue, pain, all of those things that we are used to seeing. So this is a really nice study, shows that these symptoms do persist. So we have to remember that children are impacted as well. The last speaker, Nicolas, gave a really nice overview that there are these reservoirs. This is from a review led by Amy and a bunch of us, that these reservoirs persist after acute infection. And so what we've been looking at over time is how that these reservoirs can elicit hyperinflammation and symptoms. And so we learned this early on from Miss C, where kids are infected with SARS-CoV-2. Weeks to months later, they'll come in with their symptoms, endocardial symptoms, GI symptoms, high fevers, very inflammatory. They have evidence of viruses in their gut. They have evidence of zonulin release. Zonulin's a regulator of tight junctions. And so the tight junctions are opening a little. Spike protein is leaking from those reservoirs into the blood, and we were detecting spike in the blood, and that corresponded with high inflammatory response. That's what we saw and published in Miss C. There's lots of evidence coming out to show similar patterns in long COVID. The reservoirs, increased zonulin levels in long COVID, and spike proteins in the blood. And so, we wanted to better understand what this meant, and we wanted to look in children and in adults. And part of the reason we wanted to look in children was because we know that children are affected, and we want to make sure that they're included in these important studies. But also, children are less likely to have those comorbid inflammatory diseases like, the things that we all encounter as we age, metabolic syndrome, high blood pressure, endovascular disease that can be inflammatory by themselves. And so we looked at samples from 23 children, healthy individuals, and 61 children and young adults with long COVID. You can see we actually had quite a range of participants from across the country and Canada. And what we did was we took their blood and looked at it through many different assays. We looked at some in vitro assays to see how their plasma and inflammatory cells impacted the endothelium. We looked at neutrophil assays, and we looked at microclots. Just to give a picture of the people we were collecting samples from, many of them had some sort of endovascular symptoms. And so here just lists the ones that were most common were dizziness with standing, dyspnea when climbing stairs, palpitations, many felt faint. And so you can see these symptoms were very present throughout the group. And so the first thing we did was we took their neutrophils and we put them in a dish, and we just watched what happened. So if you put neutrophils from a healthy control in a dish and you stain them with the right markers, this is what they look like. The blue, just they're sort of the cells, the nuclei there, and then as as they die over time, neutrophils do this, they'll sort of, and I didn't even capture any here, you'll see some that just happen to die on their own. If we take blood from patients with long COVID and we put it down, you'll see the cells sort of slow down. You'll see the neutrophils start to turn green. That's just they're picking up the dye. And this happens overall. So this happens throughout, very common. And actually we looked at this, quantified it. Hold on a second. Again, this is just an overview of what we were quantifying. We were counting the neutrophils, and then as they turned green, that was a sign that they were undergoing NETosis, and we quantified it. Healthy have very low levels of NETosis, as we saw. People with long COVID have high levels of NETosis. Here are the two groups in the middle. People with Miss C also have high levels of NETosis, and we've shown that previously as well. If there was spike in the blood, there seemed to be even higher levels of NETosis compared to those who have long COVID where we didn't detect spike in the blood. That being said, the spike test has undergone some iterations, and so that there might have been some technical issues there. But we also looked for cell-free DNA in these patients, and you can see there's increases in cell-free DNA, which is an indirect marker of NETosis. So then we wondered, what was driving this NETosis? We did have a theory from our patients with Miss C that immune complexes, so spike bound to antibody, activates the neutrophil. And so we took vaccinated controls, so they had antibodies against SARS-CoV-2. We isolated those antibodies. We added them to spike that we purchased, and we made these immune complexes. And then we added these immune complexes to neutrophils to see what happened. And again, we looked at the assay the same way that we were just doing. And the readout is that the more immune complexes in the blood, the more activating they are of neutrophils, the more netosis that's seen. So you can see if you don't have anything in there, no antibodies, no spike, there's low levels. You add antibodies alone, there's still low levels. That's good because many of us have antibodies against SARS-CoV-2 from vaccination or infection, but we don't have immune complexes because we don't have those reservoirs and that leaking spike. If you add spike alone, there's some variability because I think spike alone can activate some inflammation. But it's really those immune complexes. So when you add the spike and you add the antibodies, they join together and those signal for the neutrophils to be activated. The more that there is, the more activation. So then we said, well, that's a lot of neutrophils in the blood. What is it doing to the endovascular system? Because you don't want neutrophils necessarily activating in the blood. You want them activating at a site of infection. But if it's circulating through the blood, there are a lot of proteases, elastases in the neutrophils that are being released and now touching the lining of the vessels. And we did see increases in microclots in these kids. So there was more microclot burden overall. The number of the microclots were higher, and sort of looking at the distribution, there were lots of these small sort of broken up microclots, but we did see a lot of these long ones too. And we also saw a lot of cytokines, endothelial cytokines in these patients. Some were up, some were down, but it looked as though there was sort of a increase in sort of repair from these endothelium, suggesting that there is some ongoing injury. And there was also some reduction in lymphocyte chemotactin as well. And then we also saw some associations with these neutrophils and the endothelial injury and the microclot markers. And so this is just saying that they're both increased. It's hard to say that they're causal based on what we were looking at and the methods we were using, but there was increased amyloid and also increased cell-free DNA, which is that marker of netosis. So it's not causal, but there seems to be some connection, and we saw this for also the sort of repair markers as well. So in summary, just to pull it all together, there's good evidence that there's viral reservoirs. And when there's viral reservoirs, if there's spike leaking into the blood, the body is fighting it and driving these immune complexes. These immune complexes are quite inflammatory. They're doing what they're supposed to do. They're making the immune system respond to whatever they're detecting in the blood. But if this is a chronic sort of leak in reservoir, you're going to have chronic neutrophil activation. And when those neutrophils are released, that can injure the endothelium, and that can contribute to some of the symptoms that we're seeing. And so with that, just a quick teaser for later on, we do have a clinical trial underway to try to target this leak of these spike into the blood. And then just a quick reminder for people, if they are doing clinical trials, to please consider including children. There are many strategies for including children earlier rather than waiting for iterations of trials down the road. And generally, when people are on trials, they're going to do better because they're getting the care that they need. They're getting more attention, and maybe there's a benefit too. So try to include some children in your studies. And with that, thanks to the team. Thank you.
Great. Thank you, Leah. That was excellent. All right, I'll introduce our next speaker. It's Sean Miller. He is a research scientist at the Yale School of Medicine with a primary appointment in ophthalmology, and he'll be talking about human retinal biomarkers and organoid models to study infection-triggered neurodegeneration in long COVID.
Okay. So today I'll talk about, as Amy presented, the title is the Human Retinal Biomarkers and Organoid Studies. So how does this actually play a role with long COVID and other infectious diseases? So I have two affiliations. I know it's changing currently. I will be switching into the Simmons Foundation and Sunny Empire faculty and assistant professor at University of Buffalo. But I'm also maintaining my position here in ophthalmology at Yale School of Medicine. So I know everyone in this audience is probably very familiar, but we have an unsolved problem, and that's neurological long COVID. It's common, debilitating, and poorly understood. We know it's very common because there's over 400 million people with long COVID. 30 to 50% experience brain fog. But out of these patients, we have no approved therapies for neurological long COVID. So the idea being SARS-CoV-2 infection leading to persistent neurological symptoms such as this brain fog and other symptoms as well. But unfortunately, there's no objective biomarkers currently existing to diagnose neurological long COVID. So this is where I believe the retina comes in. So the retina, I view, as a window into the brain. It's a central nervous system tissue, so a component of the CNS. It has shared neuronal circuitry and it has vasculature similar to the blood-brain barrier. But what's really unique about it, it's non-invasively imaged. So it's an accessible site for early neurological biomarkers. And there's studies out there in research that are actually looking at retinal biomarkers and other indications such as Alzheimer's disease and Parkinson's disease. So the human retina, is it even altered within these long COVID patients? So we published this last year in Science Advances, but I just want to show the overall idea is that we took post-mortem human retinas from long COVID patients, and these had brain fog. These were from the Yale Long COVID Clinic as well, and then through our rapid autopsy program. And what we did from the single nuke RNA sequencing is we identified neuroinflammation. There was more microglia. There was a lot more astrogliosis. But what we really found that was really interesting, especially for me being a neurocentric individual, was that neurodegenerative, antimicrobial, and protein aggregation pathways blew up, just were extremely enriched within the long COVID cohort, or sorry, long COVID donors. So what we did with that study was, it took some time, but we got an IRB filed, accepted, to actually take long COVID patients. So these are from the Yale Long COVID Clinic. And what we do is we actually bring them over to the Yale Eye Center now, and we do a completely comprehensive eye exam. So it's multimodal retinal imaging. We have ERG or electroretinograms, which essentially will tell us even neurocircuitry of really what's going on within these neurons and how they're communicating. We've already completed 15 patients, and the goal really being is to identify retinal biomarkers associated with neurological long COVID, where we're able to take these eye exams and then go back to the actual neurological reports and correlate and see if there's associations between the two. This is a real example, and this is a recent patient that had come in, is that you can see this abnormal protein deposition. Those are like those black smears that you would see. That's not typical in a person of 45 years of age. So, and non-age-related macular degeneration or glaucoma, no retinopathy reported until essentially they came in with the long COVID to our clinic. So then what do other patients look like? Here's additional evidence of protein deposition and pathology in the human retina of long COVID patients. And we can see this drusen-like deposit or this bump you see in the retina. This is suggestive and very similar to what we would see as early signs of age-related macular degeneration. And AMD shows not only lipid droplets, but accumulations of amyloid within those drusen-like materials. So really what this found was, well, we're finding consistency with neurodegenerative pathways from our sequencing. So this suggested that there could be pathology in the retina of long COVID. Well, what about the circuitry? How are these neurons communicating in the retina? Brain fog, we think of cognitive impairment, and other neurological symptoms. But what we did here was, these are ERGs, and these are from real patients, again, and the left being a healthy control. I'm not going to get into the details of each of the flicker examinations, but what I want to say is, notice the red. So notice here, the right eye people, all of this is all green. All these numbers in green. If you go over to our long COVID patient, again, this is looking at ERG, you start seeing these differences. So this B wave alteration, you can see other alterations throughout within the eyes as well. And this is very common for what we're finding. Not only are we seeing from fundus autofluorescence protein deposition, we're also seeing changes in ERG. Okay, so we see these phenotypes. Well, how do we actually go further and understand mechanism in order for us to really not only diagnose, but therapeutically treat? So this is where the fun stuff comes in. The other stuff's fun, but this is even funner. And this is essentially bioreactors and organoids. And if you can see here in the left-hand panel, those spinning bioreactors have hundreds of organoids. What are these organoids? They come from blood draws from real patients. that we actually induce into human-induced stem cells, and then we make them into these complex 3D organoids or mini retinas and mini brains. And what's really unique about this is that you can take one organoid and actually do one drug or one drug in concentration or high throughput screening. They're scalable. So it gives us a nice human translational model to investigate this. So what we found is that if you take viral persistence modeling with our human organoids, you expose them to SARS-CoV-2, particularly SARS-CoV-2 spike protein, and we published this in last year, was the CRNAD28, you can see here in this top-hand panel where it says infection-triggered protein aggregation, is where we're finding amyloid beta aggregation within these retinal organoids, suggestive that SARS-CoV-2 is inducing perhaps antimicrobial or protein aggregation pathways. And then this is new, very recent data that we've been working on, and this is here in the bottom panel, you can see it says alpha-synuclein. Well, we can do the same thing and take retinal organoids from healthy individuals, expose them to high concentrations of spike-1 protein, and actually see that not only do we get amyloid beta deposition, we're also getting alpha-synuclein deposition. And I want to mention is we're thinking, okay, Alzheimer's disease associated amyloid beta, Parkinson's associated alpha-synuclein, is it just everything? Well, we think about co-pathology that exists between Alzheimer's and Parkinson's disease patients, and that's where this antimicrobial peptide aspects kind of tie in together. But the end result is, okay, we have these protein aggregates, but what are they actually doing to lead to brain fog? So we started to look at, and this is coming from the results that we're pulling in right now, so I'm showing very preliminary data, is that we can look at neuronal dysfunction. So we can treat with spike protein, we can go individually per individual neuron and understand electrophysiologically what's being altered. But we can also do it on network level with our organoids. And you can see down here, unstimulated, say you have a basal green level, we can stimulate and they respond. And this green is actually potassium signaling. And then we can use different modifiers that we've been looking at, and we can actually suppress signal that we're seeing that's altered at the spike-1 protein. Not shown here, but something that we've been looking at with PolyBio Foundation as well with their support. So this is something I want to show that just came out. It has not even been quantified yet. This is from this month. And this is showing us starting to drug screen human organoids. So we have a 96-well plate, and each one having its own organoid, and each organoid being scanned continuously. And what we're doing is treating with drugs to see, can we mitigate this green signal? And can we mitigate this blue signal, which would be alpha-synuclein aggregates and also amyloid beta or beta sheet fibrils. And then, so lastly, the translational pipeline, and to close out the talk and to show this is where we're at with our proposal, but also what we've been working on with PolyBio is that the Yale Eye Center is where we've completed and we're completing and also recruiting actively for more patients to our Yale Eye Center, where we're looking and using, and a real ophthalmologist is actually going through and evaluating all the assessments. And then what we're looking at now is actually, my group now is actually very largely organoids as well. And what we've done is establish an entire organoid area with multiple rooms at University of Buffalo. We're proceeding with some of this mechanistic insights and therapeutic discovery. And really, this is the overall pipeline. Can we find drugs to bring back to our donors and our patients? So we have an immediate impact, long COVID, for example, and we have broader implications such as post-infectious diseases. And some of those being even things like Lyme pans or pandas. And then lastly, I just want to acknowledge the most important people, the people who make this happen, as well as Brian Hafler, my co-investigator who works with ophthalmology reports and sees the patients, Penelope Weeks, research technician that works for me, Yale, my new affiliation with Buffalo, William Padula, who does Lyme pans and also has a long COVID cohort of patients for eye exams, and then also, of course, PolyBio for their support as well. And with that, I'd like to thank everybody.
John, thank you. It's just incredible work. There's almost nothing more important in my mind than the study of how amyloid beta and alpha-syn can form in response to infection. So just being able to do this, it's huge. All right. With that, I'll introduce our next speaker. It's Alessandra Lucchini. She is a professor at the George Mason University School of Systems Biology. And she'll be talking about identifying a Borrelia-associated subgroup in long COVID using urinary EV biomarkers.
Thank you very much for the introduction, and thank you for giving me the opportunity to speak a little bit about our project. And in the Lyme disease community, there is quite a bit of discussion on possible overlapping features, right, between long COVID and post-treatment Lyme disease syndrome, in terms of a common framework to study post-infection chronic conditions. And in terms of symptoms, patients often present with similar symptoms in terms of brain fog and widespread pain, autonomic nervous system dysfunction, exercise intolerance. Of course, the triggers are very distinct, right? Lyme disease is caused by Borrelia burgdorferi, and it's a very different microorganism. The question and the hope is that the Lyme community can learn some important lessons from the long COVID community. But are the possible mechanistic causes of the symptoms in these two patient population overlapping? And we see hypotheses include immune dysregulation and possible lingering of either antigenic debris or reservoirs of the pathogen, abnormal immune inflammation, such as neuroinflammation, vascular changes. Hypotheses are investigated also in terms of mitochondrial dysfunction and possible metabolic signaling modification. What is not commonly studied is the question: is there an overlap between the two patient populations? Right. The epidemiological data to answer these questions are not available. We don't know what is the overlap, and also, is it possible that if there is an overlap, then the Lyme disease associated antigen, if present in long COVID patient, contribute to symptom heterogeneity and possibly severity? And so with the support of PolyBio, my colleagues and I here at George Mason University would like to answer this question of the overlap, possible overlap and influence in terms of so and to do this, we are going to study urinary extracellular vesicles. So the extracellular vesicles are nano or micro vesicles, so packets of of molecules, including protein and DNA and lipids, that encase in a lipid bilayer, and they are secreted by virtually all cells in the organism, and they function as cell-cell communication. They reflect the physiological state of tissue of origin. What is important from the analytical point of view is that it has been shown that molecules associated with these extracellular vesicles are preserved from degradation, both enzymatic and chemical or physical, and maintain their activity. And plus, in the organism, the extracellular vesicles most likely, and it has been demonstrated clearly in mammalian models, they are not restricted to a single organ, but they circulate systemically. So in this figure, right, I represent the extracellular vesicles with the blue dots, and the vesicles are released by cells in the interstitial fluid of organs, and then they enter the lymphatic circulation and then eventually the blood circulation. They can penetrate biological barriers such as the blood-brain barrier or the blood-nerve barrier, and eventually they are secreted in the urine. And now urine functions as a integration of molecular information in the blood, right? Every few minutes, the whole volume of the blood is filtered through the kidney and integrates this molecular information. So the hope is that focusing on extracellular vesicles that can be collected in the urine can give us information in terms of not only the kidney and urinary tract, but other distant organs and infection, and molecular information also on the function of the immune system. So our activity has been focused mainly in biomarker discovery and validation for Lyme disease, which is a bit of a difficult proposition because the disease is not clearly defined by an existing predicate diagnostic test. And so in order to maximize our chances to find something that can be generalized to as many strains of Borrelia as possible, we compared molecular information and specifically Borrelia-derived proteins in studies from human patients and animal models and in vitro Borrelia. We performed the biomarker discovery with mass spectrometry, and then in silico we made sure that these peptides that we identified were unique for Borrelia, and then we also made sure that our targets were conserved in all pathogenic Borrelia clades. And so the next step then was to develop parallel reaction monitoring analysis, which is a mass spectrometry quantitative test, and immunoassays. So we performed the biomarker discovery and we measure many hundreds of proteins unique to Borrelia. We hone in on 3 that were conserved both in vitro in the mouse animal model and in human patients. And these 3 proteins are P66, flagellin, and the methyl-accepting chemotactic protein 5, which is the chemotactic sensor for Borrelia to follow the chemotactic signal and impart the movement signal to the flagellum. In terms of clinical validation, we have conducted a study on 60 individuals, follow them over time, and we see that the peptide concentration correlates with the symptom intensity in post-infection Lyme individuals. We have found that in active Lyme disease patients, the sensitivity and specificity is fairly good, above 95%. And now we are in the process. These are recent results where we see this peptide in a cohort of acute and chronic Lyme patients, both with parallel reaction monitoring and immunoassays. So the hope is, in parallel, as we complete the validation of these urinary antigens for the detection of Lyme disease, we can also use this test to assess the co-occurrence of Lyme disease with COVID patients. So this is an example of a tuberculosis case study where we follow 50 pulmonary TB patients over 6 months during standard of care therapy. And this is a study conducted in Lima, Peru. And we isolated their urinary EVs and we analyze the presence of the Mycobacterium tuberculosis antigen and the DNA that I'm not showing here. What I want to bring your attention on is the amount of molecular information, and these are organ-specific proteins that we could see in the urinary EVs of these patients. And we do see quite a good percent. Well, of course, the most represented organ system was the urinary system, the urogenital system, right? But we can still see a large amount of lung proteins that are interestingly differentially represented between tuberculosis and healthy individuals, but also a good number of proteins that are involved in immunity and many other important pathways. So for the COVID study, the plan is to use immunoassays and mass spectrometry to study beyond the Lyme disease antigens, markers of organ injury or neural injury in a long COVID cohort. And so we are going to measure markers associated with astrocytic activation, blood-brain barrier dysfunction, and neural injury, other markers that are related to the crosstalk between the immune system and nerves, mast cell activation, markers of vascular inflammation and endothelial function dysfunction, the type 1 interferon signaling. So the hope is to derive a molecular panel that can hopefully help define patient subgroups in a non-invasive manner. And so looking to the future, we also developed and patented a technology that allows us to collect urine at the point of need. It can be the doctor office, can be the house of the patients, and it has a simple origami folding technology that allows to discard the liquid part of the urine and ship the urine liquidless. And the device is featured with the concentration matrix that can capture the biological information and proteins and extracellular vesicles in the urine. So, and with this, I want to thank you very much for the attention, thank the team and the funding agencies.
Great. Thanks, Alessandra. We're really excited for you to analyze more samples. It's really cool how you can be able to tell where the extracellular vesicles are coming from, including potentially the central nervous system. That's huge. Okay. Well, our next speaker, Sasha Tabarnikova, who was from Akiko Isaki's lab, was actually delayed on a flight. So what we could do maybe now is just go straight into the question and answer session. And I'll start reading questions. Let me get there. Okay, we have for Sean Miller, many long haulers report increased floaters. Do you think that reflects the same microvascular and neuroinflammatory processes you're detecting in the retina?
Yeah, that's a fantastic question. To be honest, I'm not sure of too much literature surrounding with the floaters with long COVID, but I want to, because when I think typically with the when I think of floaters, I think of vitreous pathology. So something, with retinopathy, but typically I would think with floaters with vitreous, but we haven't looked into that. It's a great question, but it's something that, hopefully will be elucidated or shown over time. But yeah, we don't have any data on that, unfortunately.
That makes sense. Understood. Another question for you, Sean. There's a person that says, are retinal organoids vascularized, or are yours the ones you'll be working in?
No, they are not vascularized. It shouldn't be, just to show that it's not like a complete model of long COVID with retinal microangiography or any of that nature, but we're really looking at a neuroretina platform, which we've shown before that you actually get spike protein you get spike protein within a neuroretina.
Okay, that makes sense. All right, got it. Let's see. Look in here. Okay. Alessandra, going back to the regular testing for Lyme, could you speak a little bit on that? Like, people are asking about the difference between the testing you're doing and sort of the IgM-based or IgG testing currently done for Borrelia.
Absolutely. So the difference is that we are performing or developing a direct test. So we are detecting, we are using monoclonal antibodies that come from the mouse, right? But we are detecting the proteins deriving from Borrelia. And we have anecdotal evidence that we can see Borrelia as soon as the erythema migrans rash appears. And so a couple of weeks before IgM. When IgM is detectable, right? So it is, instead of probing the immune response and the serology, we are investigating the the direct shedding of Borrelia antigen. And we, I guess you brought this up, or some, Amy, in one of our conversations in the past. It would be an excellent question to correlate the appearance of the antigen in the urine with the immunologic response, right? And this is an information that we don't have, but it would be also important regulatory-wise, because if it's true that you can predict the seroconversion, then it might even be simpler, go to possibly the FDA and say we have a test that predicts seroconversion. So this is excellent question. And in theory, we should see the antigen before IgM. And it's going to be interesting to see how it pays out. Yeah.
That makes sense. And actually, Alessandra, in this study that you're going to be measuring these urinary Borrelia extracellular vesicles in patients with long COVID, we're getting the samples from the core clinic at Mount Sinai. And we are going to run the standard CDC existing Lyme, IgG, and IgM testing on participants. Then we're also going to run a lab that has a Lyme-6 peptide ELISA test that some people think is a little bit more specific or comprehensive. So we'll basically have 3 tiers of information. Like, how would this participant have tested for Borrelia via CDC testing? How would they test for Borrelia via a Lyme-6 peptide test made by a specialty lab that might be more sensitive? And how might they test for Borrelia vesicles via your, honestly, most innovative extracellular vesicle methods? So hopefully that will be an interesting comparison that we'll look at.
That's exciting.
Yeah, and it's part of the thinking. It's a little like the VIPER program. I think it shows the value sometimes where in the VIPER program, which is that big long COVID diagnostics validation program, we're running a lot of different forms of testing on samples collected from the same patients. And then that allows you to see how they perform head to head. And I think there's a lot of value in doing that across this space, which is like you can take some of the older tests versus some of the more new tests, things like that, to be able to understand and optimize the results. So it makes sense as a trend, for sure. Looks like we have a couple more questions for you, Sean. I think, what about light sensitivity? There is a good amount of photophobia that people experience with long COVID or related conditions. Do you think that might be tied to anything that you're seeing in the retina?
That's a fantastic question. And actually, initially, I want to say yes, a short answer. And it started from looking at proteomic data of long COVID retinas in the sequencing, is that it came up in our pathway analysis. So then going through the ophthalmology exams, we're actually finding that as well, this light sensitivity and especially dark, being able to see in the dark, night vision, it's been altered. And hopefully we'll continue to find more as the trial continues.
Yeah, really interesting that maybe even showed up in some of your data. Wow, very interesting.
Amy, I just want to say that I've been answering the chats as they came through in the texts as they came through. So I maybe I shouldn't have. Sorry.
That's okay. But do you have any takeaways, honestly, Lael, from the questions that you answered?
A lot of people were asking what would be the best strategies that I'd recommend for treating the activated neutrophils. And so I would say rather than treating the neutrophils, I would try to target the underlying cause. And I gave a little teaser to Peter Moskowitz's talk later, which is going to be talking about our sort of, where we stand with the lorazatide study, which is trying to reduce the amount of spike that's leaking into the blood. So first treat the virus. If you can get rid of these reservoirs that seem to be there, that's great. If not, at least contain them so it's not leaking into the blood and causing this neutrophil and endothelial inflammation.
That makes a lot of sense. Yeah, exactly. And lorazatide being a drug that can help gut barrier permeability. So again, what you're thinking is the virus sticks around in the gut, but spike protein it makes might be able to leak into the blood. And that's part of, again, why there might be the neutrophil activation and some of the netosis. And so if you can just prevent that gut leakage.
Yeah, that would be an important component. And perhaps there's some anti-inflammatory signals on top of that would need to at least short term be turned around. But I think trying to get as close to the source would be really important.
That makes sense. exactly. There's a good question for Alessandro Lucchini in the study where you're looking for the urinary vesicles, markers of neural and organ injury in the long COVID cohort. Are you recruiting long COVID patients with evidence of Lyme as opposed to long COVID with patients with no evidence of Lyme? I can even just answer that one because we're the clinical site collecting the samples for this study. No, what we're doing is we're bringing in just, I think at this point it's like 30 patients with long COVID who come through the clinic and are recruited. What we're interested in seeing is just in a just sample of regular long COVID patients, do any of those patients potentially have these Borrelia extracellular vesicles? And that comes down to, what we've done. We have another study running out of Sinai. We're getting blood from patients with long COVID, also an ME/CFS diagnosis, also an Ehlers-Danlos syndrome diagnosis. And we're sending samples to Ed Breischwitz's lab at North Carolina State University. They're also a chronic tick-borne, vector-borne illness lab. They're looking for Bartonella, which is another bacterial organism that can be transmitted by ticks, and Babesia in blood from those patients. There's actually precedent that they might be finding Bartonella in blood samples from long COVID patients. There are 2 case series out right now, which show that COVID or SARS-CoV-2 infection reactivated previously dormant Bartonella in a patient who was diagnosed with long COVID, and that the treatment of the Bartonella that reactivated, a little like how Epstein-Barr virus or these other pathogens can reactivate, resulted in symptom improvement for the patient. But the thing is, that study with the Ed Breischwitz team, Bartonella and Babesia are more commonly found in blood. So they're able to look for the genetic material of those organisms in the blood of these patients, whereas Borrelia is extremely tissue associated as a pathogen. It might be one of the top pathogens out there that just, burrows deep into tissue. It's a spirochete organism. It really can do that very effectively, to the point where even in acute Lyme disease, a direct detection test for blood is not even used, because it's not even thought that in acute Lyme, Borrelia might be there in blood to measure, let alone in a case where it might stick around in a person's body. So that's why we reached out to Alessandro, especially because the case of understanding if people with a long COVID diagnosis have Borrelia as a co-infection, basically in their case, we then need to maximize the technology that can pull vesicles like she's doing out of tissue, or pull the signal out of body sites and into the blood via the EV-based tools. So that's what we were thinking about. And again, what we're thinking of in the big picture of this is that, we know again that herpes virus reactivation might be an issue for some patients, but we also realize that some patients with a long COVID diagnosis might have missed a tick bite, or again been exposed to Bartonella, which can be transmitted via cats, other animals, fleas, and that we should be accounting for these other infections as also potential players in cases. That's the overall thinking. All right, let me see if there's any other questions. Let's see. I think those are the majority of questions. I could just grab another one quickly. Someone asked if there's a way to distinguish between spike protein from infection versus spike protein from the vaccine. There are 1 or 2 teams working on this in the background. There are some differences, I think, in proline residues between the spike that's in the vaccine and the spike that the virus creates. And so if you can use a method called mass spectrometry, which can look at the structure of proteins, it might be possible to pick up those differences, and you could say, oh, this is a virus spike from the virus or spike from the vaccine. So I actually know there are 2 groups I know in the background that are working on that, because it is worth doing. Okay. All right. We've gotten to 1 p.m. Now let me just see quickly. Okay. What I'm going to do is, the person who was going to speak at 1 p.m. was Michaela Loci. She's amazing, but she actually felt really sick today. And so we said, listen, you should rest. We all know we should do that when you get an infection. So I am actually going to present some slides on her behalf and just overview a couple of things about her work. So hold on. Okay, so the project that Michaela has been working on is decoding lymphoid tissue adaptive immune responses connected to long COVID. And that's Michaela on the slide. Overall, it's a really important project, again, because of the importance of studying tissue samples in patients with long COVID and related infection-associated chronic conditions. What her team does is they collect samples from the lymph node via what's called a fine needle aspirate biopsy. And they've now done this on a good number of patients with long COVID and controls. So overall, and I don't want to overspeak for Michaela, but she did present at the last symposium, so I'm going over some of the things she already said there. They're studying B cells responses triggered by SARS-CoV-2 infection in long COVID and then again in healthy convalescents, again from these lymph node samples. Now, the initial work so far has uncovered a profound disruption of B cell responses directed against SARS-CoV-2 in long COVID. And I think this is key, and it's just like I'm a broken record on this topic, I know, but these findings have only been noted by their team thus far in the lymph node samples, and they are not evident in blood from the same study participant. So it's interesting to think about that. The lymph nodes contain the germinal centers where, antibodies and B cells are produced, and it's a very important site for them. So again, it shows that being able to get to that direct site, so important for B cell activity, does seem to matter in honing in on the most signal on this front. And again, it's possible that these, dysregulated B cell responses that they're finding might explain why some patients with long COVID don't fully clear the SARS-CoV-2 virus. Still more work needed. But what's cool is that the team, and we got funding through PolyBio from a great foundation, the Wallace Research Foundation, to help Michaela add in a second part to this study. She's also looking at the impact of Epstein-Barr virus activity on the B cell responses in these lymph node samples. This is important because a lot of us see as a trend that patients with long COVID, a subset, seem to have Epstein-Barr virus reactivation. In other words, that can, be from the fact that when people get COVID, the SARS-CoV-2 virus downregulates key parts of the immune response, including parts of interferon signaling. Interferons are really important in keeping other viruses in the body in check. So that creates an opportunity for a virus like Epstein-Barr virus that might have been controlled by the immune system and essentially kept in check by the body to potentially reactivate and start to drive problems. So interestingly, in this context of research, Epstein-Barr virus infects B cells directly. So it's really interesting to think about the fact that one of the root cause reasons why some long COVID patients might have skewed B cell responses even to SARS-CoV-2 when they're infected could be because EBV is acting differently or acting in an aberrant way in some of their B cells to begin with. So now Michaela's basically doing extended work to look at whether, Epstein-Barr virus, again, which can hijack the fate of B cells and the developmental trajectories of B cells, could be resulting in some of the lower quality or aberrant responses that make response to SARS-CoV-2 and other issues in this context relevant. And I think it's really cool because a lot of what we talk about clinically when we talk about long COVID cases is a multiple hit model, where sometimes it seems like one infection that a person gets can dysregulate or downregulate the immune response in a way that makes them respond less optimally to a second infection. And so this study really tests that multiple hit phenomenon by looking at the relationship between two different viruses on these key immune-based signaling that's very important for our health. So overall, when I was looking at this research and being a big fan of the collection of lymph node samples, I just thought I would bring this study up. This is not Michaela's work. This is a different team. from the University of California, Davis School of Veterinary Medicine. But it's a really cool paper because what they looked at is they're studying feline infectious peritonitis. This is a disease in cats. It's where they get a coronavirus infection and the coronavirus persists. So it's a persistent coronavirus infection. It can become really severe and even kill the cats. So it's a major issue with the health of cats if they have this FIPV. Now, interestingly, for years, and I just learned this in the paper, the veterinary community and the research so far in the FIPV space, the thinking was that the persistent coronavirus driving the disease only persisted in macrophage cells, which are a type of immune cell in the body, but not that that the virus could persist in T or B lymphocytes. But what this team did, and it's a little similar to what Michaela's doing, is they got lymph node samples from the cats with FIPV or FIP. And so you can see that they got, in this case, samples from the mesenteric lymph nodes of the cats. And then they used some really smart methods, single cell RNA-seq, immunohistochemistry, RNA fish, to look for the persistent coronavirus that causes FIP in these mesenteric lymph node samples. And then they were able to show in those samples, using actually several methods, that in the lymph nodes, the FIP PV virus infects and enters T and B lymphocytes. These are immune cells of the cats. And they were also able to show even, for example, that subgenomic FIPV RNA was detected in CD3 positive T cells. And subgenomic RNA is produced when viruses replicate, which suggests viral replication, or in other words, active virus in the cells. And they also showed that these T or B cells containing the FIPV RNA could uniquely activate antiviral and innate immune pathways, suggesting that the virus is doing something, again, in those cells. And last, low levels of this RNA persisted in the lymphocytes of the cats, even after antiviral therapy that it's sometimes commonly used to treat these cats. So what's really interesting, and the takeaway from this paper, is that they basically paradigm shifted in one study what is understood about FIPV and the cells that it can infect, because they went further and collected the mesenteric lymph node samples from these cats instead of just looking at the immune cells in blood from the cats. So that's just a takeaway message. And last, when I did a Zoom with this team, and I've gotten to know them because I think they did great work, and they pointed me to this paper. Every time you think about findings like this, you say, well, how could we treat this, right? So let's say you had an infection in a T or B cell, and you wanted to remove it or help the cell remove the infection. Well, they pointed me to this paper in Cell. This isn't cancer, so I'm switching gears a little bit, but it's in vitro, so laboratory-based work. But what the team showed here is that they were able to take bone marrow stem cells and show that those bone marrow cells could form nanotubes with exhausted T cells and transfer mitochondria, like healthier activated mitochondria, into the CD8 T cells to basically boost their function and remove that exhaustion that they were experiencing. And so the FIPV team, is actually doing some experiments now to see if potentially stem cell-based treatments that could transfer healthy mitochondria into T or B cells could be some kind of future therapy that invigorates those immune cells to better clear persistent virus. This is still all very early work, but I think it's just interesting, even for long COVID treatment, as a thought over time. And okay, I'll stop there. It's time for our next speaker, who is Peter Moskovich, and he is at Harvard Medical School. He'll be presenting on targeting gut barrier dysfunction to treat pediatric long COVID.
Well, hi, everyone. My name is Peter Moscovis. I am a pediatric and adult pulmonologist at Massachusetts General Hospital and Harvard Medical School. I've been working with my friend and colleague, Dr. Lyonker, who I see is also on the call, on this trial that was supported by PolyBio. And we're here to give you an update on kind of the interim results. The study is just winding down, so we don't have final results to share, but we have some interim kind of blinded results. I believe some of you on the call have heard a little bit about this already, but just a few words about the rationale behind the trial. As you know, the post-acute sequelae of COVID lie on a spectrum ranging in childhood from MIS-C to, in older children, what we commonly understand as long COVID, and in adults certainly as well. And the pathophysiology, at least part of the pathophysiology, is thought to be related to leaky gut permeability. So multiple studies have shown a SARS-CoV-2 reservoir in the gut in PASC, and there's a protein that regulates communication kind of across the gut barrier, these tight junctions. This protein is called zonulin, and when it leaks into the blood, it suggests that that the permeability is not intact. It's been shown that for every unit increase in zonulin in the blood test, there's an increased risk of PASC. And as is well known to the audience, spike proteins have also been detected in the blood of adults with PASC. So putting this together, at least part of the pathophysiology of long COVID is thought to be related to gastrointestinal reservoirs of SARS-CoV-2, driving dysbiosis and alteration in the gut microbiome, release of the zonulin that reflects leaky, sort of the gut leak into the blood, and the spike protein penetrates into the blood and activates the innate immune system. Microclots are formed, driving the adaptive immune system, and increased antibodies against SARS-CoV-2, but also against the body itself. So in response to this proposed mechanism, it's been known for some time that this drug, lorazepam, can affect gut permeability. So it's been primarily developed actually for celiac disease, but there's rationale to use it in long COVID. So it's a synthetic peptide composed of naturally occurring amino acids, formulated in an enteric-coated with enteric-coated beads in a capsule. And the idea is that this would prevent the opening of the intestinal tight junction, originally triggered by gluten, inflammatory cytokines, but also likely SARS-CoV-2. So what's beautiful about this drug is that it doesn't really act systemically very well. So it really acts just in the gut, which means that there's a much better side effect profile. And the trials that have been conducted not in COVID have demonstrated very good safety and tolerability. So my colleague Lyonker did a couple trials of lorazepam for MIS-C. And again, here's the mechanism, and demonstrated, at least in a small sample size, an improved time to resolution of GI symptoms and an improved time to first clearance of spike protein. So the trial that we're reporting on today was a randomized double-blind phase 2A trial with 2-to-1 randomization. So for every one person who got a sugar pill, a placebo, 2 people got the zonulin antagonist lorazepam. We recruited children and adults aged 7 to 50 years with a known history of SARS-CoV-2 infection and symptoms consistent with long COVID. We asked them not to get a COVID vaccine during the study period, and we asked people to not participate in any other long COVID trials during the study period. And the other medical exclusion criterion, more to make sure that the drug was safe, were liver function tests and eGFR that were more or less normal. The dosing is listed there, and the period was here. So we did a total of we followed people for a total of 8 weeks. 3 of those weeks were with active treatment, and then an additional 5 weeks of follow-up. We're just finishing up the last week 6 to 8 follow-up visits. We've completed enrollment and are just completing data collection. So the endpoints in this trial were the symptom burden questionnaire, the SBQ-LC for long COVID, a quality of life survey, some orthostatic vital signs measured before and after, and for a subset of participants who were interested, we did cardiopulmonary exercise testing. So here's a schematic of what all those tests were and when we did them. We also collected some blood samples and nasal swabs. So preliminary results, we've enrolled 107 participants. The final in-person study visit was May 8th, and the final remote visits are underway. We'll complete next week. The 107 participants were about 3/4 adults and 1/4 children. Here are the demographics. You'll see the mean age was about 30. Mean number of COVID infections was about 2. Nearly everybody had been vaccinated. It was primarily a Caucasian population with some other as well. About 2/3 women. And the mean time since last COVID infection or last COVID vaccine was about 2 years. So this is the baseline symptom burden. What you'll see is that this list, these are all the domains of the SBQ, the symptom burden questionnaire. You'll see there's a lot of circulation symptoms, the sort of the POTS phenotype, a lot of fatigue reported, a lot of problems with sleep, and some muscle and joint discomfort, a fair amount of stomach and digestion, GI symptoms, and then finally, a really high impact in daily life. The blinded interim analysis, which we did after the 40th participant and was presented to the DSMB, the Data Safety Monitoring Board. So we don't know which of these people got placebo and which got active drug. Showed that so lighter color is better, darker color is worse. So it shows that there was some improvement in sleep and in fatigue and an impact on daily life. But again, we don't know who got the active drug and who didn't. So we're looking forward to diving into the details once we unblind the data later this summer. But you can see the individual patient trajectories were demonstrated a significant improvement in GI side effects and fatigue and cardiovascular side effects. Or rather effects of long COVID. So we're excited about these preliminary results, but we're waiting to see what the final results will show. So the next steps are to complete final data cleaning and analysis and to review the clinical efficacy data and the biomarkers. And we'd like to thank especially the participants who took part in this study, but also our study team and PolyBio for your wonderful support, along with the Massachusetts Consortium on Pathogen Readiness.
Great, Peter. Thanks for such a helpful update.
Of course.
Awesome. All right, I'll introduce the next speaker. It's Michael VanElzakker. He's an associate professor in the Division of Neurotherapeutics at Harvard Medical School, and he'll be talking about long COVID and ME/CFS neuroinflammation and neuroimmunology.
Thanks, Amy. Appreciate it. So, hi, everybody. Glad to be here again today. So I'm here to talk a little bit about the neuroimmunology of infection-associated chronic illness and some of the studies that we have going on. We have a lab at Massachusetts General Hospital and Harvard Medical School that sort of, specializes in neuroimaging, but we really used the PolyBio model of collecting samples from the same patients and trying to measure lots of different things in them to the extent that we can that are important. In my lab, we mostly have access to fluids, but we can use brain scanners to get at the brain, which is the most difficult tissue of all to investigate. So just a little overview of some of the stuff happening in my lab, because I think it's interesting. We have a dual PET MR study looking for neuroinflammation in patients, and that's a little bit of what I'll talk about today. We have a 7 Tesla fMRI neuroimaging study that is looking at cerebral spinal fluid flow and vagus nerve function. We got a really generous donation from a family to study cervical cranial cranial-cervical instability, and that is probably a vulnerability to some of these infection-associated chronic illness. Not everybody with these illnesses has that issue, but it seems like a subset do, and then there's also patients where that's the only issue. That's been a really interesting study, and Dr. Murakami, we'll talk about that in a little bit. Really challenging study, to be totally honest, because a lot of patients have metal in their body and then can't be scanned with the 7 Tesla very strong MRI. And then also in the middle of the study, kind of, you know how your laptop updates and you can't use your favorite app? That happened to the scanner where it updated the software, and we kind of had to go back and make a lot of changes. But we have data today that we'll show. We have another study. This one is actually NIH funded, where we're using fMRI imaging and EEG in long COVID patients and pre-2018 ME CFS patients to try and understand cognitive dysfunction. Across most of our studies, we collect sleep quality data because that's one of the things that seems to affect patients the most. If you ask them about quality of life or things that can throw them off, sleep quality seems to be really important. And so we're trying to understand what's related. So we give them an overnight sleep device to wear for the 2 nights before they come in. Pupillometry is another measure that we take where, again, like Sean mentioned earlier, photophobia is a real big issue in a significant number of patients, not in all patients, but in a significant number of patients with these infection-associated chronic illnesses. So if we can understand what sorts of other issues relate to this, we may understand mechanisms a little better. And then we have multiple forms of plasma, whole blood, and even saliva analyses that we have a lot of data on that, not all of which I'll present today, but some of which I will. And then we have another study where we're taking skin punch biopsies from patients with small fiber neuropathy to try and understand the mechanisms of what might be happening in these patients. Again, actual tissue, this pieces of skin that we're trying to understand. And then another study looking at fibrin accumulation using a PET scanner radioligand that binds to fibrin. So these are the things that I'll be talking about today. And I'll start with the dual MRI PET scan for neuroimaging. So this is an example of microglia, which is the most common neuroimmune cell in the brain. When you look at a brain, a majority of the cells that you're actually looking at are glial cells, of which microglia are one type. So it's actually not mostly neurons, it's mostly glia. And those are neuroimmune cells. They are completely involved in normal neural function, normal brain function. But when they activate, if they stay activated, that can cause problems. And I'll talk about that in a second. So they detect pathogen-associated molecular patterns. Importantly, they can detect any kind of molecular pattern from different pathogens. So in other words, they're going to have a similar response to different pathogens. That's not at all saying that the different pathogens don't matter. In fact, they matter quite a bit if you actually want to clear them and stop the ongoing provocation. And the microglia actually also respond to cytokines and other inflammatory markers. And when they do, they change shape. They actually change morphology. They activate, and then they dump out neuroexcitation. That includes cytokines, but it also includes things like glutamate, ATP, prostaglandins, things that will mess up the normal signal to noise of brain cells and cause a loss of many forms of inhibition. And they also release or produce what's called the translocator protein. And this is the thing that we can measure with a brain scan. This is what we can measure with a radioligand that binds to it, and we can tell where it's built up. So we previously published this study using that method. And this was an earlier study that we were trying to get out pretty quickly. And we compared to pre-COVID controls. Since then, we've expanded the number of patients that we're scanning. This includes long COVID patients, but also we have a group of pre-2018 ME/CFS patients. And now we're recruiting COVID recovered controls. The important question is, obviously, SARS-CoV-2 can affect some people quite a bit, make them feel sick for a long period of time, but does it affect other people as well, maybe in a less profound way or in some other way? So the patients that we're looking at now are compared to COVID recovered controls. And this is an example of long COVID patients. Importantly, our long COVID patients fulfill the ICC criteria of the ME/CFS diagnostic label. In other words, they have sort of broad sickness symptoms, including difficulty recovering from exertion. And what we're showing here, what I've got highlighted here, is an area of the brainstem that shows activation of these glial cells in the long COVID patients versus the controls you can see over here on the right. So what we're looking at right here is 23 long COVID patients versus 14 COVID recovered controls. We also wanted to compare pre-2018 ME patients using controls, and we're also able to compare it to pre-COVID controls as well. There, we also see brainstem activation in the ME. In this case, I'm calling them ME patients. What I mean by that is pre-2018. Also in the brainstem, that probably is related to the fact that there are similar symptoms, but we also see some patterns that aren't quite the same in ME versus long COVID. Long COVID seems to be a little bit more associated with areas where there's a thinner blood-brain barrier and more related to the vasculature than these patients. So why brainstem? Well, this is a cutout of the brainstem that I'll sort of blow up so you can see it. And what this shows is that this area, the MTS, the nucleus of the solitary tract, is where the vagus nerve enters the brain from the body. And that is what signals any kind of inflammation from any cause to the brain to say, hey, brain, your body is sick, go to bed. So there's a lot of structures in close proximity right there that are involved in autonomic function, inflammatory pain, sickness, nausea, some of the symptoms that are shared across different patient groups, despite having different drivers. Again, that doesn't mean that the drivers don't matter, because if you want to stop the signal, you would stop the drivers. So the vagus's highly branched structure allows local signaling detection. Different pathogens can send the same signaling, but you have to deal with each of those pathogens on their own, and that local signaling can trigger shared symptoms. So we take blood from our patients, including from a larger group of patients that didn't get PET scanned, and I wanted to share some data about that today with brain-derived p-tau217. This is a marker of risk for Alzheimer's disease. The reason we were interested in this is because there seems to be an increase in new onset Alzheimer's disease in older adults following COVID. And p-tau217 is particularly important for diagnosing Alzheimer's. This is a relatively new finding. When we looked at total tau, there wasn't a difference between the three groups, controls, ME, pre-2018, and long COVID. But when we looked at just the brain-derived, came from the brain, p-tau217, only one of the healthy controls was positive out of 49, 26 ME patients, and one was positive, so 3%. But in the long COVID patients, 10 out of 69 or 14% were positive. So I think that's evidence that it's possible SARS-CoV-2 may be kind of a unique risk. I want to leave it at that and say thank you to my group, and thanks very much to the patients who come in and are so generous with us. Thank you.
Great, Mike. Thank you. There's a lot going on in your lab. Really good overview. All right, our next speaker is David Putrino. He's the Director of Rehabilitation Innovation for the Mount Sinai Health System, and he'll be talking about non-invasive neuromodulation for cognitive impairment in long COVID.
Thank you, Amy. I'm going to go ahead and share my slides. I didn't know that I would be following on from Mike, my number one hero. So I've got some Mike VanElzakker slides in this presentation. So sorry that people are going to have to see awesome work twice. But just to start off, as we all as we all think about why people with long COVID get symptoms, one of the things we think about at the Cohen Center for Recovery from Complex Chronic Illness is what are some of the drivers of symptoms in long COVID, and what might be going on that cause some of the problems that we see in patients every single day. Then as we think about each of these drivers, we also just have to think about what are some unique approaches to addressing each particular driver, and also, when more than one driver is occurring at any one time. And to that end, I'm going to talk about a clinical trial that we completed that was really targeted toward addressing neuroinflammation and mitochondrial dysfunction in folks with long COVID. In 2022, we worked with Michelle Mongi's team and Akiko Iwasaki's team to show that even the most mild acute SARS-CoV-2 illness could lead to neurological damage by virtue of the inflammatory cascade that COVID seems to cause. This idea that COVID infection creates the production of chemicals such as cytokines and chemokines that go on to cause microglial reactivity and cortical damage, and in fact, central nervous system damage was something that we published on quite early. And to that end, and as Mike has also shared, we see evidence of this even in PET scans of people with long COVID's brain, where we see a greater propensity for neuroinflammation to be affecting the brains of people with long COVID when compared with healthy controls. So this tells us that some of the symptoms that people may be experiencing, whether they be cognitive, whether they be some of these symptoms of head fullness and headache and migraine, these could be driven significantly by neuroinflammation. Work that we've done in our clinic, just speaking with patients, has shown us that cognitive impairment in long COVID is common and disabling. It's one of the most concerning symptoms that people with long COVID tend to experience, because everyone really fears the sort of loss of independence and loss of even identity that comes with loss of cognition. And what we showed in this paper in 2022 was that over 60% of people with long COVID will report cognitive impairment that is significant enough to affect daily functions. So this is something we really need to address, because when more than 60% of patients are being affected so severely, we need to do something. So we were, very proud to work with a company called Pharion that has developed a first-in-human device that they call the Mighty device. It provides something called microtesla magnetic therapy. So this is low-energy, low-frequency magnetic fields that can reach both superficial and deep brain structures without any invasive surgery or invasive procedure. It is a wearable device that is able to stimulate neurons. In animal models that were published earlier this year, what we were able to see was that whether we're looking, sorry, not animal models just yet, in human tissue models, in vitro models, what we were able to see was that providing this microtesla magnetic therapy created a broad anti-inflammatory effect that really did seem to reduce inflammatory cell activity in inflammatory chemical activity in sample human brain tissue. They then moved from in vitro to in vivo, where we had alive behaving animals being treated with microtesla magnetic therapy, and we were able to see both improvements in the quality of neural tissue, as well as, again, this sort of broad anti-neuroinflammatory effect as the animals were treated with the MMT. So that brought us to our first in-human clinical trial of the technology. These animal results were very compelling, so we tried it out in individuals. This was a triple-blind, placebo-controlled, randomized controlled trial, meaning that people were randomly assigned to either placebo or active treatment. The assessors were blind, the clinicians overseeing the intervention were blinded, and the patients were blinded as to which groups they were being assigned into. We captured a whole bunch of cognitive assessments and patient-reported outcomes and blood biomarkers at baseline, at the end of a 4-week intervention where patients were using the device for 15 minutes every 72 hours, and then at follow-up, which was 4 weeks after the end of intervention, to look at durability of effect. 30 participants were recruited in a 2-to-1 active to sham randomization schedule. They had to have at least mild cognitive impairments scored by a neuropsychologist, and they couldn't have any other diagnoses that might affect cognition, such as a traumatic brain injury or a stroke that was causing ongoing cognitive impairment. Our primary outcomes for this study were just to look at safety and feasibility, but we then were able to look into some efficacy at the end of the trial. So top-line results, we had no device-related adverse events, which was good from a safety standpoint. We met our safety endpoint. It was a highly feasible therapy in that all participants completed 100% of all scheduled sessions. So we had 100% adherence to a treatment. And the blinding integrity was intact. So people were not able to correctly guess if they were part of the sham treatment or the active treatment group. In addition, as we were looking at some of our clinical trial outcomes, we saw that visual attention improved. And as you can see in the MMT group, which is this blue column, at the end of the therapy, they improved a little, but they improved much more on follow-up, indicating that the gains weren't just sustained, but they continued to increase. We saw the same with cognitive processing speed. We saw the same with attention and inhibition, which is a marker of executive function in the brain, these frontal lobe behaviors that cost a lot of energy to us. And we also saw emotional well-being significantly improve, while people in the sham actually experienced some significant decreases in emotional well-being. So all in all, as we were looking at some of these outcomes, it was very encouraging to see these improvements. We also received a lot of positive feedback from MMT-treated participants who were really enthusiastic about the therapy. So we're not losing any time. We're moving into a much larger multi-site pivotal clinical trial for this technology so that we can really help the company to get the FDA approval to make it available to patients ASAP. But this has been an extraordinarily encouraging first trial of the technology. I'd like to thank all of the wonderful people who help us to be able to do this work, and always thanking our patients who are so trusting and consistent in getting involved in our research. We really appreciate them. Okay, thank you.
Amazing, David. Those are very compelling results. I'm very glad the pivotal trial is in process. So, okay, I'll introduce the next speaker, who's Mark Painter. He's a postdoctoral researcher in John Ware's lab at the University of Pennsylvania, and he'll be talking about virus-specific CD8 T cells as biosensors of antigen persistence in long COVID.
All right, it's a pleasure to be here and to present today our work, as Amy said, using T cells as biosensors in long COVID. So I know we've been talking a lot about tissue today, and for sure there's a lot of value in tissue, but I want to give a short defense of the blood and show you some of what can still be done in the blood to understand what is actually happening in tissues. So really the overarching question that we're trying to pursue is, are there persistent viral antigens that are actually stimulating ongoing immune responses in people with long COVID, in everyone with long COVID, or a subset of people with long COVID? And the concept here is really to take advantage of natural properties of T cells to use them as a blood-based biosensor of viral antigens. So in every person, we have a diverse repertoire of T cells that are constantly moving through the blood and the tissues, surveying virtually every cell in the body. Each of these T cells has a unique T cell receptor that can recognize small fragments of the virus, viral peptides presented by MHC on the surface of infected cells. And so only a small portion of your T cells are actually capable of recognizing any given virus. But the cells that do recognize it, if they see that viral peptide, become activated in the tissue where the virus is present. And when they become activated, they undergo a number of changes that we can detect at the level of the proteins they express, the genes they express. And importantly, these T cells that are activated in tissues will then re-enter circulation, where we can detect them in the blood, still bearing these hallmarks of recent activation. And so the key is, can you actually fish out these rare antigen-specific cells, these rare virus-specific cells, so that then you can interrogate whether they've recently been activated somewhere in the body or not. So for this, we use these HLA class 1 tetramers. So this mimics what the T cell recognizes on the surface of an infected cell, where we load the HLA molecule with a viral peptide, in this case, like a spike peptide. We then label these with fluorescent molecules, 2 different fluorescent molecules, and a T cell that's able to recognize that viral peptide will bind to both of those reagents that we made, and we can detect it by flow cytometry as this dual staining population. And importantly, this doesn't require stimulating the cells ex vivo. So we can really take a sample from blood, immediately analyze it and preserve the state of that cell in the body and understand whether it was activated in the body without having to activate it in a dish. And so we don't just look at one virus at a time. We can actually combine through this multiplexing approach that other people use as well, really pioneered by Evan Newell's group. You can multiplex different combinations of these 2 fluorescent channels to then detect virus-specific cells from a number of viruses simultaneously from a single blood sample. So we're not just asking if SARS-CoV-2 specific T cells are activated, but also EBV, flu, etc. And we've been fortunate through PolyBio, through the support of PolyBio and the connections we've been able to make here, to receive samples from a number of collaborators at Penn, Yale, and UCSF, to be able to analyze hundreds of samples to start to ask these questions. So here's just an example of what we can see in a person with long COVID who does not have activated spike-specific T cells. We can sensitively identify these T cells, and we can look here at 2 activation markers, PD1 and CD39, and see that those T cells are not expressing these markers of activation, suggesting that they have not recently seen SARS-CoV-2 antigens anywhere in the body. But in a different participant with long COVID, we can find their spike-specific cells and actually see that these are quite activated, suggesting that the virus might be present and being sensed by these T cells. And if we just look here at the activation of spike-specific T cells in acute infection, of course, they're activated. In acute vaccination with spike mRNA vaccine, of course, they're activated. In people who recover, you have generally low activation, although there are some people where we do maybe see a sign of activation. And then in people with long COVID, Most people, we don't see any evidence of persistent stimulation of these spike-specific T cells, but there's a subset of people where we do see this activation. And importantly, we see this persist for even years post-infection. So this is the subgroup of people we've become really interested in. I'll just say that David Reig in our group has been doing very similar work with CD4 T cells, but for time, I'm not going to show that today. And very quickly, I just want to say that we also can see similar things with EBV. So we see that actually it's quite common that during acute SARS-CoV-2 infection, but not vaccination, you get activation of EBV-specific T cells, which is consistent with EBV reactivation. And in a subset of people with long COVID, you see persistent activation of these EBV-specific T cells. So that's another area we're interested in and pursuing, but for the rest of today, I'm going to focus on SARS-CoV-2, besides saying that we can look at other viruses too and see that this really is a virus-specific effect. So we don't see at least any consistent activation of CMV-specific T cells in long COVID, and we definitely don't see activation of flu-specific T cells in long COVID. So this really does seem to be a virus-specific phenomenon driven by SARS-CoV-2 and also EBV in some patients. So building on this flow cytometry data on hundreds of patients, we did a deep dive single cell sequencing where we sorted out these different virus-specific T cell populations and then can do sequencing to see every gene that they express from a number of convalescent, acute, and long COVID samples. And just showing this is each dot is a cell clustered by what genes they express. So we can see all of the typical gene expression profiles that you expect to see in human blood. And we can really nicely in the sequencing data use our tetramers to identify the different virus-specific T cell populations. So here's SARS-CoV-2, CMV, flu. And we can do this for dozens of antigens all at the same time in the same samples. And of course, what virus a T cell recognizes changes how that cell differentiates. So you can see, for instance, CMV has this classic terminal effector phenotype, whereas flu-specific cells occupy this sort of central memory. And in people who recover fully from SARS-CoV-2 infection, the spike-specific cells look a lot like the flu-specific cells. And so what we're interested in is looking at our convalescent people. If we look at flu-specific cells, during acute SARS-CoV-2 infection, those cells don't really change. And during long COVID, those cells don't really change. So flu-specific cells, no matter what is happening with SARS-CoV-2, look the same. Now, if we look at SARS-CoV-2 spike, they look like flu in people who fully recover. So these are nice memory T cells that have not recently seen SARS-CoV-2 antigens. Of course, during acute infection, those cells are doing a lot of different things because they're activated. But what's interesting is that in long COVID, we're seeing clearly a shift away from what we see in people who fully recover to T cells that are occupying these terminal effector or even activated states. And if you just look at like what these clusters represent, you see different activated cell clusters up here, this effector cluster in green down here, and then in blue, this memory cluster, which is almost totally depleted in the people with long COVID who have activation of their spike-specific T cells. And we see that these cells are enriched for a module of recent stimulation through the TCR. So of course, during acute infection, we know those cells are activated through the TCR by SARS-CoV-2. But also in long COVID, we see that spike-specific cells show signs of TCR activation, whereas in flu, we don't see that. And interestingly, if T cells are chronically stimulated, they can become exhausted, which is a state where they express a lot of inhibitory receptors and lose functionality. And if we look at some hallmarks of exhaustion, what you can see is that the cells from long COVID cluster more closely with the cells from acute infection. It's known that during acute activation, cells also can look exhausted, although they aren't. And different from people who fully recover in red. Now, they don't express every exhaustion-associated gene at a high level, but they do express some like TOX, CD101, BLIMP1, 2B4, et cetera. So we became interested in asking, these cells don't necessarily look completely exhausted, but is it possible that these spike-specific T cells are exhausted in some long COVID patients? So the definitive way to test this is to do functional assays and see if these cells are actually less functional than cells from people who fully recover that we suspect are not being chronically stimulated by SARS-CoV-2. So this is all very recent work that I'm happy to show you. where we, from a sub-cohort of our larger cohort that had these activation profiles, just to show you, we did additional profiling of T cell states and did assays to look at their ability to produce cytokines and proliferate, which are two hallmarks of T cell exhaustion. So first we took cells, we stained them immediately after thawing and just profiled their expression of exhaustion molecules. And what you can see is that, as we saw before, there's in blue acute, red convalescent, orange long COVID. You can see several hallmarks of activation that are increased in the long COVID samples, but also interestingly, this co-expression of 2B4 and TOX, two exhaustion-associated molecules, which is strongly enriched in the long COVID samples. Which really got, and this depletion of this memory signature with TCF1 and CD127. So we were really interested in seeing whether these cells could produce cytokines. So I'm wrapping up soon. But if we stimulate these cells for 6 hours and look at their ability to produce cytokines in response to spike peptides, what we can see is we can identify these SARS-CoV-2 specific cells. If we don't stimulate with spike peptide, they don't produce cytokines. And if we stimulate, we can see that in a recovered person, almost all of these cells, 9%, 7% here, can produce interferon gamma, and most of them TNF alpha. But in a person with long COVID here, we can see only about half of those cells are able to make interferon gamma. And if we quantify that across the cohort, we see a significant decrease in the ability to produce interferon gamma and TNF alpha in these people that with long COVID who have persistent activation and maybe exhaustion of their spike-specific T cells. And lastly, we also looked at the ability of these cells to proliferate. So classically, exhausted cells are poorly proliferative in response to the viral peptide. So for this, you can stain cells with cell trace violet, CTV, which is a purple dye. And then every time the cell divides, that dye will get diluted in half. And so you can track in flow cytometry how many times the cell has divided by the dilution of this CTV signal. And so we looked in people who recovered and people with long COVID. We can gate on these spike-specific T cells and see that if we don't stimulate with peptide, even after 4 days, these cells have not proliferated at all. They express high levels of CTV. And in a convalescent control, after 4 days of stimulation, we see this robust proliferation and a 5-fold expansion of these cells that are specific for spike. But in this person with long COVID, as an example, we see very poor proliferation and actually no expansion of these cells. If anything, they've gone down. And if we just look across the whole cohort at how many times these cells have divided, the cells from people with long COVID tend to have divided 0, 1, or 2 times in 4 days, whereas cells from people who recover have divided more like 4 or 5 times. And this is the division index showing a significant decrease in proliferation. And I won't go through this slide, but just to say that the proliferation and cytokine production are highly correlated with those activation markers that we've characterized in the hundreds of samples previously. So we actually do now think that this appears to be, although maybe not full-blown T cell exhaustion, we still have a lot to do to understand that. We do see evidence, compelling evidence, I think, that the cells that are activated in this subset of people with long COVID are actually showing diminished functionality and some signatures of T cell exhaustion. So just to say, in summary, we can use virus-specific T cells from blood as sensitive antigen biosensors to understand whether viral antigens are stimulating immune responses in tissues. We see possibly subsets of people with long COVID where not everybody has this activation of spike-specific T cells, but about 40% of people do. And in those people, we see real hallmarks of recent activation and exhaustion and diminished functionality, which provides possible therapeutic targets. But we have to do a lot more work to understand if these T cells are pathogenic or if you actually want them to be more potent and reinvigorated to clear the virus. So there's a lot more to do. We also hope to understand where these T cells are actually seeing this antigen. So with that, I'll thank everybody in our lab, my mentor, John Wary, Naomi, David, and Sophie who've helped with all this, and of course, PolyBio and the participants and all of our collaborators for their help supporting these projects.
Awesome, Mark. Every time you present, there's like a new next part of the story from your experiments that it's really interesting. So these kind of exhausted or differentially energized cells, it's very cool. All right. Okay, we have a little time for Q&A then. Let's do one. David, how is MMT different from transcutaneous? Yeah, transcranial magnetic stimulation?
Oh, yeah, great question. So MMT is different from TMS, transcranial magnetic stimulation, especially rTMS, which is typically the sort of treatment modality, in that the intensity of the magnetic fields being used are much, much lower. Now, rTMS works by using fairly high Tesla, like 1 to 2 Tesla magnetic fields to create a current that electrically stimulates the brain and neurons. In the case of MMT, we're dealing with 100,000th of a magnetic field intensity, and the low energy magnetic fields are not actually directly stimulating neurons, but what they're doing is they're triggering a process called mitohormesis, which actually provides slight strain to the mitochondria, just enough to trigger antioxidant and anti-inflammatory responses. So it doesn't trigger neural activity, which can cause cognitive exertion. And so we think it is much better tolerated than rTMS.
Got it. Really helpful. I even learned from that answer. Thanks, David. Okay. Mike, with the p-tau results, and I know they're still under consideration, like you guys are finalizing them, but someone asked with the ME/CFS patients, the pre-2018 ME/CFS patients, could they be neuroexhausted, and that might explain why you didn't find as much p-tau17 in them? Or do you think that exhaustion wouldn't really affect a marker like that?
You wouldn't expect exhaustion to be in play because, no brain is more exhausted than a full-blown Alzheimer's brain. And you wouldn't, if that were true, you would see that pattern in people with actual Alzheimer's, and you don't. You do see p-tau shedding, p-tau 217 shedding in full Alzheimer's.
Got it. Okay. That makes sense. Oh my gosh, I just want a question to go to Mark.
Yeah, we got to ask Mark something.
And so you can have what is your son's name, Mark?
Thomas.
Thomas. So maybe Thomas can answer. Thomas, hi. Are you going to join the team soon?
Yeah, he loves PolyBio. So
awesome.
We got to give him some work.
That's great, Thomas. Great. Okay. Well, we'll ask both of you a question. Are responses of SARS-CoV-2 N-specific CD8 T cells similar to those of Spike-specific CD8s? Besides compounding by vaccination, what do you think comparison of responses to N versus Spike-specific CD8s might tell us about long COVID pathogenesis and immune response?
Yeah, so that's a great question. We're working on developing better reagents for studying N-specific T cells and also other antigens from SARS-CoV-2, not just Spike and nucleocapsid. For the initial studies that we did look for those cells, but the reagents we had were not so good, so we can't make such definitive statements. But I think that's a really important question to understand. So far, what we've observed is mostly restricted to Spike-specific T cells. But I think that's going to be really important to understand. Is this full replication competent virus producing all of the viral antigens, or just certain viral antigens that are persisting somewhere stimulating these cells? So yes, important work to do.
Got it. Yep. Okay. And then let's see. Quickly. Did double negative AB T cells play a role, Mark?
So these would be like CD4, CD8 negative T cells, I assume, is the question that they're asking about. It's possible. We haven't looked at that at all. So in order to bind tetramer reagents, they have to, express either CD4 or CD8 to be able to interact with MHC. Those cells can be important in a lot of contexts, but we haven't studied it in long COVID yet.
Got it. Okay. All right. Well, with that, let's move on to our next session. And the first speaker in that session is Shannon Stott. She is a chair and professor of biomedical engineering at Boston University, and she'll be talking about ultrasensitive microfluidic detection of viral particles in long COVID blood.
Wonderful. Thank you very much, Amy. I really appreciate the introduction and the support from PolyBio. But I'm really excited to share with you our group's work of seeing what we can detect in the blood of long COVID patient samples. To give you a little bit of background about my group and what we try to do is we're largely a group of engineers where we work to see how we can create new microfluidic technologies that can improve patient care. I think everyone on this call would understand the benefit of a better biomarker assay to be able to understand what's happening for patients who are experiencing long COVID symptoms, both in terms of a diagnostic as well as matching patients to particular trials when they become available and monitoring overall outcomes. For our case, our particular area of specialty is looking at ultra-rare particles in various complex fluids, such as blood and plasma. And for us, we always ask the question, when a biomarker can't be found, is it the biology or is it the technology? And what our group tries to bring to bear when we're asking this question is we bring in our microfluidics expertise, but also our background in sample preservation so that if something is quite rare, we're able to make sure that those rare particles are preserved all the way through the process line. We also have experience in multiple biofluids. Today I'm going to talk about blood and plasma, but we certainly have multiple applications that are looking at urine, cerebrospinal fluid, as well as saliva, in addition to blood. We also have our own custom bioinformatic tools and pipelines, looking at how we can analyze RNA cargo. And then prior to moving to Boston University, I was faculty at Mass General Hospital for over 20 years. And so for all of the data that I'm showing you today was all done at Mass General's CLIA lab. We're really working to see that whatever platform we design can be quickly integrated into the clinical pipelines that we have. So with that, we've largely been a cancer biology lab. And so we've had many decades of trying to figure out how we can probe these complex fluids to pull up things that are very rare or in low abundance. And then when we do that, we also want to preserve the integrity of what we're isolating so that we can do interesting things downstream. One of the technologies that I want to talk to you about today is our herringbone microfluidic chip. I want to talk to you the 3 different ways that our herringbone chip works so that you can understand a little background about the technology and the time frame that we have. First, if you think about a traditional microfluidic technology, when you have any particles going through, there's no diffusion or motion of the particles. They follow in their laminar flow streamlines, entering in one entrance and then going out the exit. For us, we don't want that to be the motion of these particles because we want these particles to engage with the surface of the microfluidic chip as much as possible because we're coating the surface with something so that rare viral particles will stick to it. So the first thing that we do is we create these staggered herringbone mixers inside our device so that it creates this chaotic flow so that as particles or virus particles go through the chip, there's tens of thousands of interactions with the surface so that we increase our chances of being able to have that viral particle bind to the inner surface of the device. We also do antibody or protein engineering so that when we're coating that biomaterial onto the surface of the device, we want to make sure that our particles, and in this case particles that are very small, whether they're viral particles or extracellular vesicles, can stick and see that capture moiety, because often sometimes the surface charge of that surface will repel what we're trying to bind. And so we've done some antibody engineering where we remove our capture moiety further away from the surface so that we don't have that force field preventing the capture onto the surface. And then finally, we also have some novel nanotechnology on the surface of our device so that we also orient our capture protein so that everything's facing straight up and down so that we can have the best chance of capturing our viral particles or extracellular vesicles. So when we're thinking about an assay that can pull out intact viral particles, one of the things that we had to first ask is how are we going to pull these viral particles out? The first thing that we're doing is we're using a version of engineered ACE2 protein receptor traps. So these are something that Jim Wells' group at UCSF has developed, and they've graciously let us use for our technology. But we put those onto our biomaterial surface that's all inside our chip, where we simply flow through our blood, have those magic parts help so that we then have capture of the viral particles onto the surface of the chip. Here is a technician in our lab that's running this in the CLIA lab at Mass General, and here we're running 10 different samples at once. And the total time of this particular assay right now is about an hour for us to run it. So to see the comparison for how we do in terms of detecting virus against the existing technologies, we started by using the assay they were using in the CLIA lab to detect SARS-CoV-2 in acute COVID patient samples. So we looked to see if we can detect the virus in plasma using their traditional ddPCR technique versus that engineered ACE2 receptor trap and our microfluidic technology. So it's the same readout using ddPCR, which is a droplet PCR technology approach, and seeing can we detect the viral particles. And sure enough, when we were using our microfluidic chip, we saw that we were able to detect viral RNA that we were not able to detect using the traditional methods. We also could do this in blood or plasma. It didn't matter which one we used. And then we were also able to look at the captured particles on our chip. We also were very fortunate to receive some funding from NCATS at the time. Again, this was in the acute COVID patient setting where we were able to detect 3 viral particles in a full ml of blood. So best in class in terms of limit of detection, and these were blinded spike samples that we were sent. We ran them through our technology to be able to determine this limit of detection. We also could look at cross-reactivity. So in this case, if we put through the specific virus in the particle and we're trying to read out that particular virus, so not looking to see if it was SARS-CoV-2 RNA, but say flu RNA, we did see a little bit of cross-reactivity with some of them, but it was still at a 95% level. And then the specificity was near 99.9%, where we actually had the readout being SARS-CoV-2. When we did this in the acute patient setting, we were able to detect these viral particles in plasma at a 72% rate, saliva, as well as stool. But where the story takes a twist for the long COVID setting is when we were looking at serially monitoring these patients over their time with their acute infection, we were still detecting some viral signature many days out after their initial diagnosis. So in this case, we started to ask that question, could we detect viral particles in the blood of long COVID patient samples? And we were extremely grateful for PolyBio supporting our lab's work so that we could start asking this question. What I'd like to report to you today is some of our early outcomes from this work and from the support both of PolyBio, but also Link, in very carefully curating and sending to us a blinded set of samples that we're able to process through the technology that I just shared with you. So this is the cohort that we tested, and we've now completed goal 1, where we've used long COVID samples, convalescent control samples, and then mild acute COVID samples, and pre-pandemic control samples. And again, we were blinded to which samples were which. We ran them through our chip. We sent the results to the Link team, and then they unblinded us and told us what our outcomes were for these different cohorts. And then eventually, actually it's right now, we're going to be receiving the next set of samples where we're going to be looking at additional long COVID samples, as well as some samples that were at longer time frames from the initial time point that was sent to us. We also had improved a little bit of the sensitivity of our assay by moving to a new version of the Bio-Rad technology that we were using for our readout. I also want to acknowledge Dr. Roma Parikh, who is a postdoc in my lab, who came in, really embraced this assay, having not been involved with the previous work, and has really been an outstanding member of the lab pushing this work forward. But really probably what everyone's interested in is looking at what are we seeing and measuring in our long COVID patient samples that Link has sent us. So these are the results. And so this is a measurement of a droplet PCR readout, where the readout is a SARS-CoV-2 RNA readout. For the acute COVID patient samples, we have the highest viral load that we're measuring. When we look at the paste samples, slightly higher against a small cohort relative to the recovered long COVID patients and then to the pre-pandemic. We had set our limit of detection to 5 viral particles based on our prior knowledge. Again, we were blinded, so we set this limit of detection based on prior work. So we're continuing now that we know. We're going to be running additional samples, and then we may further refine where our assay is standing. So for our next steps, we are going to receive next week part 2 of the samples from the Link group. And so we're very grateful that they've identified these samples, and we're going to run them as soon as we receive them. And then we're also interested to look at, because we're seeing these measurements of viral RNA at these time points, really wanting to make sure we can answer that question accurately of what are we measuring when we're measuring this viral RNA? Is this viral RNA that's inside viral particles or potentially in other resources such as an extracellular vesicle? And then, of course, we're hoping to expand our cohort. So I want to first and foremost thank all the patients who've donated their blood during this time. We wouldn't be able to do any of our work if we didn't have these samples, as well as all of our colleagues, collaborators, and of course, PolyBio and Link. And thank you very much for your attention today.
Great, Shannon. Thanks very much. It's cool that you'll be sending that next, or getting that next batch of samples from Link soon. That's great. Okay, I'll introduce the next speaker, who's Frances An Lee. She's a professor of medicine in the Division of Pulmonary Allergy and Pulmonary and Allergy at Emory University, and she'll be talking about use of Mensa to identify SARS-CoV-2 persistence and viral reactivation in long COVID.
Well, thank you so much. I want to thank PolyBio for all their support for helping us with this, supporting us with this assay. I just want to give you some updates of where we are in terms of using it. These are my disclosures. We're trying to commercialize this assay, so I just want to make sure that that's you are aware of that. I think the whole concept, and in this meeting, everyone's been talking about the presence of, when you have primary infection or repeat infections, you get viral loads. But what happens during long COVID, or during long COVID or during these chronic phases, are there persistence of viruses? What I am is a plasma cell immunologist, so I study plasma cells, been doing that for about 25 years or so, and I'm interested in understanding how they come up, how we form long-lived cells, how we form short-lived cells. But what's very interesting is that during these viral infections, whenever you get infected or get exposed to different antigens, you get these plasma cells that come up in the blood compartment because they're being released, and then they go to a long-lived compartment, and then they kind of live there forever, and then your serum antibodies are all your exposures. But what we want to do is capitalize on the plasma cells that come up in the blood. I happen to call them antibody secreting cells, or ASCs, not to give it any name, but plasmablasts. And the question is, are they also released during long COVID, during infection-associated chronic conditions such as long COVID? How do we do this assay? We use this technology called Mensa that I've developed. What we do is we get a tube of blood, we remove all the historical antibodies, and then we basically culture the plasma cells that are in the blood because they're all newly generated, and we save their antibodies. So the historical record is from the serum, whereas the Mensa, which is the antibodies collected from these new plasma cells, basically are interrogated against whatever platform that you are interested in. So we asked the question, in long COVID, do you actually see them coming up? And what we did was we took a small number of patients in 2021. We ran an old assay and we said, wow, we could see some of these patients coming up positive for long COVID, whereas the serum, they start looking the same. So we also did the similar things with nucleocapsid. It was a smaller number. And so what we wanted to ask was, what happens to Mensa at different phases of the infection, vaccination? So does it really mean that there could be some evidence that there is a reservoir, there is some virus that's floating around that the body is seeing? And I don't need to try to convince this group that that's important. But when we do infections versus vaccinations, you can see that Mensas come up right after the infection. You can see it going up after vaccination because you're perturbing the immune system and the new ASCs are being generated. So you get that new antibody response, and you can see that here. And then if you get a repeat infection, this happened to be with Omicron or Delta, I think it was Omicron for this patient. The bottom line is you can see that the Mensa goes up, but the serum didn't change that dramatically. So I think this is just one way to think about it. The Mensa is something that's very acute, and whereas the serum is something that's very long-lived. And I think most people know that. Let me just show you some data, which we were excited when we looked at it during the acute infections, when we analyzed all the the acute Mensa infections. infections, you can see that with severe infections, the Mensa comes up very early as day 5. And you can see that here. So first of all, Mensa comes up early within the infection when you know that you had symptom onset and then diagnosed. And then a lot of it goes away by day 60. So after 60 to 300 or so, it really goes down. What's interesting is that during the acute phase, this first 30 days, that you can have severe infection coming up very early and very high, which was known. The antibody titers came up, and then you can see that in the mild diseases, it also came up at a later time point, like day 14, because it's a primary immune response. Whereas you can see here, this is what happens with serum. You see similar responses with the nucleocapsid. This was just looking at the vaccine responses that we had in individuals. And you can see here with the primary immunization, it comes up at around day 14, 12 to 14. And then with secondary immunizations, the peak is around day 6 to 7. And that's the traditional thing, approaches that we've seen. And it was just nice to see that we could recapitulate that. So Mensa is transient, serum is more persistent. So we took the 60 individuals, and these are all of their symptoms scores that we had with help of Max from the other groups in PolyBio, and we ran them against all the different known viruses that we had with linear peptides. We happened to test, a few of the patients just to see, is it just SARS-CoV-2 or are there other viruses that we need to be thinking about? And as many people have reported here, that we need to be thinking about some of the herpes viruses, some of those that may reactivate, such as EBV. So you can see EBV picks up a big chunk of a lot of these individuals here that basically were positive, but we saw some of the other herpes viruses. So we made a new assay for not just SARS-CoV-2, but we also added EBV, CMV, and HSV. So this is what the data looks like. And we just reanalyzed this data just to look at it more carefully to make sure all the controls are in the right place. And what we find is that in serum, there's no difference that we can see from the COVID recovered versus the long COVID, but you can see in Mensa that it's very different. And this is the original study that we did with the first 60 patients that we had. What we found is about 42% were positive in the long COVID group for SARS-CoV-2. And you can see the herpes viruses were picking up about another 60%. And then that was about a total of 70% when we reanalyze this data. So differences that we can see. You can see also the COVID recovered that there may be reactivation in some of the individuals with some of the herpes viruses. And there was one patient that we found when we reanalyzed the data that was positive for RBD. So now I just want to focus on the study that we've been doing with PolyBio. What we've done is we've run our assay transporting all these live cells to us overnight in temperature regulated boxes. And then what we've been doing is running each one of their blood samples in their study, which is the tenofovir plus I'm not going to say this correctly, the TDF versus the maraviroc and versus placebo. So those are the 3 arms of the study. And the bottom line is they were looking at primary outcomes, which was basically to this 90 and 180-day time point for clinical outcomes, and had other secondary and had other blood biomarkers to look at that. And we were one of those blood biomarkers that they were looking at. So let me just show you some preliminary data. So we've so far collected 109 patients or samples from this study. They're supposed to have 90 patients in each arm. It's supposed to have an equal number in each arm, TDF, the maraviroc versus the placebo. And then what we were running is the serum and the Mensa on them. So far, we have 109 samples, and we have run 81 of these samples, blood and serum. I'm going to show you data from 33 baseline, 25 day 90, and 180 at 22. At 180 days, we had 22 samples. So you can see here from the baseline samples, we had about 33% of them positive. So this is similar, but not exactly the same as before, when we had about 42% that was positive for SARS-CoV-2. The EBV, CMV, and HSV responses are much lower here in this study that we had so far. We only have 33 patients. We're hoping hoping to get 90, but I don't know if that's going to be up to full recruitment in terms of that. But this is what the baseline data look like. Let me just show you a little bit about the longitudinal study. So this is all the samples that we have in the Mensa. And we run it against, you can see here, the wild type Mensas in terms of the RBD, nucleocapsid, S1, S2. And then what we do is, since it's an antibody assay, we can also run the Omicron and we run Delta. And we run those separately because there's a lot of cross-reactivity and we try to pick up those responses. Simultaneously, in one assay with the wild type, we also run EBV, CMV, the two different strains, the AD, I can't remember what this is, 160, and the Merlin and HSV2. So this is what we're running for the whole assay, and this is what all the data looks like. And you can see here in the serum, most people are positive for SARS-CoV-2, most are positive for EBV, CMV, it's still about 44%, and you can see in terms of HSV around 44% in terms of the positivity. So let me just tell you a little summary, because we have a total samples from 19 of these patients where we have a full baseline, day 90, and 180 from day 180. And the bottom line is we see some patients that have positive Mensas that become negative, what we might expect if they were on treatment. We're blinded, so we don't know. We also had a bunch of patients who were negative and stayed negative. There were about 8 of those. And then there were some patients who were negative, who were positive and stayed positive. So for 3 of those in terms of what we saw. And then there were others that were negative at the beginning and then eventually became positive. There were 5 patients that we had to exclude. And I think because we weren't getting follow-up samples, or they got COVID, or there were some other issues that happened with the individuals. And then 9 of the patients, we are still waiting further follow-up draws. So this is what a positive becoming negative looks like. Typically, they're positive for one of these antigens. So this is by day now. And these are all the antigens on this axis. And you can see here they're positive on day at the baseline. Some of them become negative by day 90, and then they become negative at 180. You can see that there were about 8 patients that were negative all throughout, no matter what antigens that we tested for in the Mensa. Now, in the serums, they're positive for many of these things. This is what a patient looks like if it's positive and they stay positive. So you can see the 3 individuals here, they were positive for this RBD in the wild type, this RBD in Omicron, and this RBD in Delta. And some of this one, I think there was like low bead count, so we need to go and repeat some of these assays. The other one here was a patient that was negative to start and then became positive, but we had 4 of those. So I'm totally blinded. I don't know who is whom. And so what we're trying to do is run all the assays, and then we would love to see in terms of if our assays are helpful in picking up some of these individuals who may be responsive. So just to give you a little clue in terms of other studies that we've been doing. So we have a RECOVER study that we have where we have 100 patients that we've recruited recently at the same time as the PolyBio was being recruited last year and this year. And what's really interesting is that the numbers are kind of similar. We see that SARS-CoV-2 in this cohort of 77 individuals that we actually ran was about 35% positive. Any of the HSVs were also much lower than expected. So this was also interesting, very similar to the responses that we were seeing in the baseline samples that we saw in the Mount Sinai study. So this is just what all the data looks like. And then, so in comparison to the ones from the original study in 2021 to 2024, there may be some things that may be slightly changing, or in terms of the long COVID patients that we are seeing. And we have still another 200 from before 2024, during that whole period where we froze all of the Mensas. And so we're going to run those. And then we also are doing a study with Mike Peluso with the LINK study to compare fresh and frozen, because the way you can do these assays, I think, on frozen samples is what we've had when we did it with the UCS Link group, and in terms of the samples that we've gotten, but you have to just modify the assay to be a little different to make it equivalent. So we've been working on those, and we're just about finished with them. And so we're happy to share some of that data. So it depends on how well you freeze the cells. So as Shannon was saying in just the previous talk, how important it is for sample preservation. but the UCF samples look great. So this is just in summary, more to come. These are just some of the patients that we saw, that some of them, the positive menses became negative, and some of them that were negative stayed negative, and some of them were positive. I think this may be, it's interesting, it divided up into 3 different groups, similar numbers. I don't know if they are from the different groups, and so time will tell, and we'll have to put that all together. So I want to thank all the people, especially Natalie, Andrea, and Rachel from my group, Rachel, who's been really leading the study in our group. She's been a fantastic technician, but really functioning at a higher level. And then especially with Mike and Amy and PolyBio with all the help that they've given us. So I want to stop there.
Great,
Sorry,
Frances.
I think I went over.
Oh, it's okay. We'll look forward to unblinding that trial for sure. It was very interesting. So that's great. Okay, I'll introduce the next speaker. It's Michael Peluso. He's an associate professor of medicine in the Division of HIV, Infectious Disease, and Global Medicine at UCSF. And this is exciting because Michael is actually going to talk for 20 minutes. He's going to talk first about the VIPER program and then about time ME/CFS study also happening at UCSF.
Okay, can you hear me?
Yep.
Okay, great. Hey, everybody. I am excited to be here. It's good to see everyone. I'm going to begin by talking about VIPER, which I'm super excited about because we're able to talk about VIPER in a little bit more of a concrete way now. We're very grateful that the program, the full program, was funded by PolyBio a couple of months ago, and we're getting started. So I'll begin by just providing an overview of why I think that VIPER is so badly needed and continues to be so badly needed now in 2026. What we've seen over the last 6 years is a lot of important foundational work in different laboratories developing assays that can eventually be developed, I think and hope, into diagnostic tests for people with long COVID. But what has happened over the last 6 years without VIPER is, despite all of this being great work, it's really happened in silos. So Lab 1 works with cohort 1 and develops biomarker or test number 1. Lab 4 works with cohort 4, creates biomarker 4. And ultimately, there's no real consensus on how these different measurements perform relative to each other. There's not really an ability to merge data. We often don't have the same level of detail of who is being studied across different cohorts. And this is a problem. And so the vision for VIPER is basically to correct that problem, to have a single unified cohort where the specimens are collected with great rigor in people who have been phenotyped with great rigor. The specimens are provided in a blinded manner to all of the collaborating labs who, instead of working in silos, are really working as part of a unified team and know what is going on within the consortium. The data is generated in a blinded manner, as you just heard about, shared back with a central data repository that leads the analysis, and then hopefully we figure out how each of these assays can be iterated and improved and come to some consensus on what the optimal biomarker or combination of biomarkers could be. And so this is beginning with a focus on markers and diagnostics for viral persistence, but I think it's obvious how this could be expanded beyond that. So the primary goal of VIPER is to validate non-invasive, scalable biomarkers of SARS-CoV-2 persistence to help us diagnose long COVID, guide treatment, and accelerate therapeutics. There are 3 sort of sub-objectives here. The first is a validation objective, and that is really to look across all of the different compartments that we can access in people with and without long COVID to understand how they relate to each other, and in particular, anchoring some of the less invasive measurements on what we think is kind of the current gold standard, which is tissue-based measurements. And I'll talk a little bit about that. The second is to address this issue that Yoon in the talk she just gave kind of alluded to, which is that there can be variability over time in many of these assays. So people are positive at the beginning of their participation in a research study, and then they become negative, They could start negative, become positive. They can kind of bounce around. We hope that that is related to treatment if it's a clinical trial, but even in observational studies where we don't see an intervention happening, we do see that variability happening. And so what we really want to understand is on a granular level, week to week or day to day, how consistent are these measurements within a person? That could be related to an assay issue, or it could be related to the biology of this if people are intermittently kind of spilling over spike protein into their blood, for example. And then the third objective is to understand the impact of different therapeutics or treatments. There are three sort of big picture components to VIPER projects. I'll just touch on these briefly. So when we're talking about VIPER going forward, we're actually really talking about item 3, which is the prospective VIPER cohort, where we will enroll people, do this detailed phenotyping, and collect really lots of biospecimens on a very intensive schedule. But we're fortunate from our relationships and what we've done over the last 6 years to have 2 other really important components of this. So the first is our ability to develop and distribute what I refer to as analytic standards. This is going to be in collaboration with the blood bank in San Francisco, where they actually have specimens, large volumes of specimens from people with acute COVID who happen to donate blood before knowing that they had acute COVID. And we are able to, do serial dilutions and provide plasma with a known quantity of SARS-CoV-2 to VIPER collaborating labs. So that's going to be really important in understanding the performance of these assays. And then the second thing that's really important is we're able through VIPER to leverage what we've done in LINC and use our existing LINC biobank, which is not collected with this level of intensity as the prospective cohort will be, but there are a lot of samples available. And that is going to allow us to really jumpstart the work that is happening in the VIPER labs. I just want to, acknowledge the team. So we've developed now our clinical operations team, which is led by Lynn, who's the program manager for VIPER, Emily, who's the program manager for LINC. And then we have 2 experienced CRCs, Antonio and Emily, who will begin with the program when it starts next month. We also established the scientific advisory board for VIPER, which you can see here. It's comprised of leaders in the field who bring various perspectives on basic science, immunology, tissue biology, clinical science, broad sort of sense of what is going on in programs outside of LINC and Recover to make sure that we are sort of aligned and filling the important gaps. Mike, who's representing PolyBio, and then Ezra, who's a person with lived experience who has experience also participating in research programs like this. This SAB met for the first time this week. They are going to provide input on the design and goals of the studies. They will review the potential collaborating labs and the data that comes in from those labs and provide an annual report. And so this is going to be really important in sort of guiding our work externally. I won't go into lots of detail here, but we have a basic schedule of events that is lined up. We're really treating this like a clinical trial, even though it is an observational study. So that's sort of the level of rigor and intensity that we're trying to bring to this. Everything in the study is going to be anchored on the gut biopsy day. So that's sort of like anchoring on an intervention in a clinical trial. And we're going to make sure that we have specimens collected at precise times and processed in precise ways before that procedure, and then for at least 3 weeks following that procedure. We're going to enroll adults with confirmed SARS-CoV-2 who are either recovered or have long COVID. We're excluding people with recent vaccines and recent reinfections. They can enroll later. And because this is really anchored on the tissue procedures, people who want to participate in this first round of VIPER need to be willing to undergo the GI procedures. We're planning to enroll about 100 people with long COVID and 50 people who've recovered. And all of these people will, be based in the Bay Area and be coming, through the LINC program. I'll just mention that, as part of this, we are doing deep phenotyping of all of the participants. So we're using our standard link assessments. We're also going to use a series of validated instruments to report on symptom domains. This does not determine who is and is not eligible, but it will help us to understand and describe the population. And so we're finalizing the assessments that'll be part of this now. What we're planning to do with the biospecimen collection is to really lean in on the gut collections, which we've been able to develop that program with PolyBio funding over the last 3 years. Everybody will get a colonoscopy, and most will get an upper endoscopy. We're collecting a large number of pieces and processing them in a way that both we can work with them here, but also they can be distributed. And I think the point I really want to emphasize is that there's a lot of specimens collected as part of this per person in a concentrated period of time. So we estimate that each participant in the study will, in the end, have over 400 little aliquots in the freezer related to these biospecimens. And you can imagine the scale of the work that can support. As I alluded to, we're really anchoring on the tissue biology. I think Tim is speaking next on some of the exciting findings from our gut biopsy program. So Tim will continue to work with these specimens, but other laboratories will also be able to access VIPR specimens and work with the samples. And so we'll actually be able to look across different laboratory modalities. And then, as you heard in the last 2 talks, we've actually begun under the umbrella of Link and VIPR to share biospecimens with different collaborating labs to kind of do that foundational work to figure out which assays are looking promising and how they might be performing. And so you heard from Shannon and from Yoon. You'll hear from Tim. You heard from Mark, who works with John. And that's exciting. It's allowed us to basically develop all of these relationships and get samples into the hands of the laboratory collaborators without having to wait for the prospective component of this. So that's been quite successful. So last slide, we got our notice of award a couple of months ago. We got IRB approval for the scaled up program within a month of the notice of award. We actually screened our first participant for the prospective cohort this week, and we're planning to identify the first round of labs that will participate in VIPR in collaboration with the Scientific Advisory Board over the summer. And so those labs will begin to receive the prospective samples in the fall. And then this has actually been sort of a catalyst for a lot of discussions about how a program like this can sort of extend its reach. And I think what we have heard is that there's a lot of interest in expanding VIPR, using the foundation of the PolyBio funds, expanding this to kind of public-private partnerships. We've had a lot of discussions, with government agencies that are potentially interested in leveraging the cohort, and also expanding this beyond the initial scientific question focused on viral persistence to all of the other mechanisms of long COVID. And so we're very excited for the potential to further expand the program beyond what we'll do over the next couple of years. That is my talk on VIPR. I will switch now and talk about CHIME. Okay. So CHIME is our basically our observational program for ME/CFS that is leveraging all of the experience that we have developed in long COVID over the last 6 years to apply that to pre-COVID ME/CFS. And the rationale for this was articulated in this paper that Steve and I wrote with Maureen Hansen from Cornell a few years ago, arguing that it's really important scientifically and sort of strategically to leverage the progress that we have made and the resources we have developed in studying long COVID to understanding other non-long COVID infection-driven chronic illnesses. And Maureen actually had this beautiful quote in that paper where she wrote, "The scientific community now has far more tools at its disposal to study ME/CFS, some that could not have even been imagined 40 years ago." And so that, I think, was the inspiration for this program. The other inspiration was this foundational work done by John Chia down in LA, looking at viral persistence in pre-COVID ME/CFS. These are studies that were done over the last 2 decades, identifying evidence of enterovirus persistence in the GI tract of a subset of patients with ME/CFS. I think these findings are incredibly provocative, and that the technologies that we've developed in the long COVID program really need to be applied to sort of confirm and extend these findings. So that was the sort of inspiration for QIIME. I'll now sort of show you how QIIME is set up. And I really want to credit the operations team here, especially Beatrice and Brent, who've really led the development of the program. And then we're fortunate to have Yuki, a research coordinator, joining the program next month to help scale up. So the initial phase of QIIME participation is all done remotely. An individual signs an informed consent form. They complete a screening survey. We get sort of detailed screening information and an interview with Brent and Beatrice and Yuki, and then we initiate a medical records request to gather all of the participants' medical records for review. As that happens, we assemble a de-identified summary of the patient's medical history. And that it's presented to an adjudication committee, which I'll describe on the next slide. The adjudication committee ensures that the participant meets all of the inclusion criteria, specifically that they also meet the 2015 IOM ME/CFS criteria, which is our main criterion for eligibility. They also review whether participants meet other criteria, but that does not determine eligibility. And then provide guidance on whether additional testing needs to be done before someone is deemed eligible. Once a participant is recommended for enrollment by the adjudication committee, we conduct an in-person baseline visit with a series of questionnaires, collection of biological specimens, and objective testing. And then the goal of all of this is really to get people to the advanced procedures, looking at tissue biology, which I mentioned before. Our advisory board is amazing. They are shown here. They represent a variety of different perspectives and experiences with ME/CFS. We've been meeting monthly since January. And then, as I mentioned, at each meeting, about 4 de-identified patient profiles are presented by our team to the advisory board. They sort of review things in detail, provide a lot of commentary and discussion, and make a recommendation on the eligibility of each of the participants. And we've had 6 meetings so far, including one 2 hours ago. I've actually learned a great deal from these meetings. They're terrific. So once a participant is approved by the advisory board, they are scheduled for an in-person visit. The in-person visit is quite intense. We prepare people for this. We allow breaks as much as possible throughout the visit. So a participant comes in, they get sort of standard clinical measurements. We do a series of surveys that are divided up over the course of the visit. These were all things that went through the advisory board and actually got additional feedback from the patient community. There's biospecimen collection of blood and saliva early in the visit. There are some functional tests, including hand grip, a neurocognitive test that is computer-based, and an active stand test. And then the visit closes out and the next visit is scheduled. There's also a variety of clinical labs. And then, the most important thing is that in addition to this, a great deal of specimens go to the biobank. There's also remote questionnaires that participants can complete in sort of a discontinuous way at their own pace. And so these are things that, are frequently asked for by peer reviewers or come up in other studies. And so participants are also kind of completing these in the background, but not at the in-person visit. And then, as I mentioned, the goal of all of this is to get people to 2 specialized procedures. which is our gut biopsy program represented here on the left, and our imaging program represented on the right. The hope is that the first gut biopsy will happen next month. So where we're at with this is actually we're quite overwhelmed. We didn't quite expect the level of interest that we have gotten. So over 130 people have contacted us over the course of the last 6 months. 69 people have proceeded to completing the interest form. We've consented 30. And 29 of those 30 have completed the screening surveys. The main limitation, frankly, has been our bandwidth. And so that's why we're starting to increase our staffing. The advisory board began in January. In-person activities began in March. And then the procedures are going to begin next month in June. And so I expect if I present on this in the fall symposium, I'll be able to provide an overview of who we have enrolled to date and what they look like clinically and what has happened with their specimens. Ultimately, those specimens are planned to be used across the whole PolyBio network. Some of these faces here that you know, we're beginning a project now with Charles Chu at UCSF using his metagenomics pipeline to study these samples. And this will be something for more discussion next year. So I will end there, and I think I did both of those in 20 minutes, and look forward to the Q&A later. Thanks.
Great. So excited with these programs. They're really coming along. Okay, now we have Tim Henrich, your colleague, who is a professor of medicine in the Division of Experimental Medicine at UCSF. And Tim will be talking about characterizing SARS-CoV-2 tissue reservoirs in long COVID. And Tim, it's okay if you want to speak through part of the Q&A time. There's no problem.
Okay, sure. Thank you for having us on. I'm just going to share my screen here. Okay, so Michael had an excellent introduction to many of the clinical activities that's ongoing here at UCSF. And Michael's really been driving this amazing clinical show of getting these cohorts in and getting participants involved and really making a lot of this work just amazing and able to happen. My lab, so I'm one of the PIs of the LINK cohort here, as mentioned. And my group has been specifically kind of spearheading the pathobiology of tissue. So, for example, what types of biological drivers, or what are the prime drivers of long COVID, which we now almost definitely believe is a tissue-based disease. And so that's what we've been focusing on through LINK. Also through some recover samples and other collaborations, and as well as in some of the clinical trials that are ongoing here and elsewhere. So one of the things I do want to say is that what the tissue study has led us to believe, that we really do believe that SARS-CoV-2 persistence is still a driver of long COVID, even though we can't detect SARS-CoV-2 in every sample, especially in rigorous studies that have a lot of controls for pre-COVID samples, et cetera. But a lot of what we're seeing, and as I'll hopefully show you in the next 8 or 9 minutes or so pretty quickly, is that virus, it makes a lot of sense. And I think what we've actually developed through our gut biopsy program and some of our other tissue programs is that we now have a working model for what we feel the root driver for long COVID is. And we've even started to design clinical trials and implemented a clinical trial based on the pathobiology that we have observed. So, viral persistence can be either active replication, you can have viral activity, double-stranded RNAs, you can have proteins being made without productive viral infection in terms of frank replication, or you could have inactive either RNA, residual RNAs protected somehow in the intracellular space, or proteins as well, which may be more stable. So all of these are potentially possible. I want to talk briefly about our gut biopsy program first. To date, actually, this is a little bit outdated, but I think we processed more than 120 individual participants, initially focusing on colorectal sampling because of just ease for the participants and able to get a large number of folks. And I will be talking initially in our first study here of 56 colorectal samples from 43 people with documented long COVID through LINK, as well as 13 matched fully recovered controls. And because we have this large infrastructure that Michael has built, we've really been able to find controls that are well matched, which I think are really, really important in understanding etiology of long COVID. So we're actually now focusing our future program on response and clinical trials. for example, the Outsmart trial of antibody, in our Encitravir antiviral trial as well, where we were able to actually biopsy a large subsection of both people on drug and placebo before and after treatment, which has really been helpful, and we really look forward to seeing how this will change given what we're going to show you today about drivers of long COVID. So we published this about a year and a half ago, showing that in a kind of small study of a limited number of our first gut biopsy folks, that we actually can see persistence, especially even in the lamina propria, not just the epithelium, which has been known in acute COVID, as was shown many years ago when virus first emerged, but epithelium turns over every week or two, and so or several few days, depending on the activity in the mucosal of the gut. And so with that, we were seeing RNA, both single and double-stranded, and some within macrophages, within the lamina propria of the gut. But this was kind of what our appetite was. Ooh, what's going on? Can we really do a deeper dive? So this is a data. Apologize for a little bit of the visual confusion here, but what you can see is basically what we call a heat map. So everything that's pink or red is essentially a positive hit, which means we can detect virus somehow in the tissues. And what I've organized them by two different ways. So in green are fully recovered controls, and in white are participants with long COVID. And you can see here that when we order it by days from first infection, so their initial SARS-CoV-2 infection through link, starting at about 5.5 or 6 months after initial symptoms, all the way up to 4.5 years. So we have sampled people all across, everywhere from pre-Omicron, pre-vaccine, to post-vaccine, pre-Omicron, to Omicron, post-vaccine, to reinfection era. And so we get this nice timeline longitudinally over what we're seeing. And what you can see is actually we do find virus in the gut tissue. We see it very rarely by qPCR, very, very difficult to detect. nCounter, unfortunately, does not work for us. And I think a lot of the previous literature has been some assays, sampling issues, and also oversaturation in those assays. But what we see is using RNAscope, using chromogenic to avoid a lot of fluorescent background noise that we can see in gut tissue, as well as overlap from other bright signals from other fluorescent co-stains, which can confound actually viral persistence. So we actually moved all the way to a chromogenic staining, which we know is very clean, and have looked at that in not just the gut tissues, but our autopsy tissues as well. And so we look for both single-stranded and double-stranded RNA, and we do see that. Plus, we use something called metagenomic sequencing, which is a very deep shotgun, unbiased approach for sequencing, and we actually pre-enrich for SARS-CoV-2 and other viruses like EBV before we run these, and we actually can. It's an extremely sensitive way of looking at viral nucleic acids and viral RNA in this case across patients. So we do see it around 20-25% of the time. We will see detection all through actually the timing all the way up until 3-4 years after initial infection. That said, we see very little in the blood using the same technique. So using metagenomic sequencing, PCR-based methods, nCounter-based methods, we just don't see a lot. Although we have actually seen what we think is definitive spike protein, at least one of the individuals that had positive gut as well, using the MSD assay for spike protein. This is ancestral spike is what it's tuned for. It may have a reaction elsewhere. Now, when we look at this now from time to most recent infection, because these people are followed longitudinally in link, and so we know not just when they were initially infected, but we follow them, they get tested, and we clinical surveillance, et cetera. So we know when people have reinfection, or at least clinically obvious reinfection. And when we look at that, we actually see a bimodal distribution where we see people that have a lot of persistence by different methods, anywhere between 4 and 8 months after their most recent infection. So sometimes their initial infection in this cohort, but also sometimes after their second or third infection. So there's this cluster of folks where we see more viral persistence, more closer to the time of reinfection. But we also have these individuals, as you can see on the bottom right, that have detection from one or more of these methods, even 4 years after initial infection without known reinfection, despite being followed very closely. It's still possible people had subclinical infection, of course. So what's nice thing about metagenomic sequencing is we get viral sequence fragments from this, and we can align those to the genome to see where we are seeing the sequences. And what we are seeing here is most of them are from the ORF1AB, which is the largest part of the gene of SARS-CoV-2. We also see ORF3A. And unfortunately for the gut, we were unable to find any spike sequences. So, hard to know exactly what the strain is, et cetera. But we are able to kind of align these sequences that we get from metagenomics onto the genome. Well, just to show you what we're seeing visually, now this is our chromogenic pattern. We have to do this very carefully, by the way. We need to use pre-COVID samples. That's especially critical when you have fluorescent staining, where there can be a lot of either not just background noise like macrophages can autofluoresce, but also if you're using stains that have bright, for example, cell surface marker can often bleed in and look like viral signal at times. So we've actually gone to this chromogenic, and we always do run simultaneous pre-COVID samples to kind of make sure we're not seeing stuff that we shouldn't be seeing here. But what we do see is we see a mix of infection. We see both spike in green and also double-stranded RNA in pink here in different parts of the tissue. So we see it in the epithelium. These are the kind of the outer brush border of the gut that really interact with the intralumen. And we see both double-stranded and single-stranded in those areas. And we also see mostly single-stranded in the lamina propria. And what we think we're seeing is actually in myeloid immune cells like macrophages within these compartments. And so we actually see it in both areas, epithelium, which turns over every several days to week, to lamina propria, where you can have long-lived myeloid immune cells that persist for a long period of time, suggesting there may be some turnover or even replication in some of these individuals. Again, hard to prove definitively. So when we also look at the human gene response, this is using actually a targeted RNA array, looking at about 800 to 900 genes that we've also modified to look at viral responses to infection. We actually see that there is a dramatic clustering of people with long COVID in blue versus fully recovered up in orange on the heat map on the right. And the individual genes are, each of the columns in this heat map aren't important, but it shows that there's very tight clustering of our long COVID cohort, which means that the human response or the host response in long COVID in the gut is very distinct in people that have long COVID versus those that don't. I put the asterisks up there to show people who had viral detection in our long COVID cohort. And what's interesting about this is if you look to see overall what are the trends within these bulk tissue samples, we see that a lot of these pathways for viral response are actually downregulated, which is a bit surprising when we first saw this. So things like MHC class antigen presentation, this is how infected cells tell the rest of the immune system that they're infected and that they need to be targeted for clearance. We see reductions in genes associated with phagocytosis and cytotoxicity, cell migration in these, which suggests to us that there may be an insufficient, although there is viral sensing, there is insufficient immune response, adaptive and other immune response, to essentially clear out those residual infected cells, which could lead to chronic inflammation. When you look at unbiased data, so looking at RNA-seq data from 30 to 60,000 different human host response RNAs, especially focus, we're able to see that exact same clustering. So in where it says FR fully recovered, there's a very tight cluster. LC greater than 1 means anybody with a long COVID symptom 1 or more. LC5 means they have greater than 5 symptoms, so a lot of concurrent symptoms. But as you can see, there's, again, tight clustering between long COVID and non-long COVID, but there are different types of biological endotypes, or these different columns within long COVID. So there's different kind of biological cassettes or different processes in different people. But what we don't see, these do not correlate necessarily with the clinical symptoms or the clinical phenotypes. So we see that the clinical phenotypes really don't predict what we're going to see in terms of the biology in the tissue, which is what's driving long COVID. When you look at the peripheral blood, you don't see this at all. You see very little activity. These are blood samples taken from the same time that they had their gut biopsies, from the same participants, controlled, matched, et cetera. And using the same assays, we just don't see the viral persistence, and we just don't see those dysregulated immune responses that we do in the tissue. Well, we've done a lot deeper dive now as well on a single cell resolution, where we've done single cell RNA-seq in a subset of individuals with long COVID versus matched fully recovered controls. And we did actually enrich these for people that had viral persistence from our bulk data. And what we saw initially, if you look on the left and right, it looks like this kind of island map, and each one of these colored clusters represents a different type of cell that we're picking up in the gut. After we have to collagenase treat it, we break it up, and then we encapsulate them, and we can do sequencing on a single cell level. But there was no difference in this frequency of the clusters between our well-controlled, fully recovered, and our long COVID groups, suggesting there's not a huge difference in the makeups of the cells that are in that tissue. We also saw the same thing with high-dimensional spectral flow cytometry that we run on these. There wasn't a huge difference in the frequency of cells or the phenotype of those cells, although we did see there were more classical monocytes in people with long COVID. What was interesting, though, we also got T-cell receptor sequencing data. This is the CDR3 region, which kind of is the sequence region of the T-cell receptor that is specific to its antigen that it wants to bind to. And we actually did see clonal expansion of certain T-cell clones in people with long COVID, although one of them was actually there before COVID as well. And these are all sequences, by the way, that have been associated with SARS-CoV-2 infection over the past 5 or 6 years through the literature. So we are seeing this clonal expansion of T cells, even though the whole makeup of when you look as a whole, we don't see that difference. Well, when we look at the gene processes or what's happening in these individual cell clusters, it gives us a huge amount of information. So for example, goblet cells and these colonic, what are called TACs or TAC, don't worry what that means. These are basically cells that become the precursors of enterocytes and other cells that make up the epithelium in the gut. And so what we know about these goblet cells and these that have mucus and colonic TACs is that they've been shown to be infected during acute infection. And they also express ACE2. And so it makes sense that in acute infection, we know this happens. But what we're seeing here, and we know this from our RNA-scope data, that there is infection in the areas where these cells reside, is that they are heavily turned on for viral sensing. Their interferon responses are up, TNF signaling, NF-kappa B signaling, all these other things for all the gene pathway nerds out there. These cells look like they're infected. They are screaming infection. They are sensing this virus. Some of them upregulate RIG-I, toll-like receptors, et cetera. And so they're saying, hey, yikes, this is not good. We also see this in macrophages, and because we see infection in macrophages as well, in these tissues. And we see the same thing. They're screaming with all these different innate immune response pathways and inflammatory pathways. But what's interesting is the macrophages are actually the least number of cells. Going back up here, the macrophage is just a very small cluster compared to the plasma cells, which are these terminal cells that B cells turn into that make antibodies in the gut, primarily IgA. And what we saw actually in these plasma cells is that they're downregulating a lot of these pathways. So they're not screaming for infection. They're not screaming, hey, there's infection, I need to do my thing, there needs to be antibodies. And it turns out that plasma cells may play a very immunoregulatory or kind of role in gut mucosal immunity, these IgA-producing plasma cells. And so that was recently shown outside of the SARS-CoV-2 literature, but may be responsible for some of the downregulation of the immune responses that are preventing viral clearance that we see in the bulk data. Interesting enough, we actually don't observe any. The T cells and B cells are boring. They're just not doing much in the colon in the setting of long COVID versus non-long COVID. It's really myeloid immune cells and then these epithelial-based cells in the gut and the plasma cells that seem to be regulating most of the leading to most of the dysregulation. Well, we've also done single-cell spatial omics, so we can do single-cell RNA-seq, but actually in space. And to just show you here, we're on the right and the left where we have some of our double-stranded RNA. We can superimpose that with tissues that we now can do direct RNA sequencing within each individual cell within space on the slide. This experiment is now done. And just to kind of show you, we can also look at clusters of the different types of cells that we see on the slide on the upper left. It's just like this, again, like a country map where each one of those clusters is a different cell type that we're seeing defined by these different dimensional UMAP variables. But what's interesting, you can actually now map these directly onto space, which you can't do with just regular single-cell RNA-seq. And for example, you can see these different cell clusters, one that's very much on the epithelial border. Those are turned out to be a type of epithelial cell. And then you have other things that are more in the lamina propria or in the basal crypts, for example. And so we can actually localize where these different cells are. And for the matter of time, this is my last slide on the gut, is that to summarize, it actually shows what we're seeing with the single-cell RNA-seq data, that these cells that we know become infected or can become infected, they're not necessarily infected in all the tissues that we're seeing, but they can become infected, or the macrophages, myeloid immune cells, are kind of screaming for help because they are sensing virus. but they're also highly dysregulated. We're seeing, for example, early data showing that there's downregulation of cell trafficking markers, cytokines that we see in the bulk data, antigen presentation or response, et cetera. So we see the virus is causing cells to trigger an antiviral response, but it's heavily dysregulated, and we don't see that cytotoxic killing that we should be in viral infected cells. So that's what we see with gut. I should say with the bone marrow program, we've also been looking at the bone marrow. Why? And we actually do bone marrow biopsies. We do aspirates to get a deep dive. We actually saw a lot of bone marrow signal on some of our PET imaging scans in the past, and we know that certain immune precursors and immune regulatory and modulation can happen in the bone marrow that you can't catch in peripheral blood or even the tissue. And so we've been very interested in looking at this. I won't spare you, but we certainly find SARS-CoV-2 in people, more so in people with long COVID, although I think we also see a bit in people that are fully recovered. Like the gut, it's less common. And we're doing that same exact deep dive that I just showed you with the gut to look at that. So just going to put the mechanistic model of what we think is driving long COVID. And if you think about it from a viral persistence standpoint, that you have infection of various different epithelial cells or cells within the epithelium or near the epithelium. And you also have myeloid immune cells, some of which, by the way, in the gut act like microglia and actually control brain-gut axes. And so that's really interesting as well. Could explain a lot of peripheral symptoms, even when there's just gut persistence. And so we see this local inflammation, dysregulation, and then you have other cells that just either don't seem to be involved, and then maybe you have other cells that are actually actively trying to shut things down, and maybe doing that too well in the gut. So we think that virus gets into the tissue, the virus is sensed, there is an immune response. The virus persists, however, because it's able to evade those cytotoxic responses and persist to cause chronic inflammation. Downstream things like autoimmunity, spike protein persistence leading to endothelialitis or clot formations, or all of those could actually be secondary to ongoing viral persistence. But again, it is our hypothesis. We don't see it in every patient, but we're taking tiny pieces of gut from 21 feet of a very large tube. And so, there may be bystander effects as well, et cetera. So I just want to finish there. It's a tissue-based disease. We learn so much more in tissue than we do in blood, even in paired systems where we have tissue. We see cells that would express ACE2 become infected and look like they're infected. We see myeloid immune cells also become infected in there, which are also in a highly dysregulated mucosal immune environment. And with that, I just want to leave one more thank you because I showed up front, but really we want to thank participants for this. These are not easy procedures. We are doing colonoscopies and endoscopies and sigmoidoscopies. We're doing bone marrow biopsies. But I want to thank the participants for their potential discomfort and inconvenience because we are actually learning a huge amount right now from your sample. So thank you very much for contributing, and we really appreciate it. I think it's really going to help.
And wow, it's just incredible to see your data come along like this. There's just a lot to digest there, but the model of long COVID at the end, it's exciting. All right, I'll introduce our next speaker, who's Beverly Pang. She's in the lab of Chris DuPont and other researchers at the J. Craig Venner Institute, and she'll be talking about a tissue-based analysis of endometriosis.
Okay. Hi. So I'm Beverly, and today I'll be switching topics a little into endometriosis, a little break from the long COVID, but still continuing the theme of the tissue-specific analysis. So today I'll be talking about my work in using single nucleus RNA sequencing to study tissue-specific drivers for endometriosis. So the endometrium is the lining of the uterus that sheds and regenerates during each menstrual cycle. And it undergoes drastic changes during each cycle. So it is important to transcriptionally understand how these cell types and cell states change that lead to disease. And one such disease is endometriosis, which is when tissue that is similar to the endometrium grows in other places of the body, such as the fallopian tubes and ovaries, but it can also travel further to like the bladder and the intestines. So endometriosis is a chronic condition with varying pain levels and symptom presence. And because it is highly variable, it is very difficult and invasive to diagnose and also retrieve samples. And RNA sequencing studies of endometrial tissue are very underrepresented in very well-known data hubs. So it's important to continue generating this type of data and continue developing an endometrial cell atlas. And the data today is from a collaboration with Dr. Nicole Donnellan from the University of Pittsburgh. Okay, so this is the general RNA sequencing pipeline that is used, and today my work will be focusing on the bioinformatics analysis, where the main steps are the quality control, normalization, PCA, clustering, all of which are done in the Python ScanPy environment. So this is a little bit of more specific information on the processing of the endometrial tissue. If you would like more information, I can get you in contact with our wet lab team. Okay. So a little bit more about into the specific steps. So after pre-processing the data, we run our first round of Leiden clustering and identify our 5 broad Leiden clusters. And these data is represented on a UMAP, which is a 2-dimensional way of visualizing nuclei profiles and cluster results. So each dot on this UMAP represents a nuclei, and our data set is comprised of 94,000 nuclei, each with measurements of 36,000 genes. And then we can also color the UMAP according to certain covariates to see how they're distributed in each category amongst all of the nuclei and clusters. So this plot on the upper right is colored according to the 28 patients that we have, which are divided into 20 endometriosis and then 8 control patients. So it's a little skewed. So we can see that skew in when we color according to the endometriosis or control diagnosis. And then one of the goals of this project is to quantify differences in cell type proportions between these control and endometriosis patients. And we can already begin to see some regions on this UMAP where it does appear to be endometriosis enriched. Okay. So in order to begin to computationally annotate our clusters, we need a nice reference. So fortunately, this Human Endometrial Cell Atlas reference, HECA, was published a couple years ago that gives us a good basis for what we should be, for what we should expect to see in our data set. So this data set defines 4 broad cell lineages and 36 more granular cell types. So this gives us a good starting point for our annotation. So these are the 2 computational methods that I helped co-develop and that we'll be using for this annotation. The first method being NSForest, which uses a random forest to identify combinations of necessary and sufficient marker genes for cell type classification. So the NSForest output would be 1 to 6 markers per cluster that best defines the cluster, according to F-beta score. And then FRMatch uses minimum spanning trees for cell type matching between annotated RNA sequencing data sets. And then we can see that the bottom left is a visual example for how we would go about connecting these 2 methods and actually annotating our clusters. So the first step would be to run NSForest on the HECA dataset and generate markers. And the example here we see is for the arterial cluster. We see that there are 2 NSForest markers. And then the next step would be to use FRMatch to actually compute how similar each query reference is to each cluster. So then we can see that it is the combination of these 2 markers that would define this subcluster as probably arterial. So then we can continue applying these steps to annotate the broad Leiden clusters. So we can see that the HECA broad markers match pretty well with our UMAP annotations. So I think so far with these broad annotations, we can say that we are highly confident that these are the correct annotations. So then the next step would be to subcluster each broad method and rerun the NSForest and FRMatch pipelines on these more granular clusters. And then these are the results for that. So on the left, we have the FRMatch results, where the yellow boxes represent a statistically significant match between each query and reference cluster. And then the reference clusters are sorted and colored according to their broad lineage. So just generally looking at these results, we can see that there are some query clusters with one-to-one matches. So then these would be the quote high confidence matches. But then we can also see that there are some query clusters, for example, a few epithelial that just generally match, but at least it is generally matching to the epithelial group. And this could indicate that these clusters may not, or may be under partitioned. But then from this, you can also see that not all query clusters have significant results. So then the next step would be to look more closely and run NS-forest on our data set and then generate the NS-forest markers, which we can then do like a lit search to see what cell type they would most likely be. So this is the final annotation. I've only included the high confidence annotations for this presentation, but I think generally, I think we have some fairly confident results. We have a good representation of cell clusters across all of the broad annotations. And one example we can see is even though we may have multiple potential annotations, we can see that it's a general trend that they are very similar. So generally, we could rank this entire cluster as like the granular, luminal, or pregranular group. Okay. And then taking these final annotations, we can also look at the differences in cell type proportion between the endometriosis and control samples. So kind of bringing back that we are more enriched in the endometriosis overall because we have 20 endometriosis versus 8 control. We do have to normalize these proportions before. So this is a subset of the data, but theoretically each column sum should be 1. To make sure we're not biased one way or the other. So then we calculated the fold change between the proportion of endometriosis and control nuclei that makes up each cluster. And then we can rank these according to these fold changes where values of less than 1 mean control enriched and then greater than 1 is endometriosis enriched. So then the next step would be to begin understanding why certain broad or granular annotations are enriched in one way or the other. And one example of this would be it makes sense for a immune cell type, so that's colored in red, to be endometriosis enriched because it's enriched in a disease. But then it is less obvious why venous or arterial, so the artery or vein clusters would be control enriched. And because this is a pilot study, ideally we want to sequence more samples and also publish a paper diving a little deeper into how these why we think these specific clusters would be enriched one way or the other. And I'd like to thank PolyBio for funding and my team at JCBI for helping me with this analysis. Thank you.
Great, thanks, Beverly. Definitely cool methods on the tissue. All right, I'll introduce our next speaker, who's Nadia Roan. She's a senior investigator at the Gladstone Institutes and a professor of urology at UCSF, and she'll be talking about cytolytic virus-specific CD8 T cell responses in long COVID.
So thanks for having me again. So today I am going to share a brief update on some of our work looking at immune dysregulation in the context of long COVID, specifically looking at cytolytic viral-specific CD8 T cell responses. And so our studies in long COVID very much focus on women with long COVID. And the reason we are focusing on this population of individuals is because even though it's the case that men are more susceptible to severe acute COVID-19, it is women that are much more susceptible to developing long COVID, and specifically premenopausal women. And so our study works with the LINK cohort, which, as everyone here knows, is headed by Michael Peluso together with Tim Henrich and Steve Deeks. Was started back in early 2020 and today has recruited over 1,700 participants. And so our PolyBio funded study focuses on what we sometimes refer to as tolerogenic tissues in women with and without long COVID. And so these tissues are multiple biopsy sites from the gut, as well as from the female reproductive tract, including both the upper tract, endometrium and endocervix, as well as lower tract ectocervix. And we have multiple questions that we are asking with these specimens. One is, do we detect the presence of a SARS-CoV-2 reservoir, and if so, in what cells? Also, what is the nature of immune dysregulation in these tissue sites as compared to peripherally, and what are the features of SARS-CoV-2 specific immune responses? And some of you may recall that Marta Calvet-Mirabend, a talented postdoc in my lab, last time at the PolyBio meeting, presented some early data using single-cell sequencing, where we could actually detect SARS-CoV-2 in cells in all 6 of these tolerogenic compartments. And those are early data, and we're actually still in the process of recruiting right now. To date, we've fully recruited for 1 long COVID participant and 3 controls, and we're actively moving forward with that. But I'm not going to say more about the tissue-based study because we want to wait until we've collected all of the specimens so we can do an integrated analysis. But what I will talk about today instead is blood-based analyses that we have been performing on just blood specimens, so no tissues for today. Okay, so specifically, we are using blood specimens to look at immune dysregulation and focusing specifically on CD8 T cells. So why these cells? Well, these are key antiviral effector immune cells that have been reported by us and other groups to be perturbed in the context of long COVID. And so back in 2024, we reported that SARS-CoV-2 specific CD8 T cells have numerous phenotypic differences in those isolated from people with long COVID as compared to people without. But those CD8 T cells that we had characterized were identified by a technology using that's known as intracellular cytokine staining. And one limitation with this technology is that we are characterizing the features of these cells not in their original baseline state, but following ex vivo stimulation to identify the cells. And in fact, to date, there have been rather limited numbers of studies examining the original phenotypes of viral specific T cells in the context of long COVID. And so we decided to look at this a little bit more closely, looking not only at SARS-CoV-2 specific CD8 T cells, but also CD8 T cells specific for EBV and CMV. And so why these chronic herpes viruses? Well, as I think many here know, both have a relationship with long COVID. And so in the case of EBV DNA levels have been reported to be elevated in those with acute COVID-19, in those that are more likely to subsequently develop long COVID. In addition, EBV antibody titers have been reported to be elevated in those with long COVID. Similarly, CMV antibody titers have also been reported in some studies to be elevated in those with long COVID, but paradoxically, other studies have also reported that CMV antibody titers have been inversely associated with specific forms of long COVID, specifically neurological long COVID. Okay, so in order to identify and characterize these viral-specific CD8 T cells, we used a tetramer technology. It was introduced earlier by Mark Painter, but essentially it's 4 peptide MHC complexes bound together. And the reason we have 4 of them is to increase the avidity effects, but it allows us to identify antigen-specific CD8 T cells, including those specific for including those derived from SARS-CoV-2, EBV, and CMV. And so we were using this technology in the context of what's called combinatorial tetramer technology. It's an approach that was originally developed by Evan Newell when he was in Mark Davis's lab. But essentially, we are using CyTOF as a readout. CyTOF is a high-parameter immunophenotyping method. And essentially what we do is we dedicate 8 of the CyTOF channels for barcoding. And that allows us to, with these 8 dedicated channels, look at up to 56 specificities of antigen-specific CD8 T cells. In addition to doing this, we can also characterize all of these antigen-specific CD8 T cells with a lot of phenotyping markers. So we designed a panel that has 29 phenotyping markers looking at 56 specificities of viral-specific CD8 T cells. And the viral-specific CD8 T cells covered SARS-CoV-2, EBV, CMV, and also influenza. Okay, so we applied these specimens on individuals in link with versus without long COVID. The recovered individuals are shown as R. The individuals with long COVID are shown as LC. And one of the first things we did was to look at just the distribution of classic immune subsets amongst both total CD8 T cells as well as CD8 T cells corresponding to all of these different viral antigens. And these classic subsets include central memory cells, effector memory cells, etc. But overall, the conclusion was that we actually did not see huge differences in the distribution of these subsets amongst all of these CD8 T cells. But to really leverage the high dimensional nature of these data sets, we then implemented unbiased clustering. And you can see we can cluster both total CD8 T cells as well as all of these viral specific CD8 T cells into all these different clusters. There are 24 of them when we use unbiased clustering mechanisms. And this here on the right-hand side just shows the 24 clusters as well as the various markers that define each of these clusters. And so then we asked, well, are there certain clusters that are preferentially enriched in individuals that had recovered as compared to those with long COVID? And in fact, we did find 2 clusters that were enriched in the fully recovered individuals as compared to the long COVID individuals. This was cluster 9 as well as cluster 11. Now, interestingly, when we looked at the phenotypic features of these 2 clusters, they actually shared quite a bit of features, including that they all had low levels of the exhaustion markers in our panel, PD1, TIGIT, and CD39. And so that's consistent with the notion that recovered individuals have a lower frequencies of these cells that are essentially they have higher frequencies of these cells that have lower exhaustion markers. So consistent with the notion that you have a more exhaustive profile in individuals with long COVID. Now, we also asked the reverse question, are there clusters that are preferentially enriched in those with long COVID as compared to the recovered individuals? And we did find clusters that were enriched in the long COVID individuals. And in fact, we found multiple clusters, and they differed depending on the types of specificities we were looking at. So, for example, the ones enriched amongst the SARS-CoV-2 specific CD8 T cells were clusters 2, 13, and 19. Those are CMV were cluster 5, and those are EBV were clusters 19 and 20. Even though these are all distinct clusters, one interesting thing is that they all share features and that all of these clusters preferentially express the cytolytic markers granzyme B, as well as CD29, which has been used as a marker of cytolytic CD8 T cells. And so this prompted us to next ask the question of what if we just gate specifically for these cytolytic markers using manual gating approaches? Do we see that it is elevated in those with long COVID? And so we gated on CD8 T cells expressing just CD29 or just granzyme B or the combination of the 2. And you can see overall, we do see that the individuals with long COVID have higher frequencies of these particular CD8 T cells. But the even more interesting thing was when we stratified these data and separated into participants that were either female or male, you can see this phenomenon is much more prevalent in the female participants, where you really see this elevation of these cytolytic CD8 positive T cells, whereas in the male participants, we didn't really see that. So this notion of preferential cytolytic CD8 T cells in the context of long COVID may be preferentially in women with long COVID. Now, cytolytic CD8 T cells are known to develop during acute viremia. They serve the purpose of helping to eliminate acutely infected cells. And so we postulated that perhaps this elevation of these cytolytic CD8 T cells in the context of long COVID might reflect an inability to shut down this activity after acute infection. And so to begin to look at that, we looked at some longitudinal specimens from an individual that had fully recovered versus an individual that had long COVID, from the acute phase all the way out to hundreds of days post-infection. And you can just focus your attention on these, the orange line, which is the SARS-CoV-2 specific CD8 T cells identified by the combinatorial tetramer. And looking here specifically at the levels of granzyme B, one of those cytolytic markers. And you can see in the fully recovered individual, it starts off high, the cytolytic SARS-CoV-2 specific CD8 T cells, and then it goes down over time, as you would expect if the virus were effectively cleared. But in the individual with long COVID, you can see that it starts off at a certain level, and there's no evidence of it kind of going down over time. And in fact, it tends to increase over time. And so what we also decided to do was to look in an entire cohort of acute individuals, people with active COVID-19, and compared the frequencies of these cytolytic CD8 T cells in those individuals as compared to recovered and the individuals with long COVID. And what we found was that the individuals that had active COVID-19, that they had high levels of these cytolytic CD8 T cells amongst both the total CD8 compartment as well as the SARS-CoV-2 specific CD8 compartment, but that it went down as you would expect in the fully recovered individuals, but we didn't really see it going down in the individuals with long COVID. And so to summarize what I talked about today, so combinatorial tetramers allow for simultaneous phenotyping of many specificities of viral specific CD8 T cells. What we found was that overall, the CD8 T cell classic subset distribution did not differ between individuals with versus without long COVID. We found that CD8 T cells with specificities for SARS-CoV-2, CMV, and EBV all preferentially express cytolytic markers in those with long COVID. And we think this may reflect an inability to shut down cytolytic CD8 T cell responses that are elicited during acute COVID-19. And with that, I want to thank the folks that did the work. So Tongsui Ma was the one that did this work in my lab, but we worked closely with Evan Newell's lab, specifically Hiju Ryu, who used to be in Evan's lab. Also want to thank the entire LINC team, particularly Michael Peluso, our funding source, PolyBio, and Amy Proal, and I will stop there. Thank you.
Great, Nadia. Really cool data, including the sex differences in some of the results. So very interesting. Okay, I'll introduce our next speaker, who's Chiara Gianarelli. She's an associate professor of medicine and pathology at the NYU Grossman School of Medicine, and she'll be talking about SARS-CoV-2 persistence in vascular tissue and cardiovascular risk.
Thank you, everyone. It's really a pleasure to give an update on our project on SARS-CoV-2 persistence in vascular tissue and consequences in terms of cardiovascular risk. We are very interested in cardiovascular complication because there is a broad spectrum of cardiovascular manifestations that have been associated with COVID-19, and these include stroke and myocardial infarction, and we're very interested in tackle this problem. And we given the importance of cardiovascular complication, we answered the question. We wanted to answer the question related to the underlying mechanism that sustain this increased risk for patients, not just during the acute phases, but also in the long term, up to 2 years after acute infection. And what we found is that SARS-CoV-2 can have a very specific and direct effect in modulating inflammatory responses within human coronary vessels. And we discovered this by using different approaches, but substantially we were able to document the presence of viral RNA in human coronary atherosclerotic and non-atherosclerotic coronaries by quantifying viral RNA, and we found that this was unrelated to the degree of disease, presence also in normal vasculature, and not so affecting other structure, for example, perivascular tissue. And then we decided, with the support of PolyBio, to move on in addressing the question of whether these manifestations with increased inflammatory potential at the lesion sites, at the human vasculature sites, was persisting over time. And to address this question, we leveraged our patients enrolled into studies that are called ATHEROIN and ATHEROCORE. For this analysis, we used over 140 subjects, and this comprised patients where we collected coronary plaque samples or carotid plaque samples. And based on their history, we could classify them on whether presence of infection, prior infection, COVID-19, others because they were enrolled before COVID-19 first cases were reported, were classified as COVID-19 negative, and then we also have some that have an unknown COVID-19 status. And this is another representation that also outline how we can integrate clinical data like their age, sex, BMI, but also other cardiovascular complications into categorize these patients in these three groups of COVID based on COVID-19 status with at a higher resolution. And we approach the problem of trying to understand how COVID-19 can increase cardiovascular risk over time by using a multi-omic approach where we analyze carotid, coronaries, but also the blood of these subjects. Using Luminex XA, bulk RNA sequencing, and single-cell RNA sequencing. For today, I would like to tell you how we substantially wanted to address the problem of having a robust analysis to help us identify the status of these patients based on a very specific readout. And we use the Luminex serology that is able to really distinguish patients based on prior infection with a validation that goes up to 2 years in terms of specificity and sensitivity. And this is based on the assessment of different strains of SARS-CoV-2, but also other viruses that are listed here. And these are the results that we had. And this heat map really, you can appreciate, really helped us having a very robust classification in terms of whether the patients had a history of COVID-19 or not in the pre-COVID pandemic and COVID pandemic subjects based on the detection of the S and N antibodies against S and N. And down below you have different others, viruses, serology that is included in the assay. And of course, you can appreciate that we achieved better resolution, allowing us to move on with different additional studies. Again, this is another quantification that really shows that you have an persistence of different antibodies against different type of serotype of SARS-CoV-2 infection. Okay, so based on this analysis, we looked at our transcriptional analysis that includes blood, tissue, so carotid and coronaries, and we wanted to look at what were the major contributors to the variants of that we found in this context. And of course, there is an intrinsic difference in carotid and coronary tissues that is the main contributor to variants, but COVID history is the next one. And it's even explaining even more than smoking status and sex or hypertension or hyperdyslipidemia, the differences that we see in our transcriptional data. And again, this can be appreciated even more by looking at the different principal components analysis and how they contribute to the differences across covariates. And COVID history, again, in this portion of the panel to the left, you can see that is the second contributor to differential expression analysis across covariates. So the next question is, what are the specific molecular signatures that emerge from these plaques? And we looked at different programs that are consistent with chronic viral antigen exposures and sustained inflammation in human coronary plaques. And we found a very strong signature related to IL-6, IL-1 beta, cytokine signaling, but also responses to viruses. And these are upregulated in atherosclerotic plaques of COVID-19 patients, positive patients, based on the serology assay that I explained. And you can see that the top are cytokine activity, sensory perception of smell, defense responses to virus, IL-1 beta, IL-1 6 that are important in cardiovascular disease because they are validated to have very strong causality with cardiovascular events and disease in patients, whereas other signature that have to do more with homeostasis of cholesterol and lipid metabolism were downregulated in these patients and upregulated, of course, in COVID-19 negative patients. We were also able to detect viral RNA using this deep sequencing in some of the human atherosclerotic plaques and paired blood, and you can see that this detection is displayed here in coronary plaques, where you see that in this set of patients, there was a background level of detection, but in some of them, the detection was higher. And this was also in post-pandemic patients. And we detected this in some of the subjects that were classified as COVID-19 negative, perhaps because they had a history longer than 2 years of infection. But we're still looking into that. And here you can see the vaccination status and the COVID infection status as well. What's interesting is that when we looked at whether the this signature, this viral RNA was detectable in the blood or in the plaque, you can see that there was a mismatch for most of them, but in some cases, there was a detection in both conditions in relation to patients with coronary samples. For the carotid, this is even higher in this context. And we were then interested in understanding what are the underlying mechanisms potentially influencing this highly inflammatory, persistent inflammatory response. So we looked into the possibility that transposable elements might be the underlying this mechanism by which this inflammatory response is dysregulated. And we found that indeed we were able to map transposable elements in human plaques from patients with a history of SARS-CoV-2 infections. And you can see here the different classes and families of transposable elements that we mapped. But in essence, we were able to see a very clear, distinct signature of transposable elements that were upregulated in COVID positive plaques from patients who had a history, and they all belong to the LTR family. And then we have a few that are downregulated in this context. Particularly, it was interesting to see that when we infect using this ex vivo human vascular explant model, human tissue, we can see over time really clear activation of transposable element in these tissues that peaks at 72 hours. And when we looked at whether there was an overlap between the acute infection and those that were upregulated in long after the acute infection, there was a partial overlap. But also very distinctive signature, speaking for perhaps some initial activation of transposable elements that's appropriate for the acute infection, but then the activation of some additional programs that are more specific for COVID-19, long COVID. And specifically, we mapped the transposable elements that are activated in carotid plaques of these subjects, and we were able to see that they are particularly enriched in myeloid cells, that are the cells that in previous studies we found are specifically targeted by the virus, and they are infected in human atherosclerotic lesions, as well as we proved that in vitro. indicating that there is a real interest in or influence of these transposable elements in particular cell types that are the ones that are infected by the virus. There is also a functional aspect to whether the infection can, in fact, alter some function of macrophages in this context. And in these initial studies, we show that there is an impaired efferocytic capacity of primary human macrophages when they are infected. And this is a very important and fundamental function of macrophages in maintaining tissue homeostasis, and in particular in atherosclerosis, being able to clear up lipid accumulation in these lesions. And when this function is impaired, this lesion grows and there is increased risk for cardiovascular disease events such as heart attack or stroke. So in essence, we can conclude that there is a lasting mark of SARS-CoV-2 infection in atherosclerotic lesions, and there is an inflammatory, vascular inflammatory reprogramming, and also the persistence of detectable viral RNA in some of these subjects. And we are now interested in pursuing more mechanistically and larger scale our analysis on how transposable elements are selectively upregulated in these plaques and preferentially activated in myeloid cells, indicating that there might be some long-lasting innate immune senses mechanism that are related to viral persistence that may have implications for other long COVID manifestations as well. And with this, I would like to thank PolyBio for supporting our research and giving us the opportunity to perform these studies, and of course my lab for performing the studies, and in particularly Natalia Heberhardt in my lab, who has performed most of this work. And thank you so much for listening.
Great. Thank you very much, Chiara. With the topic of cardiovascular consequences of COVID in mind, our next speaker is Xian Zhang. He's a professor of medicine at UCSF, and he'll be talking about post-mortem mapping of SARS-CoV-2 persistence and sudden cardiac death.
Hi, everyone. I think I'm the only clinical cardiologist here on the panel. I'm not an immunologist or infectious disease expert, but via our study, I think we can provide some unique insights into what we don't capture in the hospital. So, as we all know, heart disease is the #1 killer, and sudden cardiac death is a substantial component of the that #1 killer. However, what we understand from sudden cardiac death derives from the small percentage that get into the hospital. And what we've been doing is examining deeply all of the sudden cardiac death cases that never come to clinical attention because they go to the medical examiner or the coroner's office. And so in San Francisco, we've been running this study for the last 15 years. Every single case gets reported to the medical examiner, so there's no bias. Death or taxes, we capture every single case in the county. They all go to the medical examiner, my collaborator, Dr. Moffat, who's been an amazing partner in our study. And by virtue of that, we can capture every single case and do a comprehensive autopsy to understand the underlying causes of all these cases. And San Francisco being a very international city, we capture national and worldwide. So it's really a snapshot of what's going on throughout the world, not just in our city. And so a few years ago, we published the first results of our study. Again, here I'm highlighting Dr. Moffat, the medical examiner in San Francisco. And not to take you through every single one of our methods here, but we track every single death, 20,000 deaths over a 3-year period. And she achieved an incredible 97% autopsy rate of all of these out-of-hospital cases. So truly no bias as opposed to other autopsy studies of selected cases occurring out of the hospital. And so here's the punchline of the first report we had, which is that nearly half of what's been called sudden cardiac deaths are not even cardiac. You can see here, acute heart attacks only accounted for 10% of the burden of sudden cardiac deaths, and we've uncovered the myriad causes of sudden cardiac death or presumed sudden cardiac death throughout our study. And this has led to a number of discoveries. I won't take you through all of them here, but HIV, device problems hidden among sudden cardiac deaths, sudden neurologic deaths, and overdoses, for example. So how can we take this study to tease apart exactly what's happening in long COVID? Well, my incredible team reaches out to the family members of these victims of sudden cardiac death and asks for consent to transfer specimens that were collected at autopsy to our lab. And now we have over nearly 450 cases with autopsy specimens spanning all the underlying causes of sudden cardiac death. And importantly, we are able to sample at autopsy the deep tissues beyond what's accessible in the hospital, for example, beyond the coronary plaques, deep into the tissues, in the brain, in all the deep organs. And so the theme of our study is find the reservoir. And the nice thing about our study is that we have nearly 10 years of pre-2020 epidemiology and tissues to study. And so by virtue of our study, we can hope to find the causes of long COVID. We examine these deep tissues, look at viral persistence, immune dysregulation, interactions with chronic diseases, and then again, we can look at endothelial dysfunction, but also go beyond that and look into the tissues. So here's a adaptation of what I showed you earlier. You know, a lot of our autopsy specimens so far in the world of COVID research comes from hospitalized cases, whereas what's not captured out in the community has been missing, and this is where post-SCD is really poised to capture these results. Now, here's an example of why this is an important distinction. In our study, we captured trauma deaths as controls, and we have about half of our trauma deaths died instantly at the scene of the trauma, and then about half survive to get hospitalized but then die in the hospital. And here what I'm showing you is the transcriptome of deep tissues in instant trauma victims versus hospitalized trauma victims, and there's a massive immune response just from the life-threatening trauma or the critical illness. So that can really confound results if you only look at hospitalized autopsy specimens. And so here in our study, we've now processed 70 total cases post-2020. 65 are undergoing some phase of our deep sequencing. And then again, we have nearly 400 matched specimens pre-2020 to examine whether there's any confounding in what's occurring in the natural environment. And so here's just our preparation sequence. We start with our tissue sample, we do extraction, automated library preparation, we do targeted probe and enrichment and biosequencing, and then we have a process of deep analysis of both non-human and human reads so that we can assess the host response element of this. So, so far, what our study design is that we can look across tissues and across people who have been captured with our study. And what I want to show you first is that we started with myocardium. And because of the depth of our sequencing, we can go and see if any of these cases, for example, are false positive or false negatives. So far, I'd like to highlight for you the first approximately 60 cases we've done in the post-2020 period. And we found 4 out of the 53 cases, so 56 cases so far, had acute COVID in the heart. So this first case is a 24-year-old male whose cause of sudden death was a occult overdose. He was COVID positive at autopsy, but we found COVID in the brain and in the heart, pending enrichment in that particular case. Second case was a 70-year-old man who died of also an occult overdose. He was also COVID positive, and we found COVID in the heart and the brain in that victim. And here was the one later case in 2024. This gentleman died of a cardiac hypertrophy cause of sudden cardiac death. He had no evidence of COVID, but there was COVID in the heart. So in this particular case, truly maybe occult long COVID reservoir in the heart. And then the one surprise was approximately a year into the pandemic, we had a 32-year-old male who at autopsy we adjudicated his cause of death as myocarditis without yet knowing our sequencing results. And here you can see that he had COVID that was tested positive. We found in the deep tissues of the heart that caused myocarditis as his cause of sudden death. He was COVID negative at autopsy, so this was not in the setting of an acute infection, at least not in the first 2 weeks. So this is a truly out of hospital surprise cause of sudden death. So far, as I mentioned, we've had 56 cases we've tested, and if you extrapolate, this is approximately 5% total positive myocardial testing positivity. And then if you look and you subtract the acute COVID cases, this is approximately 4%, if you will, extrapolation of COVID reservoir in the heart so far. So what are we doing with these deep tissues? What we have also saved is FFPE blocks, and we can go deep into the tissues and look at spatial profiling of where the virus might be. For example, I'm going to be showing you here the example of the one case with who was positive in the brain, and we can see that we were able to map SARS-CoV spike RNA within the neural tissue in this particular case, and these kinds of deep tissue experiments are pending in the heart and other tissues. And so far, we're performing NGNS in all tissues from all time points. As I showed, we can detect RNA transcripts and DNA from not just SARS, but other eukaryotic viruses, so we can evaluate interactions between other viruses and SARS-CoV-2, and this is a much higher sensitivity than other methods such as nCounter, for example. And so with that, I'd like to thank particularly all victims of sudden cardiac death and their families in San Francisco County, Dr. Moffitt and my co-PI, Dr. Henrik, and also, of course, our funding support from PolyBio. Thanks for your attention.
Thanks, Ian. Those were some interesting cases. Okay. Our next speaker I'll introduce, it's Henry Van Brockland. He's a professor in residence in the Department of Radiology and Biomedical Imaging at UCSF, and he'll be talking about whole body imaging of T-cell activation in long COVID.
Thank you. Yeah, and thank you, Amy and the PolyBio Foundation for the opportunity to speak here today at this symposium. And what I'm going to talk about today is some of our work in the looking at immune activation by positron emission tomography imaging in our long COVID interventional trials. But just want to start off by sort of reiterating some of the themes that we've already heard today about SARS-CoV-2 and its persistence in tissues for extended periods. And as you can see, it's very challenging to get access many tissues that may be related to long COVID. And so we believe that non-invasive imaging plays in a really important role in characterizing, identifying, and categorizing the various aspects of long COVID. And you've already seen some of this today with Mike VanElzakker's talk using non-invasive imaging approaches. And this is something that we're putting forward in our work here. We have many different ways, and specifically going to focus in on the immune system. And we have many different ways of interrogating the immune response, and also looking at the sort of physical as well as functional aspects of this immune response. And today I'm going to focus in on looking at the sort of the functional response that we see in T cells and specifically activated T cells, and hone in on FRG as a mechanism for and looking at T cell activation. The way that FRG works is basically a molecule here. It's an analog actually of nalarabine, which is a chemotherapeutic. They found that colleagues at Stanford found that it actually gets across into T cells, gets taken up and is either phosphorylated in the cytoplasm or in the mitochondria. And in cases where the T cell is not activated, this phosphorylation can be reversed by SAM-HD1, and then the free or unphosphorylated FRG can get back out of the cells. But in the case of T cell activation, where SAM-HD1 is downregulated, the phosphorylated FRG actually, the phosphorylated FARG actually stays trapped inside of the T cells. And so where you see this accumulation is where you have your activated T cell activity. And they did a lot of work to validate this. Dr. Gambier, who developed this and has unfortunately subsequently passed, as well as his wife, started a company called Cell Site Technologies. And since he was at Stanford, he was unable to actually do some of the first in-human studies just because of conflict of interest. So he approached us here at UCSF and asked if we would be able to assist in first looking at uptake in healthy volunteers. And then some of the first patient studies that they were interested in doing was following chemoimmunotherapy in cancer patients and looking to see the immune response to that treatment. So I show here an example of female and male subjects. These are healthy controls that received the FARG and were scanned over a period of 3 hours. You can see here over on the right some of the normal tissue uptake. So we have pituitary, heart, liver, kidneys, of course, clearance organs, parotid, submandibular. So this just gives you sort of an overview of what you see in the healthy volunteers. In the setting of long COVID, we got into this because we had actually noticed and done a study looking at the HIV reservoir. We had developed an antibody molecule based molecule that was actually looking at the HIV cell coat protein, GP120. But at the same time, we also started to investigate whether the FARG, which we had in our hand and already had an IND for imaging in humans, we started looking at that as well in our HIV subject and noticed that we were getting a lot of overlap in the HIV reservoir that we were seeing with our VRC01 molecule, our antibody, with the signal that we were seeing from the FARG. And so this, we did that study before long COVID, before COVID set in. And then as soon as COVID hit, one of the ideas that we had was, well, if we can see the reservoir with the and see reservoir or T cell activation in HIV, can we also see it in long COVID? And so that's what got us started in this particular area. To date, we've looked at 79 participants. We've reported on the first 24, and I have some of that data I'm going to show here today. You can see over quite a range of time from infection, up to 15 long COVID symptoms reported in some of the patients, a median of 6. And we had our 6 pre-COVID healthy controls, and we had 6 with reported no long COVID symptoms reported. We did a number of assessments. I'm not going to go through all these but we asked a number of different questions and looked at the data in a number of different ways so that we could try to understand what some of the characteristics were of the long COVID and the uptake of FARG. The demographics of the first 24 subjects, we had 15 pre-Omicron, 9 post-Omicron, the mix was about 50-50 males and females. And I don't know what we're up to 79 subjects now, so I don't know what the demographics are for the full cohort. But we have also the days post-infection, and we also included patients that were vaccinated and understanding what the vaccine may have done in terms of changing the uptake that we saw with the FARG. Here's just 3 patients. One pre-COVID control on the left, and in the middle and the right, long COVID from a couple of months out to a couple of years. It's at this level, it's very difficult to see the differences. You really have to do a little bit deeper dive and take axial slices and look. But we do see some gut uptake here, at least in this patient here at 63 days after infection. And it's something that we see in several of the patients. But when we do take and go tissue by tissue, and here are some images, we saw uptake in the nasal turbinates, parotid glands, which are even elevated over what we see in the pre-COVID controls, uptake in lumbar marrow, hilar lymph nodes, lung parenchyma, again, in areas that we do not see uptake in our control subjects. And you can see in many of the tissues, red is post-COVID, The gray is the 6 pre-COVID controls that in many of the different tissues that we looked, in the brain, in the lungs, in lymph nodes, marrow, liver, spleen, parotid, and glands. You can see the uptake in the COVID patients was significantly higher in many of these cases to the what we had for the healthy controls. And if we broke this down in symptoms, I'm not going to have time to go through this in great detail, but you saw increased uptake in long COVID symptom phenotype positive patients versus the controls, and in many cases, even in cases where the long COVID symptoms weren't associated with the phenotype, we actually saw elevation there as well. And so, can we tease out some of the various components here? So fast forward now to some work that's been done more recently, where we've looked at different uptake in different long COVID in patients with different long COVID symptoms. On the left, cardiopulmonary, and you can see again, this is now an increase in the FRG uptake here in the rectosigmoid wall and the colon, so in the gut, and these COVID patients that we're comparing it to now are not our 6 pre-COVID controls. These are now patients that have recovered from COVID and are experiencing no long COVID symptoms. Okay, and so now we have a more recent set of controls that we're comparing these to, and you can see that the increases here are still significant. If I go over to the right, this one is actually interesting. In 7 patients that had olfactory long COVID, we saw this increase here in the posterior nasal pharynx, and you can see it's quite different from the 59 patients here that had no long COVID symptoms. And I put this reference down here at the bottom, and it's actually a reference to a study that was done a couple of years ago, where they actually showed that there was this sort of migration of long COVID or COVID, the virus, viral components from the skeletal marrow into the brain. And so is it plausible that perhaps what we're seeing here could be the reservoir that might offer entry into the brain in this region here in the nasal pharynx? It might be possible that that's where some of this is making that migratory walk. So now to the topic that of the subject of the talk, and that is a few slides now on our trials that are ongoing here with the link cohort, the Outsmart trial, which is sort of pre and post imaging of patients that received Arium-002. It's a monoclonal antibody. So we had 36 individuals with long COVID that were enrolled, and 24 received the Arium, 12 received the placebo, and a sub-study of these 10 individuals, 6 that received Arium, 4 placebo, that were injected with the FRG. We saw no differences between the monoclonal antibody and the, sorry about that. We saw no difference between the monoclonal antibody and the placebo prior to treatment, with the exception of the cauda equina. So that's shown up here. So these are the baseline. There's no significant difference between between the Arium and the placebo images. But here you can see the cauda equina. This was significant, but it was even more significant post-treatment. So we saw a significant decrease in the Arium patients here in the temporal lobe, the thoracic spinal cord, and the submandibular gland, in addition to the cauda equina. And just to take a little bit deeper dive on the submandibular gland, you can see here that 5 out of the 6 patients actually, I guess 4 out of 6 actually saw a decrease in uptake of the post-treatment with Arium, and in our actually placebo controls, 3 out of the 4 actually saw what looks to be a little bit of an increase in uptake, and that you can see also made out over here in the images. So here's placebo before and after, and here's baseline in Arium before and after. You can see here the decrease and here the increase in these particular patients. In another trial that's ongoing right now, 40 long COVID patients were given either encitravir or saline infusion, and 24 of these patients have been imaged, and both pre and post administration. The data is being analyzed, and I'm sure if given the chance in the fall to report out, that you'll hear more about the results of this trial here. So just brief summary. Inflammation and dysregulated immunity persists for years following SARS-CoV-2, as we've seen today. We have a couple of trials that are ongoing now where we're incorporating this FRG imaging to see what tissues are affected, what the effect is based on the therapeutic that's being given. So with the Outsmart trial, we've observed CNS and glandular tissues post-treatment seem to have some decrease. The Prevail trial is ongoing, but I think one of the things that we're seeing here is that the imaging may be an important tool to measure the tissue-based whole body immune activation and inflammation in long COVID treatment studies. With that, I'd like to again acknowledge this slide, some you've seen before. Obviously, acknowledge PolyBio for their support of the long COVID work. Without PolyBio, none of this would have been possible. And of course, you've seen this in a few slides now, the link cohort and that wonderful curated cohort of patients that are now available for applying to these studies. With that, I thank you for the opportunity to speak.
Amazing, Henry. Thanks. Very interesting. All right, I'll introduce our next speaker, is Isin Safik. She's an assistant professor in the Department of Immunobiology at Yale University School of Medicine, and she'll be talking about defining SARS-CoV-2 RNA reservoirs and neutrophil-driven inflammation in long COVID.
Thanks for the opportunity. I'm going to echo a lot of the themes that we've heard today: myeloid cells, macrophages, neutrophils, and then the tissue biology. But we take a pretty unique approach to study these, and we utilize something called a humanized mouse model. So these mice are genetically re-engineered mice that allow human immune system to develop in them. So we put in a lot of different human factors into these mice, and these human factors allow human immune system to develop. In addition to these human factors, actually, we can transplant these mice with human tissues. We've done it for lung and liver, and we're optimizing ways to study the intestine. And we can edit the immune system in these mice, approach, take over multi-omic approaches. And eventually the idea, the hope is to find a model disease that are relevant to human and study disease mechanisms, but also use these mice to as a platform to test therapeutics. And we've used these mice in the context of infectious disease, fibrotic disease, and autoimmune and inflammatory disease, and cancer, and we call them a Mr. G6 family of mice for the genes that we have modified in these mice. But just to summarize very briefly, in humans we have an abundance of myeloid cells in circulation as well as in tissues, and most of the existing humanized mouse models fell short here. So that's why we developed a mouse model called Mr. G6, as you can see, that mirrors the human immune response. And just to go deeper, it's not only the proportion of these cells, but also the phenotype of these cells are very similar between these humanized mice and their human counterparts. We've looked at lung myeloid cells and compared how they resemble to their human counterparts. We've done it for liver macrophages. We've done it for tumor-infiltrating immune cells. More recently, we've done it in the context of human neutrophils in a mouse model that we've just developed that enables human neutrophil development. And just as a proof of concept, I'll go back to severe COVID-19, and we've studied severe COVID-19 and the macrophage response in these mice. These mice show the same disease steps or histopathological steps that we see in patients. They start with infiltration, but they end up with fibrosis. We can study the human immune response in these mice, particularly focusing on the macrophages, and we can test the causal role. So in the context of macrophages, we've actually shown that human macrophages can be infected with SARS-CoV-2. There are 3 different mechanisms. 2 of them we've described, either to ACE2-mediated or antibody-mediated infection, and another group at ABU have described a CD163-mediated one. What happens is that SARS-CoV-2 replicates in these macrophages, and it activates an LRP3 inflammasome, which is a very inflammatory form of cell activation, which leads to pyroptosis. In the process, there is also maturation and secretion of really inflammatory cytokines. This is what we uncovered in mouse, but we went and validated this in human autopsy samples from Yale New Haven Hospital, and we can see that a very significant portion of the macrophages at the time of that are actually infected, and we quantified this by looking at RNA-dependent RNA polymerase as well as other viral antigens. So recently, we've been focusing on human neutrophils, and we're trying to understand how they contribute to viral persistence. Similar to macrophages, neutrophils do express the receptor that is required, ACE2, and to test whether they can be indeed infected, we went back and used a reporter strain of virus. And as Layal has done, we've taken healthy neutrophils and kind of instead of doing immune complexes, in this case, we have infected these macrophages with SARS-CoV-2 and used the reporter virus. So MNG, which is neon green, is only present in cells that are infected, and you can see a very clear persistence, about 40% of neutrophils having the signal. We can reduce the signal if you use antivirals like Paxlovid, but we need to really use high doses of Paxlovid to be able to do that. A similar observation was there for remdesivir, which makes us think maybe if we want to really clear these myeloid reservoirs, we need to come up with unique antiviral strategies. This was done by Sophia Peng in my lab, but we wanted to make sure that there's actually viral replication happening in these cells, and we've shown this in macrophages, and we wanted to see neutrophils as the second cell type, a myeloid cell type. And we collaborated with Michelle Lua from Anna Pile's lab here. She developed primer sets so that we can look at subgenomic RNA and also characterize the subgenomic products from these neutrophil infections. And we can see subgenomic products as early as 4 hours post-infection, and they align to the SARS-CoV-2 genome. We don't see every species that we see, for instance, in the virocell, and that's ongoing investigation right now. So, well, I've told you a lot about humanized mice and, our in vivo settings. So what we can do with this mouse model is we can preferentially target monocyte-derived macrophages and have a model that lacks these macrophages, or we can target alveolar macrophages, have a model that lacks these alveolar macrophages in addition to monocytes and monocyte-derived macrophages, or we can have a model that lacks human neutrophils. And what we wanted to see is if we have any of these cells, let's have a mouse model that just has neutrophils or just has alveolar macrophages, the monocyte-derived macrophages, do they contribute to viral persistence measured by a subgenomic RNA? And indeed they do. So if we just have any type of myeloid cell, it is enough to have sustained viral persistence in these mice. But what is very interesting, if you have a mouse that lacks all of these myeloid cells, the virus is eventually cleared, suggesting that myeloid cells themselves are significant reservoirs, or they contribute to this infection either by increasing the susceptibility of the epithelial cells or preventing the clearance of these viruses through inflammatory mediators that we are currently investigating. So what we think right now is that myeloid cells are targets of SARS-CoV-2 infection. In the acute phase of the disease, when we look early, the predominant cell type that is infected is actually indeed epithelium, shown by the red cells here, the red epithelial cells. But when we looked at the chronic phase of the disease, what we've seen is sporadic epithelial infection and very abundant myeloid cell infection, whether it's a macrophage or a neutrophil. And now what happens is when we deplete these cell types, when we get rid of the macrophages and the neutrophils, all of these myeloid reservoirs, indeed the tissues can clear their viral reservoirs. And we reduce even the epithelial infection in these mice. So currently, this is our working model for lung, and we're currently investigating what the mechanisms are, whether the myeloid cells are themselves a reservoir, or do they increase susceptibility of the epithelial cells to infection, and how they contribute to viral persistence. But this was a common theme, and throughout today. We're also interested in the intestines, and we're currently establishing intestinal infection models similar to what we have established in the lung, and testing whether macrophage involvement is sufficient to increase susceptibility of the epithelial cells and what happens when we perturb these states. With this, I'll take everyone. This has been a very collaborative effort. We've been funded by PolyBio as a very collaborative team. Akiko Iwasaki, Anna Pyle, and Craig Wallen have been instrumental in all of the data that I've shown. But because of time, I haven't had a chance to talk a lot about our collaboration with the Link cohort, particularly with Tim Henrik and Michael Pelusa, where we're trying to identify and test the immune complexes in the context of long COVID and how they shape the in vivo phenotypes.
Great, thanks, Ethan. That's cool work you'll do with the Link group for sure. All right, I'll introduce our next speaker. It's Eduardo Retigui. He's an associate professor of chemical and biomolecular engineering at The Ohio State University, and he'll be talking about BARA, integrated nucleic detection of viral antigens and nucleic acids in long COVID.
Thank you again for the invitation and for allowing us to share our work in SARS-CoV-2. First of all, I just want to make a small introduction about our lab. We are a technology-driven lab, basically, in which we are trying to develop is microtechnologies and nanotechnologies for the characterization of circulating biomarkers, and also in terms of characterization of viruses. And actually, that's how we started to work in SARS-CoV-2 during the pandemic. I'm going to focus today in one of the assays that we have developed that is called the BARA assay. And basically, what I would like to start is how we actually designed this assay. So we work largely also in extracellular vesicles, which are actually nanoparticles that cells release in circulation. And we found out that, like many others, that there's a lot of analogies in terms of these extracellular vesicles and actually viruses. Like, for example, they have properties that are very similar in terms of the size. They have surface markers. They have the cargo, which is the RNA. So I think that was actually the initial step for us when we start to design our assay. So the way that basically it works is we work with individual virus particles. We don't lyse anything in our assay. And it works by immobilizing these virus particles on a surface, right? Which is, you can see it here, this biochip. So we can, for example, put it here, the virus particle. We can immobilize also, as you have mentioned before, the extracellular vesicles that these biofluids may have. And then our detection consists of basically integrating antibody on the surface, and then we have designed probes that go directly inside the virus and then can basically detect the different regions of the messenger RNA. And so basically, we're trying to combine an antigen and a molecular test in one. But actually, we also can do not only in the viral particle by itself, but also in the extracellular vesicles. So the readout that we observe is based on fluorescence. So, for example, we can directly look in terms of the spike protein, which is on the surface of the virus. So these are individual particles. So we can look into the RNA, which is inside the virus, what I was showing you before in the cartoon before. And we are also interested in this co-localization of the signal, especially if we are interested in seeing if both or if the virus actually is intact. And more interestingly, also, we can look on this signal in terms of the RNA that is present in the host EVs of a patient, right? So this is in the EVs. So this integrated workflow is what we call it, like this VARA, which is the biochip for antigen and RNA assay in terms of COVID. So this has been designed with a very requirements of the FDA, since we submitted an emergency use authorization for this assay during the pandemic, and by doing that, I think actually the characterization of the assay has been done with many controls. For example, here for the determination of our limit of detection in terms of like the antigen, also in terms of the RNA, each one has individual signals, right? So then we validated this against the standard assay, which is a PCR, and the ELISA assay, in which we found that actually we have a much higher sensitivity for what we wanted to characterize for the protein and RNA in the virus. Since we have been also talking about samples that can be frozen, not only fresh, we also did a very deeply comparison in terms of like the performance of the assay is actually maintained across these two different variables. We also validated in terms of different mutations that are present in the virus to actually demonstrate that there is not too much variation in terms of performance. We have also tested cross-reactivity, right, in terms of other different viruses, bacteria, fungi, and we have also conducted studies that had to do in terms of interference. Especially this was when samples were basically saliva, right, which could have some effects on performance of the assay. So this is a fully automated assay, as you can. I'm showing you some steps here, and the capabilities in the order of 200 and around like 250 samples that can be performed in the platform. So in terms of clinical results, the first cohort that we ran actually was in the active phase. And as you can see, we divided this in terms of antigen and RNA for these two different types of samples here. And when we combine actually both of them, I think the sensitivity that we got is outstanding. It's almost 100% of performance. Then we also did tests that has to do in terms of like asymptomatic patients, just to see how it will perform the assay, in which we found that actually very good agreement with tests that have been done with PCR. And what's interesting in here, in terms of long monitoring for long COVID, with this assay, we run like a pilot of 7 patients here that were different time points, but this was after the active phase of the virus. This was done in collaboration with Dr. Jason Goldman of the Swedish Health Institute. And what we try to do here basically is if we can actually measure those signals after the active phase. But in this case, we largely focus on the population of which is the extracellular vesicle, right? So we didn't identify actually virus in circulation, at least in these 7 patients that we did it here. But once we have this confirmation that we have the EVs, then we actually can prove them for actually the viral RNA. So here's one example from one of these patients in which we compare with one healthy donor here, and then for 3 time points from one of the long COVID patients. So in which we can see that clearly we can identify this RNA signature. So, and then we can also do something that is very unique to this assay, which is the colocalization of signals. For example, we can colocalize a surface protein versus this viral RNA. And this actually is very important because one of the aims that we are actually working with PolyBio is trying to see if tissue of origin is actually one big aim that we are trying to perform. And here is it's also this same 7 cohort of patients here, but more like for each one of the different time points. So it's very clear that over this periods of time we can actually measure this viral RNA present in EV. So what we are doing right now is actually we finalized. We're starting to probably run some of the additional samples that we are very grateful for receiving from this organization. Which is going to be in the order of, I think, around 150 plasma samples. Some of them are going to be paired, and our main goal is basically trying to determine viral persistence, the comparative amount of the viral signature in these different biofluids, and replication with the different RNA probes that we have. So I think this is just a short presentation. So I think I just want to summarize that we actually are developing this single EV platforms or EVP platforms that have a very remarkable sensitivity that allowed us to ask questions beyond just the traditional finding the signatures. We are also interested in actually where these signatures are coming from. And hopefully, we can, in the future, I can show you about how these results with this long cohort of patients arise. Thank you very much. And these are my
Great, Eduardo. That's a really helpful overview. It will be great that you'll be using these methods on blood and cerebrospinal fluid from long COVID patients. That's cool. All right, I'll introduce our necker. It's Mario Murakami from Mike VanElzakker's lab at Mass General and Harvard Medical School, and he'll be talking about using ultra-high resolution brain scanning to understand cerebrospinal fluid flow in craniocervical instability patients.
Hi, Amy. Thank you for the introduction. Can you see my slides? It is
yes.
Okay. Okay, thank you for the introduction. Thank you, PolyBio, for the invitation. I'm Mario. I'm Mike's postdoc, Mike VanElzakker postdoc, and I'm going to present my project. So I want to start talking a little bit about the MCFS. As everybody stated, MCFS is this big umbrella of conditions, and the diagnostic criteria is basically symptoms. So it englobes a lot of causes that are different from each other. Most of them are post-chronic inflammatory conditions after an infection, but this specific vulnerability is mostly mechanical. I'm talking about the craniocervical stability, a very interesting cohort of patients, and for some reason they have the same symptoms of those post-infection disorders, brain fog, pain, fatigue, autonomic dysfunction, and this is a gap in the literature. We don't know why this mechanical compression of the brainstem causes symptoms like neuroinflammatory conditions. We have two main hypotheses in the literature. It is the compression itself of the brainstem, because some nuclei, like the nucleus of solitary tract, is located that, and the vagus nerve is located that, and we addressed this issue with other projects that we discussed in the last symposium. But there is another hypothesis, that is this compression also causes the improper clearance of the CSF. Just talking about the craniocervical stability is when there is a loose in the joint between the skull and the spine, the C1 and C2, because of some condition, starts to have a hypermobility, causing this compression of the structures in the neck. And talking more about this second hypothesis about the CSF flow, there are some neurological disorders that involve the accumulation of metabolic waste in the brain. The normal physiology of the neurons and the glia cause metabolites that sometimes accumulates in the brain, causing symptoms. And to wash out all those metabolites, we have a garbage disposal system. It is the cerebrospinal fluid that flows in the perivascular space. We call it the lymphatic system. And inside this red circle, we see a very interesting area. It is the fourth ventricle, and it is the area that we think that is compressed by the CCI. And we believe that some pathological brain disorders, and it is driven by this accumulation of metabolites because of some sort of damage in this garbage disposal system because of the improper clearance of the brain tissue. And we think this is the case of the CCI. So we're gonna evaluate more this garbage disposal system, the CSS flow. Here it is an image of a dynamic, physiologically enhanced CSF flow, and we believe that there are two main physiological pumps: the cardiac arterial pulsation that pumps the blood, and also the respiratory movement that causes the transmission of the pressure. This moves the brainstem and also causes the flow of the CSF. In order, just to put in perspective, I zoom in on this region of interest, the fourth ventricular aqueduct, and we can see that it's really a thin area. So all the CSF exit the brain with the metabolites and waste in this region, in this thin channel. So in the past, researchers developed those sequences that were developed to evaluate the flow of the CSF. You guys can see in black, and this gives the CSF flowing in the perivascular space, and inside the red circles, the four ventricles that are a very thin area, and we think it is compressed by the CCI. This image is called phase contrast enhanced imaging. And here at Martino Center, researchers researchers developed a more advanced and more technological sequences. This is a sequence developed coupling the phase contrast enhanced with fMRI, a 7-test fMRI, where we could also calculate the flow direction and velocity of this CSF flow. And we use those advanced imaging sequences in our study design. This is a pilot study. So we collected 8 subjects, 3 healthy controls and 5 CCIs. We scanned using this ultra high resolution fMRI with 7 Tesla scanners. We collected those multimodal images. I'm going to talk a little more better about them later. And we also collected some physiological signal, the heartbeat and the respiratory movements to check the amplitude of velocity in this channel. We used a very high advanced package of software to analyze those data. So I'm going to explain better this methodology. This is a slice in the brain right below the 4th ventricle, and we can see in this yellow circle a dot that is pulsing, a yellow dot that is blinking. This means flow of CSF in that thin region, in that thin tube in the below the 4th ventricle. And I could manually draw an ROI in this region that is pulsing, that is blinking, and calculate the velocity. I used phase signal and bold signal to extract those blue waves, regress out a neutral region that is the cerebellum in this red, and calculate this green wave, which represents the velocity inside this tube. And we also acquired some physiological data using a platysmograph, so we could calculate the respiratory movements, and I could use the peaks of those respiratory movements, those red dots, and in between them I could see the maximum velocity and the minimum velocity and calculate the amplitude, what means the acceleration or the quality of the pumps of those physiological drivers in the region of interest, that is this thin region below the 4th ventricle. Doing that, we could collect and produce some preliminary results. Comparing healthy controls with CCIs, we saw that there is a decrease of amplitude in CCIs, which means that in this specific region that is blinking, there is a decrease of amplitude, decrease of velocity, which means that is a compromised structure of CSF flow in CCI comparing with healthy controls, which means that maybe this is part of the mechanisms which drives the symptoms, the brain fog, the pain, the fatigue, the automatic dysfunction. We think there is an improper clearance of the CSF because of this compression of this region that is very thin and very delicate. And this is part of the mechanisms that explain why CCI produces symptoms so similar to ME/CFS. So very straightforward takeaways. We think that the CSF flow is disrupted in the CCI patients, and we think that this disruption explains accumulations of metabolites producing neuroinflammation, driving probably those neurological symptoms that those patients suffer. And as future direction, we want to extend this pilot study, increasing the cohorts to improve the statistical power, and also add more different groups to see across different conditions, other conditions that affect ME/CFS, maybe chronic Lyme, maybe long COVID, maybe other ME/CFS subjects. And the good news is that maybe this can be a target for future therapeutics because CSF flow is addressable, and it is so this was one of the projects that we developed here at VanElzakker Lab. I want to say thank you to Mike, our CRCs, Evelyn and Seiji, Martino Center, MGH and Harvard Medical School to provide all the necessary equipment, and also PolyBio for funding this project. And thank you everybody and the subjects and patients that was helping us in this project. This is just one of the few projects that Mike is involved in and I'm working on. And thank you, Annie.
Really cool, Mario. It's cool how you can use the scanner to image the cerebrospinal fluid flow so well. So really cool excited to see the data as it continues. All right, our next speaker is Mark Elia from Invivyd. He is the chairman of the board at Invivyd, and he'll be talking about monoclonal antibodies as targeted therapeutic strategy for long COVID and post-vaccination syndrome.
Okay, great. Well, thanks for having me. I mean, it's a pleasure always to be able to talk to you and talk to the people that are interested in polybio. And I thought I would just point out that Invivyd, maybe a little bit unique in this group, is a monoclonal antibody biopharmaceutical company, publicly traded. We have a product we commercialize today called Pemgarda, which is a monoclonal devoted to the prevention of COVID-19 in immunocompromised persons. But that's really our warm-up act. We're pretty focused on these medicines for a lot of reasons that I suspect your viewership would understand. But I'll just spend a couple minutes talking a little bit more about how you and I met and some of the work we're doing together in the area of long COVID. Going back to the beginning of the SARS-CoV-2 pandemic, it was pretty clear that if you studied any of the host pathogen interaction that defines a coronavirus in a human, you could make a vaccine. You just couldn't make a good one. And I think that was relatively knowable out of the gates, whether it was something we all wanted to accept and understand quickly or maybe grindingly painfully. I think we're reaching those realizations at some rate here in society. And so we started this company around a very, very sophisticated discovery technology that allows us to make antibodies that are a little bit different and a little bit better than what a human bee suite is capable of making. And so we do that. The first one, as I mentioned, is a product that's really not suitable for scale. But one of the interesting things about it is that it has been commercial for now 2 years. And around about a year after we launched this medicine, we began to see reports from the field of various humans who had accessed the medicine, whether it's they were immunocompromised themselves or whether or not they were working with a physician who was willing to write it off label. They were experiencing some treatment relief from long COVID symptoms. And this was, of course, interesting to us. And it was particularly interesting to me because to this day, I still personally suffered from some aspects of long COVID at the level of cardiopulmonary function. And so I'm always hopeful, but deeply skeptical because these are complex syndromes with attractive hypotheses, but no definitive etiology. My own personal concern is that a really robust COVID infection may well be a form of trauma that leaves a form of unmodifiable damage behind. But my hopeful heart is very much informed by some of the work that Amy and others have been doing to articulate, for example, persistent virus or persistent antigenemia in certain compartments that we could access via monoclonal. So in the big picture, Invivyd exists today as a biopharmaceutical company fully integrated from discovery through commercial. In the area of long COVID, we took the time to form what we call the SPEAR group, which is our sort of in-house group of clinical experts and scientific experts. Amy is a member. Her colleague David Putrino at Mount Sinai Core is a member. Akiko Iwasaki and Michael Peluso at UCSF. And we are essentially moving rapidly now to trialing antibodies in long COVID. And I would just linger for a moment to point out that people with an interest in complex viral syndromes or post-acute viral syndromes may know this has been tried before in long COVID. Most recently, the results were less than impressive, but I wouldn't key in on those results too much because the antibodies used were inactive against most recent lineages. And by contrast, what we are aware of is that people have achieved treatment success in the past with Regeneron antibodies. We believe they may be experiencing some symptomatic relief with Invivyd antibodies, and we're thrilled to give it a real shake in the near future with what we expect to be an extraordinary quantum of antiviral punching power in the form of our newest antibody at relatively high dose for a relatively protracted treatment period. So I say all this, and I hope it's of interest to your audience because I think what we're finding in life generally here on Earth is that there's really no such thing as a virus you want, and there's certainly no such thing as a virus you want to live with long term. And yet when clearance is impaired, suddenly you start to realize, well, there's only a limited set of tools you can really use to go after these things. And thankfully, monoclonal antibodies are potentially one of those things. So our mission is broader than even just COVID. It's broader than long COVID, but since monoclonals have demonstrated clinical utility in the prevention of disease, they've demonstrated utility in the treatment of acute infection, and they may be showing us some hints that there is potential for a therapeutic signal in a post-acute setting, this would all be in some ways really exciting and a little bit comforting if this were not some profound and deeply complex sort of anatomic and physiologic mystery, but instead the consequences of protracted exposure to viral replication, inflammation, so on and so forth. I'm looking forward particularly to the results of our upcoming study, not just because I think it would be wonderful to have a treatment option for people, I do think that, but it would also go a long step to deconvoluting the contribution of some of these viruses to human outcomes long term. Because I think one of the issues that I think Amy and PolyBio are very effective at evangelizing out there, and yet which is still so understood even in the infectious disease community, is that the ID doctors are typically attuned to treating an acute infection and then moving on with life as though everything is fine. COVID is yet another place, along with Lyme, along with very much so, of course, the herpes viruses that have all kinds of, relatively well characterized untoward long-term consequences. This is a place where maybe we can finally, pharmaceutically speaking, put the bat on the ball in a convincing way, and in so doing, accomplish a bunch of things at once that I hope pave the way for us to do similar work in a lot of other viral contexts, whether it's Lyme, whether someday if the technology would allow for it, certain herpes viruses like Epstein-Barr and so on and so forth. These are things that humans really shouldn't have to live with, but for the time being, we still do. So again, I know this is probably a lot less visually stimulating than watching really interesting charts and pictures of fun anatomic spaces imaged in some interesting ways, but I'm personally super excited about actually translating some of this basic science into immediate pharmaceutical consequence. And frankly, if it doesn't work, if our study fails to generate a consequence, I think that's a really important learning the field can move on from with some more definitive sense for what is and isn't true. And we can get back to maybe even the better way of preventing long COVID and post-viral syndromes, which would be to prevent those infections in the first place. So I look at this upcoming work as a really, really important next step for the field. And I know all of us at Invivyd are incredibly grateful for the work that PolyBio does. And we're really excited to have Amy's support sort of on a more day-to-day basis, playing around with study design and so on and so forth. So I guess to your audience, look forward to hopefully seeing that shortly. And, depending on how study progress goes, hopefully we're going to be in a position to update the world on some of this, in less than a year from now with a result of some kind. So that's most of what I have to offer here today. But I'm happy, of course, Amy, if you have any questions that you think people would be interested in, or if anyone else does, I'm happy to answer. Or, Memorial Day beckons. I just want to point that out.
Well, we have one more speaker after you, Marcelo. So Marcelo is coming, but I see Francis. We can do 2 minutes of questions. Francis, I see your hand. What are you thinking?
I just want to ask one quick question. I know that you develop broadly neutralizing antibodies, and I'm just what is the platform that you use, since you don't have slides? And just to learn how you're generating these.
Sure.
Because it seems like is that the crux of the technology? I'm just trying to understand. That's all.
Of course. Yeah. No, I appreciate the question. The central trick that we get up to that most other antibody players don't is we don't rely on a mammalian system for our discovery. Okay. So classically in the pharma industry, one would either go hunting in serum for an interesting IgG candidate, or one would inoculate a mouse, humanized mouse. And our view was always that in infectious disease, when you do that, you're playing with fire, because you're, of course, selecting your pharmacophore straight out of the evolutionary pressure pool that moves these viruses around. So as I'm fond of pointing out, any idiot in the pharmaceutical industry can make a COVID antibody. After all of us do, personally. We make them all the time. The challenge is, of course, if you're limited by the peculiar, let's call it habits of the mammalian bee suite, you are biasing your drug discovery toward evolutionary pressure, which you don't want. So at Invivyd, we use a yeast-based library. Essentially, you can think of it as combinatorial chemistry in a biologic context. So if you had any kind of starting point whatsoever, whether it's a blank mAb, a mAb directed towards a different viral target, a self-target, or you could also start with convalescent sera and just look for a starting point. What we do is we essentially use structural principles to walk our antibodies into epitopes and geometries that mammals don't typically like to drug, in effect. So our targets are always going to be highly conserved regions that are essentially cryptic or immunorecessive. And so if you know your SARS-CoV-2 spike protein, ours would be sort of class 1-4 antibodies, which are sort of the weirdlings, and they're very uncommonly made in nature. And so when we do that, we hold ourselves to a relatively high standard. We would always like to impose very potent neutralization against any lineage that has ever had clinical penetrance. Because to our mind, when we're doing that, we're integrating the sum total of the evolutionary journey that the host and the pathogen have undergone over these last 6 years. And we are availing ourselves of these kind of like quadrillions and quadrillions of variant explorees juxtaposed against human and mammalian serology to essentially refine. Like, you can think of our antibodies as having been honed over time to try to access that target space. And, look, to your point, it actually relies on a completely different discovery paradigm than most pharmaceutical companies enjoy. And it was built for this purpose.
Cool.
It is cool. It's fun.
All right, very cool, Mark.
Yeah, it'd be great to learn more about exactly what the technology is at some point.
Well, we're a little cagey about that because it wouldn't be so fun to have you go do it yourself, and then we'd be sad, right? But I agree. Anytime you want to learn more, happy to set up time.
That'd be great.
Cool. All right, Mark, I could pick your brain forever, but let's give Marcelo the last 10 minutes of this symposium. So thanks so much, Mark. And we're just so excited for the trial of VYD2311. Researchers are, patients are, you name it. So
I'm excited.
So yeah,
I can't wait.
Everyone.
All right.
So all right.
Thanks so much, guys.
All right.
Bye-bye.
See ya. Bye. Okay. And then our last speaker is Marcelo Freire. He is in the Division of Infectious Disease at the J. Craig Venter Institute, and he will be talking about proteomics revealing immune markers in a long COVID vagus nerve stimulation clinical trial.
Thank you. Can you see my trial? My trial? No, my slide.
I can't, Marcelo. I can see your
Well,
slide.
I love what Mark was talking about. It gave me a little nostalgia when I was Cold Spring Harbor, work with antibodies from chicken bone marrow and yeast libraries. So fun stuff. So it's great to hear the advancements here. So today I chose to speak about this proteomics study because it highlights one facet of my lab. We are looking obviously in long COVID subjects, which we know by now that it's multi-system and has multiple medical paradigms how to treat it. And we are very interested in immune response. But more importantly, what I want to show you today, how are we measuring in our lab and how different this is, and how can we think of not just one molecule at a time, but an entire network that impacts the human body, whether in disease or healthy. Okay, that's in the slides. Oh, what happened to my slides? Okay, is that what's gonna happen? Hold on. 1 second. Okay. Okay, so the paradigm shift in biology is not just thinking of one specific molecule, but in fact, multiple molecules. Here's just an example of, let's say, proteins from the virus or host response or post-translation modification proteins, but they might unify in certain networks through their interactions that could be biological, mathematical, or involvement between cell-cell interaction or some chemical mechanism. So that defines the complexity that we have today when we think of inflammation. And inflammation is present in acute COVID, but also in long COVID. And the question is, why when we suppress inflammation, the patient is not healed when we suppress with current medication. So there's several challenges here. How do we understand? How do we recover these patients? In our lab, we are very interested in the salivary proteome. So saliva is a fluid that's non-invasive and has a capability to provide a lot of information. So here's acute COVID era, and we determined the proteomic signatures of acute, and it was so cool to watch the neutrophil talks and all that, because in this study, we had shown that in salivary proteomes, we had found that neutrophil NETs were highly prevalent, but only in moderate and severe cases, but not in mild. By that time, we already knew that a chronic condition would come up, obviously, from all the other viral diseases that induce chronic conditions. But the question is, are those things persisting through a long COVID? And we did that in acute in terms of proteomics, but also in terms of oral and host microbiome. And we learned that saliva, it's highly predictable. Here you see saliva in blue here and the combination in black, and the blood this is an AI model for predicting the patients and predicting severity, and here's the blood. So we could appreciate how saliva or a combination of microbial and immune markers in fact can predict the severity of the disease and the yes or no of the condition. So we have a database called salivary proteome, and our plan is to redefine how we think of these interrogations that we have today, so we can look into more detail what is being impacted. So for example, health and disease is one, but then the other is a clinical trial, multiple time points. We are talking about monoclonal antibodies here. And how are we evaluating these patients? And thinking of saliva as a primary trial endpoint, and that will be a great goal for me to get there, and making sure that we use reliable messengers to give us this information. So salivary proteome contains 3,209 proteins at this moment. We have matched blood and saliva, and we're looking into also the peptidomics, which goes a little further for how we investigate saliva peptides. We have published the methodology. We use mass spec, and we dissect the proteins into peptides and then predict which proteins are present there in that saliva. We can either do that with the microbiome or the host immune response, or both. And that can allow us to have some prediction of health and disease, and the reference of this step-by-step of how we prepare our samples. We optimize the protocols for over 10 years to get to a point where every study that we do the same protocol in the lab, allowing us to have the highest amount of proteins per sample. David Putrino at Mount Sinai has led this clinical trial of vagus nerve stimulation, and there's a bidirectional relationship between the brain and the body. And this neuromodulation therapy, it's thought to improve some anti-inflammatory reflex or stimulate nerves that could then activate systems in the body to improve patients. So these are 50 subjects, and there are two arms, active treatment and placebo. And saliva samples were tracked through 5 visits. The 5th visit was 12 weeks, so before and after. And we looked between treatment active versus sham. Obviously, we want to see if there is a placebo effect, but also within treatment, visit 5 versus visit 1, so you can understand the specific response from the same subject from the baseline, measuring that protein expression, directly causing a stimulation versus placebo group it's in the first visit, and then the visit 5 in which they did not receive the actual vagus nerve stimulation. So here is actually the core of the results as I summarize for you today. It's an inflammatory inversion. So you have here the sham group on the left, visit 5 versus visit 1. So what you have here is an upregulation of the sham group, the placebo group, with 139 proteins. And it shows a down expression of 32 proteins. Out of the thousands of proteins that we found, those are significantly upregulated when the patient is under the sham cohort. When the patient's under the active group, which they receive the vagus nerve stimulation, and in that case, then we have a decrease, almost twofold, of the upregulation and an increase of downregulation proteins. Now, the work that we're trying to do in our lab is investigating the networks that are dominating the systems here. One, obviously, is inflammatory immune response, but we're also interested in adaptive immune responses and some other T cell signaling. And that we are using this pipeline from the salivary-proteome.org, which is a database that was initially funded by the NIH and led by my lab and my group, where we are dissecting, comparing to other data sets. So just as an example for the data that I showed you today, from these sham results, we have increase of upregulation of serum amyloid A4 protein. And the clinical significance of some proteins like these present in saliva is that elevated serum amyloid A4 protein correspond to increased relationship in rheumatoid arthritis, oncology, arteriosclerosis. So proteins like this are highly clinically significant and persists after sham control. Meanwhile, with the active response, we have top-down regulated proteins, including several inflammatory molecules, such as sorry, I couldn't, somehow my screen disappeared for me, such as the gamma cyanonuclein protein, which has a dual role in cancer and neurodegeneration. So this protein, in fact, if persisted, upregulated through the 12 weeks, this protein is highly regulating neurodegenerative diseases in brain, and it's linked to several prion-like manner structures, and it's involved in other cancers throughout the body, colorectal cancer, breast cancer. So proteins like this were completely downregulated in this vagal nerve study in the activated group. So our hypothesis at this moment is supported. This is the first time we do a clinical trial with the salivary proteome, in which we are investigating not just health and disease, but in fact, response to therapeutics. So the vagus nerve stimulation here is altered by the expression of salivary protein associated with inflammation in long COVIDs. And we hope that these promising results can help, future trials. We hope that complex trials with drug trials can allow us to monitor saliva and actually investigate these networks more carefully and understand what is the baseline of that patient, and after multiple measurements that you cannot do in bone marrow or blood, what is it changing and when is it changing? So that kinetics is also very important. So I want to say thank you to the people that directly work in this project and to the Mount Sinai team, and of course to PolyBio and Prebys Foundation, NIH, and my team at JCVI. So thank you.
Great, Marcelo. It's good to think that there are some salivary biomarkers we can track in trials. So it's very good proof of concepts. All right, cool. All right, well, that is the end of the symposium. Everyone did a great job. It was truly interesting to hear about all the data. We're so grateful for all the patients who have participated in all of these studies. It's incredible, and you make everything possible. And we're also so thankful for donors to PolyBio that support this work and make it happen. So thank you to everyone. Take care, and we'll see you in 6 months at the next PolyBio Symposium. Bye.